to this meeting of the Independent Citizens Oversight Committee, the CIRM Board, and the Application Review Subcommittee. Here we are in Sacramento, California. And I'm going to ask that we call the meeting to order, and we'll start with the Pledge of Allegiance if Mr. Scott Tosher will do the honors and lead us. Thank you.
Sure. The flag is by the podium. Please stand if you are able. I pledge allegiance to the flag
of the
United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Thank you, Scott. Would you please continue and call the roll?
Sure. Iyad Al-Masri, Kim Barrett,
Present.
Dan Bernal, George Blumenthal, Maria Bonneville,
Present.
John Carruthers,
Present.
Monica Carson.
Present.
Sorry, Monica, are you muted?
Present.
Thank you very much. Marguerite Casillas,
Present.
Judy Chow,
Present.
Leandra Clark Harvey,
Present.
Shannon Dahl,
Present.
Anne-Marie Deliege,
Present.
Isabel Daron.
Here.
Thank you. Mark Fisher Colbury,
Here.
Elena Flowers.
Present.
Oh, sorry, that was so quiet. Judy Gasson,
Here.
Jeff Golden,
Present.
Vito Imbasciani,
Present.
Rich Lahara,
Present.
Pat Levitt,
Present.
Hala Madanath,
Present.
Linda Malkus, Carolyn Meltzer, Chris Miaskowski,
Present.
Adriana Padilla,
Here.
Joe Panetta, Joyce Sacchi, Marv Southard,
Present.
Shauna Stark, Carol Watson,
Here.
Yael White, Kevin Zhu,
Here.
Keith Yamamoto.
Here.
Great, thank you very much. Mr. Chair, we have a quorum.
Quorum. Thank you, Scott. The first order of business is to consider the consent agenda, which is a little longer than usual today. It consists of the minutes from the December 11th and the January 29th meeting of the ICOC, appointments and reappointments of the scientific members to the Grants Working Group. And for those, I presume everyone has looked at this. There's one new appointment of a Dr. Mark Fitzgerald, MD, PhD, from the Perelman School of Medicine at the University of Pennsylvania. There are 6 reappointments of scientists. And there's also a very interesting graph appended to this report that quantifies the representation of women scientific members on the Grants Working Group. And you'll see that the attendance and participation of women scientists on the GWG has nearly doubled over the time span of 2020 to 2025, reaching a total of 50%. There are other items on the consent agenda. One is a recommendation from the Governance Subcommittee regarding the process of evaluating the chair, the vice chair, and the president and CEO for this upcoming year. And finally, there are some amendments to the funding mechanism for CIRM related to the Education One or EDUC One program. So the way the consent agenda works, if anyone wants to discuss anything that I just mentioned, it needs to be abstracted by you by voice. And then we can close out the rest of the agenda and then proceed to a discussion of the item that you would like. Do I hear any abstractions to the consent agenda items 3, 4, 5, and 6? Not in the room, and perhaps anyone online? No. Okay. Can I have a motion to close the consent agenda, please? Okay.
Leandra Clark Harvey.
Leandra has moved.
Second.
Maria seconded. Scott, you can
Board comment? Public comment?
public.
All those in the room in favor say aye.
Aye.
Opposed? Abstentions? And I'll poll the members on the Zoom. Monica Carson,
Aye.
Judy Chow,
Aye.
Isabel Derone,
Aye.
Mark Fisher Colbury,
Aye.
Judy Gasson,
Yes.
Jeff Golden,
Aye.
Rich Lahara,
Aye.
Pat Levitt,
Yes.
Hala Martinat,
Aye.
Chris Miaskowski,
Yes.
Marv Southern. Okay. Carol Watson
Yes.
and Kevin Zhu.
Aye.
Oh, and Keith Yamamoto.
Yes.
Great. Thank you. That motion carries.
Thank you. Thank you, Scott. Board member Shannon Dahl has a comment on the consent agenda.
Yeah, I had a comment about the grant working group committee. I think we had, just looking at the new appointment and the reappointments and looking back at the list, I'm really impressed by the group that we have reviewing our grants. And there's such a critical piece of the organization and moving the programs forward. I'm excited about the new appointments and the renewals. As we look at our portfolio at large and the way in which we're advancing more programs into later stage, I think it would be helpful to just take a step back and say, do we need to add additional later stage expertise to the group to ensure that every time we're going from phase 1 to BLA, that we have enough reviewers in the room with that perspective. I see we have some on our list, which is great, but they're smaller in number. So I would just ask that the team take that into consideration. Thank you.
Thank you, Shannon. We're going to move now to agenda item number 7, the consideration of applications that have been submitted to CIRM in response to our Discovery Program announcements, or DISC 4. And this presentation is going to be led by our Vice President for Portfolio Development and Review, Dr. Gil Sembrano. Thank you, Gil.
Everyone. So yes, I'm going to present to you the recommendations from the Grants Working Group as it relates to the DISC 4 program. So let's get started with that. We always begin with our mission to accelerate world-class science, to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. And this is important in setting the stage for what we do and why we do it. Of course, to help us achieve that mission, we have a strategy with very specific goals that we want to achieve. As it relates to discovery programs, those discovery programs are set up to support comprehensive discovery research, and they also encourage collaborative, multidisciplinary approaches where possible. And in particular, the DISC 4 awards that are based on the original REMIND program are very much intended to support cross-disciplinary, very collaborative and integrated studies across a range of technologies and approaches to address bottlenecks and other knowledge gaps in human disease. So for the DISC 4 awards, anybody who applies, we expect that they're going to achieve one or more of the 3 outcomes listed, which is to discover novel mechanistic insights or advance understanding of human disease pathobiology, extend understanding of disease mechanisms across affected human populations, and or identify and validate novel therapeutic strategies, targets, and biomarkers. Now, the way the DISC 4 program was designed, it was designed using preference topics for each cycle. And so the a board approved preference topic for this particular cycle was neurological diseases. And so that includes conditions and disorders of the brain, spinal cord, and peripheral nervous system. All right, so a little more about the program structure itself. The duration of the award is 4 years. It offers 13 or up to 13 million and an additional million with matching funds from the applicant. The applicant team itself includes the principal investigator, along with a minimum of 4 co-investigators who work collaboratively to achieve hopefully something that synergizes and can achieve more than either one alone. So the allocation that we have for this particular cycle is 84 million total, which supports roughly up to 6 awards. And so let me tell you a little bit about then the review process that's utilized for the DIS4 applications. So this utilized a pre-submission process at the beginning, at the onset. So these were short 3-page pre-submissions that were reviewed by the CIRM staff, and the set of preferences that you've seen before were applied here. We had 138 pre-submissions, and of those, 24 were invited to complete a full application. Those full applications were assessed for eligibility. All were eligible and went on to the merit review stage. And we are going to then share with you the recommendations from the grants working group that evaluated those. So this is just a reminder of the composition of the grants working group panel. It includes scientific members who evaluate the applications based on broad subject area or methods expertise, and they are the ones who enter the scientific score for every application. And we also have what we call grants working group board members. So those are ICOC patient advocate members who sit on the grants working group. They offer their patient perspective on the significance and potential impact of projects, and they also provide oversight on the process itself. They do not enter scores. We also have visiting specialists. Those are scientific members that we invite to fill knowledge gaps when necessary for a particular set of applications, or even maybe one application at a time. And they provide their insight to the panel on applications. The scoring system that we use is on a scale of 1 to 100. A median score of 85 to 100 means that it has exceptional merit and warrants funding. A median score of 1 to 84 is not recommended for funding. The scoring that is utilized is holistic and based upon all facets of the expert reviews, and we do encourage reviewers to use the full range. All right, and the criteria that are the basis for the score are highlighted here. These are just the high-level elements of the 5 main criteria, which include the significance and potential impact of the project, innovation relative to the current state of research for the project, the scientific rationale, the project plan and design, and the applicant's consideration of impact across affected populations. This table summarizes the recommendations from the grants working group as it relates to this set of applications. So for those 24 applications that were evaluated, 8 of them were recommended by the working group, 16 were not. The total budget requests for the 8 applications totals 108.5 million. And as noted earlier, the funds available for the cycle is 84 million. So this exceeds the amount that we have available to support all the projects. So the scoring and the review sometimes results in a minority report, and that happens when any application that's not recommended for funding by the grants working group but has had 35% or more of the members score in the funding range. Those include a minority report, which is basically a paragraph that summarizes the minority position, and we usually check in with those reviewers to ensure it mirrors their perspective. In this particular cycle, there were no applications that qualified for a minority report. All right, this table summarizes the 8 applications that were recommended by the grants working group. It shows them in rank order in terms of the median score, which ranges from 85 to 90, along with the budget request. The other thing to make note of is you see the Y and the N in the second to last and third to last columns. So the Y means yes, those are the number of grants working group members that scored 85 for above for that application, and the N means no, which those are the ones that scored below 85 for that particular application. And so you'll notice that in particular for the last two, which the CIRM team is recommending not to fund in order to accommodate the budget, had a difference of 8 to 6. So 8 members scoring 85 or above, while 6 scored below 85. So it's just something to take note as I describe these. So this CIRM team recommendation is just to give you a little bit more about the rationale. We looked at all the top applications, and in particular, those that had a score of 85. There were 3 applications that I showed you on that list that had a score of 85. And looking at those, the CIRM team is recommending that 2 of those applications not be funded, which is DISC 419196 and DISC 419226. And this is, solely due to the fact that we don't have enough budget to cover. I want to emphasize the fact that all have merit. So we're not suggesting that any of these do not have scientific merit as indicated by the working group, but we had to develop a rationale for why we would recommend the group we did. So the DISC 419391 did have a greater consensus on merit. So that's the 11 members scoring 85 or above versus 3 that scored below, while the other 2 had 8 to 6 split. So a little bit more about the 2 applications that are not recommended. So this is 19196, or not recommended by the CIRM team. They are recommended by Grants Working Group, just to be clear. We're not recommending this one largely because of the score. It is the application itself, in terms of the topic, is focused on neurodegenerative diseases, particularly ALS and frontotemporal dementia, and aims to understand how RNA localization relates to the disease state. Reviewers noted an excellent team, innovative approaches, impact of the data set to be produced, but there was a risk that was noted that while the mislocalization of the RNA is a common observation in ALS, the reviewers did note this several times, and you can see it in the summary, about the uncertainty of whether this actually plays a causal role in ALS. And so the risk is that the effort expended here may be limited if it turns out that there is no causal relationship. Also, we already mentioned the score of the Grants Working Group consensus being 8 to 6. Is that oh, there we go. All right, and so then the next application, 19226, is the other one that we're not recommending for funding. So this application is focused on understanding cytokine responses in the brain and microglial mechanisms in the context of Alzheimer's disease. Again, reviewers noted an excellent team, innovative approaches, and the potential creation of a high-value data set. For some reviewers, the enthusiasm was offset by some concerns related to the lack of power calculations and other technical elements. There was also some concern about the fact that the projects were a little disjointed, didn't seem to relate clearly to one another as to why they were put together as proposed. And again, this also had a GWG group vote that was eight members voting 85 or above and 6 that did not. All right, so with this, then we can transition to the Excel spreadsheet, the table that shows you all of the applications for discussion. And Chairman Imbasciani, I'll just turn it back to you for discussion.
Gil, Dr. Sombrano, thank you so much for that presentation and the explanation of the deliberations you went through. If I can have the temerity to summarize Dr. Sombrano's presentation, we have a lot of applications, some of which were not recommended for funding by the GWG. The Grants Working Group did recommend 8 for funding, but that budgetary request would have exceeded the $84 million we have available. In fact, it totaled $108 million. And as it happens, there is a 3-way tie at the bottom. Gil very eloquently described the differences between those 3 and what guided the team to recommend one over the other. Therefore, even though the Grants Working Group recommended 8 for funding, we can only fund 6. And it's going to be incumbent upon the board to choose that. So to simplify this from a motion point of view, I think it would be easiest if we had a motion not to fund those from number 9 down. That is, not to fund the ones that were not recommended by the Grants Working Group. He's showing that now, all the ones that fall below position 8. Scott, do you want to clarify?
So moved.
Just from a process standpoint, because the program is what we call oversubscribed, at the moment, for those voting members of the Application Review Subcommittee who have a conflict with any application, it's considered to have a conflict with all of them for the purposes of making a motion and seconding a motion and for the discussion. When I call your name during the roll call vote, your answer will be aye or nay with respect to the motion, and then add, except for those applications with which I am in conflict, some version of that. So at the moment, the voting members of the ARS with a conflict and thus should not participate in the making of the motion or discussion are members Dahl, Bonneville, Durone, Deliege, Flowers, Fisher-Colbury, Miaskowski, and Watson. Beg your pardon? No, that means there is a conflict, so please refrain from.
I just want to, this is Jeff Golden. I think I have a conflict with one of them as well. $19444.
Yes, 19350, you have a conflict.
Thank you, Scott. Okay, so Chair would like to entertain a motion not to fund those applications not recommended for funding by the Grants Working Group.
Moved.
John?
I will second Dr. Barrett's motion.
Sorry, you're not a voting member of the.
It has to be coming from a member of the Application Review Subcommittee. That's quite all right. That's right.
Second.
Correct.
I'll make that motion.
Okay, we have a
Okay.
motion and it can be seconded by
I will second Marv Souther.
Marv. Thank you. Any discussion on this early motion not to fund those grants not recommended by the GWG from board members? Board members online? Or anyone, any member of the public like to speak to this? There's a gentleman in the audience that might have.
All right, so we're going to now open the floor for public comment. If you're joining in person, please approach the podium. Oh, the microphone.
It's this microphone over here, sir.
If you'd like, you may state your name for the record and please keep your remarks within 3 minutes.
I'm actually discussing about proposal 19196. Should I wait until later?
Correct. It's not part of this motion.
Okay, yeah, thank you.
All right, any participants on the phone that would like to talk about these applications, please press *9 to raise your hand. All right, we have one member of the public, number ending in 3564. If you'd press *6 to unmute yourself, if you'd state your name for the record, and please keep your comments within 3 minutes.
My name is Laura Miller, and I am commenting on 19391. I am a person living with ALS. ALS being that disease that slowly takes away a person's ability to move, speak, and eventually breathe. For most of us, about 90% is.
Okay, ma'am,
Did you get her name?
we're not on that application yet. We're gonna save your comments for when we get to that application number.
Thanks, Lana, for that clarification. Yeah, to the speaker, we haven't, the application you would like to speak to has not been put on the table for a motion, but don't go away. Okay, if there's no other comment, just verifying with board support? No. No other comments? We can proceed to a roll call vote on this motion.
Yes,
Maria
except,
Bonneville.
yes, except for those with which I have a conflict.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl.
Yes, except for those with which I have a conflict.
Thank you. Anne Marie Deliege,
Yes, except for those with which I have a conflict.
Isabel Derone,
Yes, except for those with which I have a conflict.
Mark Fisher Colbury,
Yes, except for those with which I have a conflict.
Elena Flowers,
Yes, except for those with which I have a conflict.
Vito Bassiani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes, except for those with which I have a conflict.
Adriana Padillo,
Yes.
Marv Southard,
Yes.
Carol Watson,
Yes, except for those with which I have conflict.
Kevin Zhu.
Yes.
Thank you. The motion carries.
Thank you, Scott. Okay, now we are left with 8 applications that have been recommended for funding by the GWG, but as Dr. Sembrano has explained, our budget will only support 6 of those. We are oversubscribed. So we can do this in several ways. The simplest way would be if someone moved to accept the recommendation of the CIRM team to fund the 6 that have been recommended, and not to fund the 2, although they tied in that low score.
So moved.
We have Marguerite Casillas has moved.
I'll second the motion.
That's a second there.
Again, a voting member without a conflict. So those with a conflict at the moment are Dahl, Derone, Fisher Colbury, Flowers, Miaskowski, and Watson. So the remaining voting members
Are eligible.
are eligible.
I second.
Thank you.
Thank you. All right, so any discussion on the motion? From board members, Anne Marie
Yeah,
Deliege.
just, Gil, thank you again for that great presentation. We realize that it's really a challenge when we come to an over budget sort of scoring. We received a number of letters about these applications in general, particularly these 2. Can you summarize some of the concern expressed in the letters and how we would address them? I know it's a general question, but maybe you've read the letters, I assume. And,
Yeah, so,
yeah.
so we did read the letters. I mean, I think they each have.
Hear you. Could you maybe speak up a little bit? Can we
Okay,
hear more about-
so I will,
Thank you.
sorry. Yeah, maybe. Sorry about that. So yeah, so we did read the letters.
Sure.
And so I think they each, advocate for their project, highlighting elements of, in some cases, the scientific elements that the grants working group highlighted and how they've addressed some of those concerns. Of course, we always note the fact that, unless the grants working group actually sees them, it's difficult to know what they would think of these responses. So we don't evaluate them from a scientific perspective to confirm that, they would have changed the outcome. But, they are noted there. Some of them highlight elements of the fact that, these are an important area. I mean, two of them in particular that are on the line relate to ALS. They both happen to come from the same institution, actually. But, they are both important projects, both highlighted as meritorious by the grants working group. So I think the difficulty is also that even with these letters, it becomes difficult to determine, where one draws the line and how one recommends them. So, I mean, that at least is what I can offer at the moment.
Thanks, Gil. We have comment, Board Member Casillas?
Yeah, in one of the letters, about those two applications that came from the same institution, they proposed like, well, take a little bit of money out from this one and put it to that one. I mean, is there even a mechanism for that?
It's something that we do not recommend. We do not want to, take funds from other projects in order to allow, or even if there are funds remaining. So you would have about 3.5 million remaining out of the 84 if we fund the top 6, is not sufficient to cover the scope and intent of the project. These projects were reviewed with the expectation that if they were to be funded, they would be funded in full and would be accomplishing the goals that they set out. I think providing any less would really hamper significantly their ability to do that.
And there's no reason those two applications that aren't being recommended couldn't reapply?
Right, so we are going to have additional cycles of Disc 4, noting though that, there are preference topics for each, but we are not preventing applicants from coming into any of these cycles, so they can reapply. We have actually in the batch one application that is in the top 6 that was an application that fell in the same situation under the Remind 4 program that is now being funded, assuming this goes forward for that program.
One question, I think I know the answer, but still I will ask. The envelope for this cycle has been fixed. Is there some flexibility there? Is that the budget for this year with an additional budget for the next cycle review? How do we consider that?
So the budget for the next cycle has not been set, so that'll come in June. So this is for one cycle per year. So the cycle is, or the budget for this is fixed.
I don't see any hands from board members online. We can go to public comment. I do see a telephone call and we have a gentleman in the room. Yes. Sir, if you'd start again, tell us your name again and thank you.
No, it's a tied for the top 6. We are number 7.
To fund the top 6. Oh, sorry, I only heard to fund the top 6, so not fund the other 2.
We need to use a microphone to make a record, please.
Thank you. It's to accept the the motion is to accept the recommendation of the CIRM team on all 8. So he is in order to speak.
Okay, can I start? Okay. All right, good morning to members of the subcommittee. I'm Stanley Chi from Stanford, representing 19196. So ARS is a devastating crisis for California. Despite decades of efforts, the survival rate remained unchanged. We are here today because our team is a first-time partnership between Stanford and UCSD, and we are ready to bring in a new therapeutic solution to your portfolio named Spatial RNA Medicine. The grants working group raised a critical question. Is RNA localization a cause or a consequence of ARS? We now have the answer. In recent 2025 data, we proved that RNA being in the wrong location kills neurons. And crucially, we also shown that using our technology to move one key RNA to the neurites, we can trigger the axonal regrowth by 80%. We are now able to rewrite the RNA geography to save the neuron. We know the subcommittee is facing a difficult time today, and with remaining funds in the allocation, I want to mention that our team has never received CIRM funding. While our proposal is comprehensive, we are so committed to the mission that we have developed a narrow scope, flexible budget model. We are prepared to work immediately with CIRM to fit our scope into the available funds. We believe that it's important to bring this unique first-class platform into the community now, rather than waiting for another year of waiting, because for patients, waiting is not an option. If you look at the current CIRM portfolio, you'll find many work in gene editing and cell therapy, but you'll find 0 projects addressing spatial RNA medicine. By funding 19196, you are not just funding another study, you are filling a massive gap in California's portfolio with a technology that doesn't exist elsewhere. It's not just a tool, it's a medicine to stop ALS. Finally, I want to share why this project is so urgent. We received direct and moving requests from patients who are asking to join our team, not as subjects, but as researchers, to find a cure for themselves. They aren't just looking for a treatment, they are looking for a way to fight back. For these families, the funding is not just a budget line, but it's a literal fuel for their hope and their belief that a breakthrough is finally within reach. Let's not leave the hope to chance. So I hope the committee will consider, let's break this tie in favor of innovation and let help us make the spatial RNA medicine a reality for California families.
Thank you for your remarks.
All right, we have 2 people on the phone. It's Laura Miller ending in 3564. Let's try this again. If you press *6, and you can have 3 minutes for your public comment. Ms. Miller, are you available? Oh, there you go.
Yes, thank you. Again, my name is Laura Miller. I'm commenting on 19196. And I am living with ALS. ALS is a devastating disease, and despite decades of research, we still don't understand enough about how this disease works. Consequently, there have been no treatments developed to stop its progression. This project explores something scientists are only beginning to understand, that in ALS, molecules inside nerve cells may be going to the wrong places. If researchers could learn how to correct that process, it could open an entirely new way to treat neurodegenerative diseases. Breakthroughs in ALS research don't just help people with ALS, they may unlock the biology of many neurodegenerative diseases. So, as someone living with this disease, I strongly support funding innovative research like this. Thank you.
Thank you very much. All right, we have one more public comment. Well, I have two more. Last four digits is 6378. If you press *6 and state your name if you would like and start your 3 minutes.
Yeah, this is Michael Snyder. I'm the contact PI of 19391, the one that was recommended for funding. And I'd just like to say, first of all, that we're obviously pleased that the reviewers and CIRM staff liked our application and overwhelmingly supported it, 11-3 in favor. We do believe this research is highly impactful, not just for the science, but for its translation potential. And we have a very strong and experienced team that we believe can pull this off. The genetic basis, as you just heard, of sporadic ALS is very poorly understood. And we have new innovative AI methods that people really haven't done before. And they do find new genes and potential ALS targets. And we've already found many genes with these kinds of approaches. And so we think these are going to be very, very powerful. In fact, we've even found one potential target using this kind of approach. So we are confident that it can work and be very impactful. The reviewers did raise some concerns. It seemed like the biggest one was whether we would have access to samples, and we obviously do. We have a member of our team. He's part of Target ALS, and even in his own biobank, he has many potential tissues that we can use. So I think their major concern isn't really a concern. This gets to the technical issues, and other technical issues are pretty easily addressable. So the bottom line is we do think our project's hugely innovative and can easily translate into things that will be highly impactful for ALS patients. And so we're, again, quite confident we can pull this off and have a great impact on this very devastating disease.
Thank you, Dr. Snyder.
All right, then we have a public comment ending in 4883. If you'd press *6 to unmute yourself and state your name for the record. You have 3 minutes.
Hello, everyone. My name is Sarah Doherty, and I'm the Senior Director of Research with the ALS Network. I'm speaking in support of 2 applications today, which I realize now might not help with your tiebreaker, but it's really important information. So regarding item 19391, this proposal addresses an unmet need and one of the most critical challenges in ALS research, understanding the genetic and molecular drivers of sporadic ALS, which we mentioned represents the vast majority of ALS cases. By integrating machine learning, large-scale genetics, neuropathology, and stem cell-based functional screening, this project takes a highly innovative and comprehensive approach to identify new therapeutic targets. What makes this approach especially powerful is that it moves beyond observation to true functional discovery, linking genetic findings directly to disease mechanisms. The ultimate goal is to generate new genetic targets for sporadic ALS, an essential step towards developing therapies that can benefit the broader ALS population. This exceptional multidisciplinary team representing leading California institutions has the potential to build a strong pipeline of targets that could meaningfully impact patients. For the ALS community, this kind of discovery-driven research is essential to unlocking new treatments and advancing the field toward real progress. And then, perhaps unconventionally, in support of 19196, also an ALS project, this proposal represents a bold, innovative approach to tackling ALS. In this instance, rather than focusing on genes or protein, as Dr. Q mentioned, this work explores how the spatial organization of RNA within cells contributes to disease, and so critically, how to correct it. This is a fundamentally new way of thinking about neurodegeneration. This work opens the door to an entirely new therapeutic strategy space that goes beyond current approaches. Importantly, these mechanisms are shared across multiple neurodegenerative diseases, giving broad impact. This is exceptional team is grounded in a highly collaborative environment. The project has both scientific rigor and innovative edge that is needed to drive meaningful progress. From a funder's perspective, this kind of forward-thinking research is what is needed to move from incremental advances toward truly transformative therapies. I thank you for your time and truly request that you consider both of these proposals.
Thank you to the speaker. I see another call coming in, Lana.
We do. Okay, the phone number ending in 7366. If you would press *6 to unmute yourself, and if you'd like, state your name for the record, and you have 3 minutes. I see that you're unmuted. You can start speaking.
Oh, yes. Hi, thank you. This is Justin Ichida. I'm one of the co-Is for 19391. I'm from USC. I've been working in the ALS field for over a decade now, and I think what we're beginning to see is that this disease is reversible if you identify the right target. For example, the ASO targeting SOD1 has really we've been seeing that it can halt the disease and in some cases improve improve the motor function of patients with SOD1 ALS. I think the challenge is that most of the patients with ALS are sporadic, and we don't yet know what the right genetic targets are for their disease due to the complexity. And so what we've seen with some of the new techniques with AI and multi-omic analysis is that we can actually now use those to better harness the genetic information and sequencing that we have to find these new genetic targets for sporadic ALS. We've had some success already with one of them, and we are now looking to do more. And I think this grant would be pivotal in enabling us to do that and come up with a series of strong genetic targets for sporadic ALS where there's a huge unmet need. So thanks very much for your consideration.
Thank you. Lana, I think I do not see any more hands raised.
You are correct.
Okay. Any board member want to make any final comments before we proceed to a vote?
Yeah, Gil, I'm sorry.
Yes.
Gil, I'm trying to see how we can offer a creative solution for the 2 that are proposed not to be funded right now because of the tie. Is there only one cycle review for DISC-4 every fiscal year, or are there more than that? And does it mean that if these applicants want to reapply, this will be in 1 year with an unknown budget and a known scope of proposals? Or is there something that can be done about that?
Right. So there is one cycle per year. And so the scope of the next one, maybe Rosa can speak to it because that's part of the proposal for a little later.
Can you hear me?
I'm sorry.
So the next application will be open in April for these 4. And as we will be proposing, of course, is to the prerogative of the board, but as we will be proposing today, Dr. Shepard will be presenting the focus recommendation, and it's actually a much larger systems scope that we will discuss later. So it would include these therapeutic areas. Most likely they could be able to reapply. And we have one a year that is an $84 million, but we also have these 5, which is $50 million investment. So in total, we are investing around $135 million in discovery every year through those 2, and they're like smaller awards can come through these 5, which is also open.
So I think what we're saying is that these 2 applicants could reapply in April, although the review process will take a year. That's right. And if they were granted if they were accepted, they would receive the money in.
When is the ARS scheduled for this
I
one?
think it would come back a year from now, so it would be next March ultimately to the board.
Okay.
It's a long process because there are the pre-submission as well. So we have like the pre-submission, then they are selected, then they are invited, then the full team puts together the proposal, then that's reviewed and yeah.
Okay. Let me clarify this. A positive vote, a yes vote on this motion will be a vote to fund Disc 4 19200, 19271, 19334, 19371, 19291, and 19391. Okay?
And to
And
non-fund
not fund,
19196 and 19226.
that's correct.
So for the following members, please vote in the same manner that I described earlier, yes or no, except for those applications with which you are in conflict. Those members are Dahl, Derone, Fisher-Colbury, Flowers, Miaskowski, and Watson. Maria Bonneville,
Yes.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes, except for those with which I have a conflict.
and Marie Deliege,
Yes.
Isabel Derone,
Yes, except for those with which I am in conflict.
Mark Fisher-Colbury,
Yes, except for those with which I have a conflict.
Elena Flowers,
Yes, except for those with which I have a conflict.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes, except for those with which I have a conflict.
Adriana Padilla,
Yes.
Marv Southard, I'm sorry, Marv, I didn't catch your vote.
Yes.
Thank you. Carol Watson, hi, Carol, I believe you're muted,
Oh, I'm so sorry. Yes,
except for those with which you are in conflict.
except for those with which I'm in conflict.
Thank you very much. Kevin Zhu,
Yes.
great. Thank you very much. That motion carried.
Okay, all right. Thank you, Scott, and thank you, everyone, for the discussion. And thank you, Gil.
Thank you.
I'm going to invite Dr. Haley Lamb up to the podium where we consider agenda item number 8, applications submitted in response to our CLIN 2 program announcement.
Thank you, Dr. Imbasciani, and good morning to the board. I'm here to present. Is the microphone on?
Yes.
Should I speak closer? Is that better? Okay. I'm gonna move this down a little bit so it's closer to me. All right, good morning. It's my pleasure today to present the Grants Working Group recommendations for the CLIN 2 Clinical Trial Awards. As always, we begin with our mission, accelerating world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. Our strategic goals for the CLIN 2 program focus around Goal 4, propelling 15 to 20 therapies targeting diseases affecting Californians to late-stage trials. We've been funding clinical trial programs for a while now, and the updated CLIN 2 program has a couple of changes that allowed us to support emerging novel clinical trial designs, incentivizes market access strategy development and pre-commercialization activities, and prioritizes innovative therapies for diseases that affect Californians. The overall program objective is to support the completion of interventional trials for innovative stem cell-based or genetic therapies addressing serious unmet needs and with the potential for transformative benefits to patients, families, and the healthcare system. A little bit about the program structure. So this program is offered 4 times per year with a max duration of 4 years And the funding limits depend on the award stage and whether the applicant is a for-profit or nonprofit. The program expects awards of somewhere ranging between 9 to 16 awards per year. And the board has approved for this current fiscal year a total of 135 million. 42 of that has been expended thus far. A little bit about the review process. So the CLIN 2 program allows applicants to submit a full application, and those full applications for this current round had 18 applications go through the qualification process. This allows the CIRM team to apply the program preferences and for the grants working group to apply the value proposition evaluation. From this process, the top 7 applications advance to our typical internal CIRM eligibility review and go to the scientific merit review by the grants working group. From there, they get recommendations that come to here for final funding decisions. A little bit on how the grants working group evaluates the applications. So the composition is up to 15 scientific members of the grants working group that provide expertise on disease areas, regulatory, manufacturing, and clinical operations. We have our access and affordability experts that provide an evaluation along those areas. We also have our board members that provide a patient perspective on an impact on and provide oversight onto the process. We also have scientific ad hoc specialists that come in and provide expertise on specific areas of the applications. The scoring system for the clinical program is the same as our other scientific programs, using a scale of 1 to 100. A median score of 85 to 100 is a recommendation from the group to fund if they are available. And a median score of 1 to 84 is a do not recommend for funding. The scoring is meant to be holistic and based on all facets of the expert reviewers. And the grants working group is encouraged to make use of the full range of the scoring to signal their enthusiasm for the project overall. They evaluate these projects based on 5 review criteria, the first being value proposition, second, rationale, third, the project plan and design, fourth, the project team and resources, and finally, the population impact. A note on access and affordability. So all applications in the CLIN 2 program are evaluated by subject matter experts in this, and their reviews are presented at the grants working group meetings along with the scientific evaluations and the board member reviewers. These may inform the scoring of the review criteria, and there is recognition for especially the early stage trials that any deficiencies in these programs can be addressed during the award period. So to summarize the grants working group recommendations for this recent round of clinical applications, 7 applications were reviewed and 3 are recommended for funding for a total applicant request of 31 million even. And as I mentioned before, so there's a 135 million application for the fiscal year, and we currently have 92 of that available based on the first round of CLIN 2 approved about 42 million. So we're at 92 for the current fiscal year available. In terms of minority reports, as Gil described earlier for the DISC 4 program, the requirements for minority reports are the same for all of our programs. And for this particular round of CLIN 2, there are no minority reports in this cycle. And to present the CIRM team recommendations, the CIRM team concurs with the grants working group recommendations to fund the 3 applications listed here, CLIN 2 19270, CLIN 2 19119, and CLIN 2 19416 for a total of 31 million. And I'll pass it, can we show the Excel spreadsheet, and I'll pass it to Chair Imbasiani? Thank you.
Oh, there it is. Good. Okay, thank you. Thank you, Haley. So while there might be changes in the future to how we consider clinical applications at this stage for funding, I'd like to maintain the system we've always done, which is to consider the clinical applications for funding one at a time. So There are 3 recommended by the team, and the chair would like to entertain a motion to accept one of the ones that have been recommended for funding.
So moved.
You just specify which one, Leonda. You could do them in order.
19270.
Yeah, it's not an eye test, I know.
I know, I'm like, do I say that? I got it right. Thank
It
you.
is, it's CLIN 2 19270 is the motion.
It felt like an eye test, but thank you.
Yeah. And do I have a second?
I can second.
Hey, okay, thank you. We have a motion and a second. Any discussion from board members? Haley, you might want to maybe say something. Yeah, Anne Marie?
Yeah. It's simply a question. We have had all the documents, but for these 3, can you tell us or remind us which stage they are? Is there entry into phase 1 to phase 2? Where are they along the line where we'd like to see them progress towards close to phase 3?
Right, so this particular application actually it has an active CLIN 2 clinical trial right now, and this project would be an expansion of that existing trial based on FDA feedback.
Okay, for each of the 3?
Oh, so that's, I'm just speaking of 19270, which is the current application under discussion. Do you, I'm not sure if I can speak to the other 2, or is that okay? Okay, yeah.
Any other discussion? On 19270, anyone in the audience or in the public want to speak to this? If not, Scott, I think we can proceed.
Just one moment.
Yep.
All right, I won't be calling the names of those who have a conflict with this application. Maria Bonneville,
Yes.
Marguerite Casillas, excuse me.
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Kirkshin, Anne Marie Duliege.
Yes.
Oh, I'm sorry, Shannon, you're fine. Shannon Dahl,
Yes.
Isabel Duro. Wait,
Yeah.
excuse me, I'm sorry.
Oh.
Have to, I'm reading across. No, Isabel. Apologies. Mark Fisher Colbury,
Yes.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Adriana Padilla,
Yes.
Marv Southard. I'm sorry, Marv.
Yes.
Thank you. Carol Watson
Yes.
and Kevin Zhu.
Yes.
Thank you. That motion carries.
Thank you. We can now consider one of the other 2 remaining applications that have been recommended by the CIRM team. Yeah, Haley's in place in case anyone has any questions. So if a board member would pick one and nominate it for funding.
I'll nominate 19119.
Yes, thank you very much.
Second.
And we have a second from Vice Chair Bonneville. Discussion from board members. This is the application on Pitt-Hopkins syndrome.
To address the question earlier about the stage of the program, this one is a phase 1 first-in-human trial.
That case, first in patient trial, that's right?
Yes.
Okay.
Fine, thank you. If not, anyone any member of the public want to speak on this? Okay, Scott?
Maria Bonneville,
Yes.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Anne Marie DeLiege,
Yes.
Mark Fisher Colbury,
Yes.
Vito Bassiani,
Yes.
Rich Lahara,
Yes.
Adriana Padilla,
Yes.
Marv Southard.
Yes.
Great. Carol Watson,
Yes.
Kevin Zhu.
Yes.
Thank you. And the motion carries.
Thank you. We have one final application recommended for funding by the CIRM team. And that would be 19416, blinding eye disease.
So moved.
I'll second.
We have a motion and second. Any discussion from board members?
Again, to address the question earlier, this is also a first in human trial. First in patient.
Okay. Any member of the public want to comment? Are you ready to go, Scott, again?
Mhm.
Thanks.
Marie Bonneville,
Yes.
Marguerite Casillas.
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Anne Marie DeLiege,
Yes.
Isabel Derone,
Yes.
Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes.
Vito Bassiani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
and Kevin Zhu.
Yes.
Great, thank you, and that carries as well.
Thanks, Scott. So the final part of this agenda item would be to close out the recommendations. So Chair would like to entertain a motion not to fund the remaining applications.
I'll make that motion.
Thank you. Board Member Casillas moved and
Second.
Leandra seconded. Thank you. Anyone?
Is there, may I ask a comment, question?
Yes, open to comments.
Just, let me check.
Okay,
Okay.
I'm okay. I just had a general comment. I noticed that, with these applications, I noticed the score cutoff, but also noticed the amount. I was wondering if someone can explain, was this the amount for just this round, or is it all the clinical applications? And then does that go back in the general pool, or is there going to be another clinical round?
There will be one more clinical round for this fiscal year.
Okay.
Coming.
And it's the remaining funds that go into this, or was it
Whatever is
money for
left
this
after
round?
today, right, will be applied to the third round for this fiscal year.
Okay, so there was no limit on the amount on this round. It's just that that's for all rounds.
The 135 approved by the board is for the entire fiscal year, which for this particular fiscal year covers 3 CLIN 2 rounds. I hope that makes sense. And
Okay.
one round has already been voted on and approved last year, and
Got
then this is the second, and there will be one more coming to
Okay.
you. Yeah.
Thank you.
That was good to clarify. Any other questions from board members? There is a hand raised from the public, Lana, and we could take it now.
Yep. All right, we're now opening the floor for public comment. If you're joining us in person, which there's quite a few of you guys, one at a time, please approach the microphone right here. If you'd like, state your name for the record, and we will keep you to 3 minutes.
Good morning. I'm Karen Gensler, PI for CLIN 2, entitled Trileucovax or TLV, a first-in-human acute myelogenous leukemia vaccine for relapse prevention. We appreciate the grant review and also the opportunity to speak in front of the ARS today. AML is the most common leukemia, affecting about 1,800 Californians per year, whom I've been treating for the last 35 years. While about half of patients achieve remission, without transplant, most patients relapse and die. I want to make 3 points very important for the ARS CLIN 2 funding this morning. Point number 1, new strategies to prevent relapse are urgently needed. We developed our novel immunotherapy, TLV, thank you, to eliminate persisting leukemia and address this need. By engineering patients' own leukemia cells to make TLV, we are creating a personalized therapy that could be used for any patient. This strategy is based on the established biology of graft versus leukemia effects after stem cell transplant, where donor immune cells can eliminate residual leukemia. The goal of TLV administration is to stimulate the patient's own immune system to recognize and kill the residual leukemia that causes relapse. Our peer-reviewed preclinical data strongly supports this concept. Point number 2, the phase 1 clinical trial is ready to go now. With 7 years of CIRM funding, we have built the infrastructure for the manufacturing of TLV, which my colleague Dr. Donald Cohn will describe. We've also obtained the necessary regulatory approvals at our institutions and by the FDA. The importance of our approach is supported by the fact that the FDA granted TLV both orphan drug and fast track designations in recognition of the novelty of our immune therapy and the critical need that it addresses. Point number 3, our major goal is to make TLV broadly available to all Californians. Again, any patient can potentially receive TLV. This is in contrast to stem cell transplants, where access for older patients is limited by the need for a match donor, high risk, and rigorous health requirements. Once manufactured, TLV can be available and shipped to an outpatient therapy in virtually any community. The 9 open letters submitted to the board last week strongly support our scientific validation, regulatory and manufacturing readiness, and a clear path to clinical and commercial development. In summary, our clinical trial is urgently needed and critical to addressing the devastating illness. It is ready for enrollment now and will maximize access and availability to patients all over California. I therefore respectfully urge you to please fund TLV this morning. Thank you for your attention.
All righty. Next person up, you have 3 minutes.
Good morning. My name's Kevin Harrington. I'm an AML survivor. I'm here today to strongly urge the board to continue funding Dr. Gainsler's True Loop Vax program. The 2 worst days in an AML patient's journey are the day of diagnosis and all too often the day the disease returns or relapses. The journey between those 2 events is daunting, in my case, an intensive chemotherapy, induction, consolidation, and if you reach and maintain remission, a bone marrow transplant. That in itself can be lethal. As a survivor, I know that my recovery was a brute force miracle of modern medicine. It doesn't end there, however. Post-BMT, multiple ICU stays, intubation, blood clots in both legs, arms, lungs, requiring 2 operations, multiple ambulance rides to support the mounting hospital stays, GI bleeding, repercussions of an infected BMT donation, C. diff, and a significant deconditioning after steroid treatment for GVHD, resulting in time spent in a rehab facility to learn how to walk again. While this may seem like a worst case scenario, after spending significant time over the years in AML and BMT online forums, it's not unusual, nor is it unusual for the large number of patients who have survived a BMT or less fortunate who are transplant ineligible to fall into relapse. Their option to go through that again or simply wait. Remission is not the end of fear. It's the beginning of a different kind of watch. Every blood test, every bruise, every fatigue triggers the same response. Is it back? To this day, I still pore over and trend my test results. Also want to mention the hospital staff who, despite never-ending revolving door in and out of treatment, continue an amazing level of care while wearing multiple hats, long shifts on call, making rounds, and in many cases developing the very research that saves our lives and for the first time delivers real hope to our community. Their dedication to both patients and innovative research like TLB shows how vital the support is for programs that combine compassion and scientific progress. By funding initiatives like this, we honor not just the patients, but also the staff, and the commitment drives these breakthroughs. I ask on behalf of all AML patients, particularly the 50% that will relapse, to fund the True Low program. Thank you for your time.
Thank you very much. All right, next person up, please step close to the microphone and state your name. And you have 3 minutes.
Good morning. I'm William L. Brown, a retired venture capitalist and biotech CEO. And I want to talk about two things, time and leverage. I have a rare leukemia, one for which there's not enough data to suggest a definitive treatment. For all AML patients, and for me, there's no certainty of a cure. We live in hope each day that one will be announced. 1 in 3 leukemia patients have AML. Over 130,000 AML patients will die this year alone. For each of them, time will run out. The development of new pharmaceuticals, even by the best scientists, takes time, and time costs money. I've learned that you can't hurry science, but we have here a drug candidate where the time has been invested, and human trials can begin within 3 months of funding. No other drug or drug candidate reproduces all of the features of Trileucovax. Existing therapies are toxic, unpredictable, and not a reliable cure. Trileucovax is a personalized vaccine that can be made for any AML patient, shipped anywhere, and administered in community hematology clinics. Its tumor cell approach could become a generalized platform for vaccine delivery. Recognizing the potential for a cure and the need to move quickly, the FDA has fast-tracked Trileucovax for phase 1 trials. Each of us here today is motivated to leverage the CIRM's funds to treat as many patients as quickly as possible. So how can we achieve this leverage? To do so, we need to involve Big Pharma to complete phases 2 and 3 and to do the marketing and distribution. Big Pharma has become very particular in what they will fund. And if you don't have phase 1 data, your candidate won't be considered. My experience in the biotech industry tells me that positive phase 1 data will generate sufficient Big Pharma interest to complete the development process. Maybe more importantly, Big Pharma will be intrigued by the possibility of a generalized vaccine delivery platform and the leverage it will create for new development. And this leverage will extend beyond Trileucovax by earning UCSF the funds to continue the development of new discoveries. For the over 200,000 worldwide AML patients, most of whom will otherwise die.
Thank you very much. Anybody next?
This combination.
Thank you, sir. Thank you. Anybody else in person for public comment? All righty. Please step up and close to the mic so we can hear you and state your name, and we'll start your 3 minutes.
Thank you very much. I'm Sean Ainsworth with IMUSoft. Really, I pardon me, just want to express my thanks to CIRM and the board and the GWG for the support that you've provided IMUSoft. It's been transformational for us. It's allowed us to accelerate our lead program into the clinic. We've seen some great excitement from the community as a result of that, and we very much appreciate the continued support. I would add that we take the trust that CIRM and the California taxpayers place in us very seriously, and we will be good, disciplined shepherds of that trust and be transparent with our progress. And we're very honored to be part of the CIRM portfolio. So thank you very much.
All right. Right, if you are on the phone for public comment, please press *9 to raise your hand. Anybody else left in person for these two applications at the bottom? All right, I have a phone number ending in 7541.
Haley.
If you press *6 to unmute.
Thank you, and hello. My name is Wes Miller. I'm a Chief Medical Officer at Epicrisper Biotechnologies. Thank you for allowing me to publicly comment on the reviewer responses to our application 19378. As a California-based startup, we view ourselves as a crucial partner in helping CIRM realize its mission to develop not only a diverse portfolio of effective and transformative therapies, but also to make such medicines accessible and affordable to patients. Our application is focused on EPI-321, the first and only epigenetic editing therapy in the U.S. being evaluated in an ongoing first-in-human and first-in-patient clinical study. EPI-321 was developed as a potential cure for FSHD, one of the most common forms of muscular dystrophy affecting about 1 million people globally and approximately 5,000 Californians alone. Currently, no disease-modifying options are available for patients, representing a major unmet medical need. EPI-321 is a potential one-time treatment for patients who desperately need a treatment for this chronic disease and has been granted orphan drug, fast track, and rare pediatric designations by the FDA. Several reviewers expressed concerns that planned EPI-321 clinical doses in the low 10 to the 13th vector genome per kilogram IV range would be insufficient for biological effect and clinical benefit for FSHD. However, 3 lines of evidence support the contrary and must be stressed. First, people mosaic for FSHD exhibit clinically attenuated to absent disease in the setting of 30 to 50% normal nuclei. This suggests that definitive and durable silencing of DUX4 in only this fraction of nuclei may be sufficient to abrogate clinical FSH. Second, and aligning with these natural history observations, EPI-321 non-clinical efficacy data from various unique FSHD patient muscle cell lines showed deep suppression of the DUX4 pathway, reduced apoptosis, and rescued muscle contractile function at biodistribution levels in this range of 0.3 to 0.6 vector genomes per diploid genome. Third, published clinical data from over 80 individuals administered various IV doses of AAVrh74 predict that these planned EPI-321 clinical doses will exceed this muscle biodistribution threshold, and preliminary data from the ongoing first-in-human study are beginning to confirm this prediction. Finally, the company is dedicated to developing novel therapies that can be accessible and affordable to patients who currently have no therapeutic options. Recent improvements in process development have brought down the cost of manufacturing by upwards of 60% already. However, continued efforts to improve the manufacturing process are needed to bring down the costs in late-stage clinical and eventual commercial settings. We strongly believe this squarely aligns with CIRM's mission. I hope this helps address the remaining reviewer concerns and that CIRM will strongly consider our application for funding this cycle and help to bring EPI-321 to patients who need it most. Thanks so much for your time.
Great, thank you so very much. Is there any other public comment? I think we're good, Chair.
Okay, so clearly there's a lot of interest in the 2 applications that were not recommended by the team. If there is interest on the part of board members in funding one of those, we need to defeat the motion on the table. And preparatory to that, and with respect to the closeness of the scores and our remaining budget, I'm going to ask Dr. Lamb if you would comment a little further on your deliberations, on your recommendations.
Thank you. Yes, so the CIRM team did an internal check of both of these applications. I will note just additional information for the board. Let's see, so addressing both of them separately, 19378, the FSHD application. This application does not have, or this indication, the CIRM portfolio does not have any active awards in the PDEV or the CLIN2 portfolio. And then regarding 19191, this is the AML application. This one, the CIRM active portfolio has no PDEV application or awards rather, but there are 2 CLIN 2 active awards in AML, but with a different modality from the current application, just for additional information for folks, if that's helpful at all.
Thanks, Haley. Does board members probably have some questions for Haley or?
Yes, first in general, I wanted to thank the members of the public, scientists and patients who have since this morning, including for DISC4, had the courage to come to the microphone or to their phone to explain the urgency and the importance of the program. And I want to assure that the board at large is very sensitive to that, and thank you for this. Second, I wanted to know if you could tell us a little bit about the comments that were made by the grants working group, particularly in regard to application 19191, the autologous AML vaccine.
Yes.
Because this one had relatively a low score compared to what's the minimum threshold to be considered, 80 versus 85. Can you tell us about what justified this low score when we hear otherwise that this is an intervention that is ready, that has been approved in essence by the FDA to start? There's a lot of positive things about that.
Yes, I think the reviewers agreed on those points. It's a needed option for AML for those that are ineligible for the standard of care transplants and cannot undergo the high dose chemos. I think the concerns from the grants working group mainly were around the design of the clinical trial. So the concerns, I think the primary driver of the scoring, I guess, there were some minor concerns around the manufacturing, just being complex, and possible concerns with the yield of the product. But I think the major concern, going back to speak to the trial design, was they believe that the design of the trial would ultimately be safe as a phase 1 trial, but it would be difficult to interpret the data and understand whether it would go forward as a go, no-go decision. That's kind of sort of summarizes the grants working group sentiment in terms of how they voted. Basically, it would be they would be able to determine whether the product was safe, but not whether they wouldn't be able to understand whether it works and whether to move forward to the next stage of phase of trials.
Isn't that the main goal of a phase 1 study, to determine safety, but not necessarily to have a signal to move ahead? The signal is given by a phase 2 trial with a more complex design. So it's hard to expect substantial efficacy information, of course, at the end of a phase 1 inpatient study.
Yeah.
Could you provide the same perspective on the other award as well, other grant?
For just so we're clear, 19378 for the FSHD, right? So for this one, also high unmet need, and it's a potentially a one-time treatment with a novel approach on the epigenetic side. The concern from the grants working group here focused mainly on the toxicity of the potential toxicity of the treatment, which was also commented by the public comment as well. And again, they thought that the proposed doses for the trial were again likely to be safe, but not likely to make a difference in the disease in terms of disease modification.
I guess it's the same comment Anne Marie made about the other one. Safety is the goal of phase 1.
Scott, could I ask you to repeat the motion? Or Beth?
The motion.
On the table.
The motion is to not fund the remaining 2 CLINs, so those would be 19378 and 19191.
You know, I'm asking fellow board members or members of the CIRM team to help us. How can we address the issue where the comments from the grants working group was that there would be insufficient data at the end of this trial to give an indication of efficacy, not proof, but indication. And we'll recognize that the design of phase 1 trial cannot generate that.
Yeah, so I think that is the understanding from the grants working group, that it is a safety trial. I think their hope is that the hope would be that there could be some signal and some data from some of these trials, especially for the more rare conditions. But I think I would also say that the applications are scored as in these clinical reviews across all 7 applications. So there is some consideration for the relative enthusiasm that they might have for projects across the clinical. And Dr. Zambrano.
Yeah, so
No, I'm sorry.
sorry. So I think one other thing to note is one of the things that we stressed with the grants working group is that we're looking for novel transformative therapies. So part of it is that, it is not necessary, as was noted, that an initial phase 1 trial show any efficacy or necessarily any signal. They're often designed to assess safety. But I think they are also looking at, is this a therapy that's likely to, either when it goes beyond the phase 1, to be effective and to have a meaningful impact on the patient population. And so I think that is part of the calculus that is in there. And so the reason they look for at least a hope for some signal of efficacy is that it's going to also signal that this is really truly a transformative therapy. Now, that doesn't mean that, of course, that, we can't fund a proposal that is focused on safety. I think that is okay. I think the other thing to consider in terms of just the budget, so that you're aware, I think Haley noted the budget is at 92 million currently. This would bring us down to about 60 million. Is that correct, Haley? If we funded the top 3 for one more cycle. So I think, in terms of, is there flexibility in the budget to include an additional or 2 additional? You could, knowing that the next cycle could be impacted, but at the moment each cycle is using up about maybe 40 million per cycle. So the remainder would still likely be adequate. So I think that is something that you can also take into account.
Right, so just to look at the numbers, if these 2 applications were funded, that would be an additional 20 million for this round. And that would bring the total total awarded for this round to 51 million, and out of the 92 remaining. So that would be 41 remaining for the last round, if these 2 applications were funded.
Thank you, Haley. In order to, in a sense, prompt or force further discussion among board members, I'd like to amend the motion. And I'd like to amend the motion to include funding application CLIN 2 19191.
Only that one?
We could do them one at a time. I think it would be cleaner to do it one at a time.
So the motion is to amend the motion on the table to fund
Thank you.
19191. And is there a second?
I second, yeah.
Mark Fisher-Colby? Mark, you need to unmute, I think, yeah.
Sorry about that. Is it okay to make a comment?
It is.
The floor is yours.
Yeah. Hey, yeah, I just wanted to comment that the reviewers are extremely familiar with the fact that phase 1 is intensely focused on safety, and there's almost never going to be a determination or even a nod in terms of whether or not there's a signal because of the highly limited number of patients. So the reviewers very much factor that into their consideration. That's part of the DNA of the review cycle. So I didn't want there to be an intimation that there is a clear wait for a signal out of a phase 1 trial, because everyone knows that's highly unlikely. And this is then a circumstance, just to give nuance to the previous comments in the context that people are looking at what's next and what is the risk profile for achieving the phase 2 trial work, and what are the characteristics that in the phase 2 where a signal might be able to be determined, come into play. So I just wanted to clarify some of the comments related to how people make their determinations on the scoring. Thanks.
Thank you, Mark. John Carruthers?
Yes, thank you. I thought you were going to make the motion on 19378 as the one to, because the scores were so close. So I was wondering if Rosa can add, I'm sorry, someone can add comments. My understanding that 191, the score was lower from some of the previous comments. I was wondering if there was some additional detail. I got confused.
The door is not closed on.
Okay.
Not at all.
I mean, I don't think I can make a motion, but it would seem like 19378 would be the one I would include, not 191.
Yeah, thank you, Dr. Carruthers. So for 19191, in our view, that's how we agreed with the grants working group recommendation, is that it's hard to justify funding as submitted. The application, as it was provided by the very good public speakers, it addresses a very real need, but the record supports the grants working group view that the trial is not sufficiently differentiating actually or interpretable for a 12 million first-in-human award. The biggest issues are not that the biology is not plausible. The reviewers did say that the rationale is sound and the preclinical data are supportive. The problem is that the application didn't convincingly show that this vaccine is meaningfully better than prior AML vaccine efforts. And the proposed trial may not generate the clear go, no go answers. So with that, our strong recommendation is they can come back and address the reviewers' comments and reapply. One of the things that we will have today in the discussion that we will be providing with regards to the preferences going into guiding principles is what are our guiding principles? And we are going to review what the guiding principles for CLIN2 are. We want transformative therapies that differentiate from what's already out there, right? And we have a limited amount of funding and time, right? So that with that, our recommendation could be what the grants working group said, address those issues and come back. And there are 4 instances of clinical applications a year.
Thank you.
Thank you, very helpful.
Yep. Any other questions from board members? I presume we're going to head to a vote on the amendment first before the main motion. Any member of the public want to comment on the amendment? Please. If I remember, Dr. Gensler, yes?
Permitted?
Yes. Floor is yours.
Thank you very much for all this discussion. I think what has gotten buried is the very promising data that we do have. In our proof of concept data, we looked at the ability of this vaccine to eradicate active aggressive leukemia, which it did in 80% of models of
very aggressive disease. I know that's a mouse model. However, in our in vivo data, we were able to take patient immune cells after remission induction and co-culture them with the vaccine and see specific leukemia-lytic effects that did not target the normal bone marrow in most patients. And while these were variable, they were striking because the immune effects were seen in remission patient cells who'd received lymphodepleting therapy. So I think this is very strong evidence that this could have activity. And it is very different, and I'll now be a little bit scientific, from other vaccines because we have provided the co-stimulatory signal of CD80 to actually trigger cytolytic T cell effects. And for that reason, it is different from every other vaccine on the market and can be made for any patient exclusive of HLA, ethnic, or other descriptors of patients. So this is a universally applicable vaccine that we know how to make. Unfortunately, Don Cohn was not able to get on and tell you that the manufacturing is highly developed and efficient. And so it is very attractive to have a therapy that really any patient could get in their own community without traveling to a major medical center with the financial cost and difficulties attendant. Thank you for letting me speak.
Thank you. Our President and CEO, JT.
Yes, thank you, Mr. Chairman. Thank you, everybody, for your commentary today. I just want to, as a generic matter, note to the board that the grants working group in any application sees all of the data that is subject matter of commentary on particular applications, and through rigorous process comes to a conclusion, which is either to fund or not fund. It's impossible, as a board sits here today, to hear commentary that addresses the science that you can evaluate as to whether that is persuasive over and above what the grants working group in rigorous and robust debate is already contemplated. So I just make that point, not with specificity to any particular application, but just generically as you consider motions like this. Thank you, Mr. Chairman.
Thanks, Jonathan. Yes, Adriana?
During the GWG deliberations, did the questions go out to the project coordinators about the challenges that were being noted so that they could re-deliberate?
So the review process allows reviewers to ask questions to the applicant and respond if there are any. I am not 100% sure on this particular application, but I would have to double check.
I also feel there's a lot of science that's here that I can't assess. I really need the GWG to assess that.
The grants working group also has the option to, if there are sort of score driving questions, allows us to call out live during the meetings to the applicant if there is an outstanding question that the answer to which would influence how they recommend the application in the final scoring as well.
I have a question. Can you also clarify if there's another, you mentioned there's another round of reviews for this CLIN 2 program this year with the budget. What is the timeline for that? So if somebody was going to resubmit so that the GWG could review again with these responses, when would, how would that unfold?
So if from this cohort, if, for example, there were applications that are not recommended, they could come in at the end of April. There's a deadline at the end of April for new applications for which these would be eligible, or any projects that aren't funded are eligible to submit at the end of April. I think it's April 28th.
And the duration of the review cycle, because what matters is when they could, if a group reapplies, when there would be the review and the duration of the process, and when they would hear about a potential outcome of the reapplication.
Right, so for the April, I believe it would be coming to September ARS.
Okay,
I mean, it would come to the board in September.
okay.
Marguerite?
Yeah, just to Adriana's question that, yeah, so I'm a patient reviewer. We are notified of when questions are due. I feel like the CIRM team does a good job to encourage us to ask questions. And every time I have gotten good responses from applicants. So that piece is working, I think.
I don't see any more comments from board members, including those outside the room. Any member of the public? Final comment? No. We can proceed to a vote on the amendment, Scott.
So to be clear, this is a motion to amend that we're voting on. So there will still be another step. Ultimately, you will still be taking a vote on the underlying motion, assuming there isn't another motion to amend with regard to the other application. So the results of this vote, I'm not going to call on those members who have a conflict as to either of the applications because this affects that pending motion. So again, this will just be a motion to whether the original will be amended. So an aye vote will essentially be to fund application 19191 and then not fund 19378. That will be the new shape of the underlying motion.
Keith, before I see Keith's hand up.
Yes, I don't know if I can.
Wait. No, Keith has a conflict.
Thank you, Keith. I'm sorry, but I think you're conflicted on this. Okay, sorry to interrupt.
So I'll take the vote now. Marguerite Casillas,
No.
Judy Chow,
Yes.
Leandra Clark Harvey,
No.
Shannon Dahl,
No.
Anne-Marie Deliege,
Yes.
Mark Fisher Colbury,
No.
Vito Imbasciani,
Yes.
Rich Lahara,
No.
Adriana Padilla,
No.
Marv Southard,
No.
and Kevin Zhu.
No.
The motion to amend does not pass.
Okay, discussion again on the original motion. Are we finished with the discussion on that? If any board member wants to make a comment or a member of the public? Do you see any hands, Claudette? Or any? No. Okay, Scott, we can go to the original motion. Unless there's another.
Can you repeat the original motion for clarity, please?
Certainly, the motion is to not fund applications 19378 and 19191. So for the following members, Bonneville, Durone, Flowers, Miaskowski, and Watson, you have a conflict, so please indicate your vote, again, with the exception of those with which you have a conflict. Maria Bonneville,
Yes, except for those with which I have a conflict.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Anne-Marie Deliege,
No?
Isabel Derone.
Yes, except for those with which I have a conflict.
Thank you. Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes, except those with which I have a conflict.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes, except for those with which I have a conflict.
Adriana Padilla,
Yes.
Marv Southard, Carol Watson.
Yes, except for those with which I have conflict.
Thank you. Kevin Zhu.
Yes.
Thank you, and that motion carries.
Great. Thank you, and thank you, Haley, do you want a break? Is that?
We would love a 10-minute break.
Yes, you may have.
Thank you. So we'll see you at 10:30.
I presume we can convene again? Okay, we're going to start imminently. Okay, we're going to take up where we left off and direct everyone to everyone's attention to agenda item number 9. This is going to be a discussion of amendments to the Community Cares Center of Excellence Concept Plan, or Infrastructure 8. And the presentation will be led by the Assistant Director of Patient Access, Dr. Jeff Lomax. Jeffrey?
Thank you, Mr. Chairman. Beth, are we okay sound-wise? Thank you. And I'd like to thank the CIRM leadership for giving us the opportunity to present this item, and acknowledge the patient access team for their effort in pulling these materials together and all the thought behind it. So again, this is for your consideration, the reopening of our Infra 8 program, which is more commonly known as the Community Care Centers of Excellence program. Just a brief reminder of the contours of this program. It is mandated by Proposition 14 to establish the Community Care Centers of Excellence. The aim of the program is to expand the capacity of our existing clinical trial network, which is embodied in the Alpha Clinics network, with the aim of promoting access to both human clinical trials and treatments that arise from those trials, particularly institute-funded programs. And a main criteria for prioritizing or for the program itself is to ensure there's geographic diversity in those centers. And so we know that geographic diversity is critical to making treatments broadly available to patients, as we heard from a number of comments, that proximity and access go hand in hand. So this program is intended to achieve that outcome. I'm going to update you on the status of the program as it stands at the moment. In October of 2025, you approved 3 awards pursuant to the Infra 8 program. Those awards totaled a $27 million commitment. The original budget that you authorized in the concept plan was $36 million. So there was remaining funds available through your initial authorization for this program, or those funds are still remaining. I also want to give you a perspective of the current status of our clinical infrastructure program as it stands right now, and that's illustrated by the map on the right side of this slide. We've got the Alpha Clinics in red and the community care centers in blue. And I think what we're trying to get across, and that map illustrates quite nicely, is our current footprint is within the state, but south of Sacramento. So there's about 1/3 of the area of the state. There is no clinical footprint to serve patients. Well, went the wrong way. There we go. And during the October meeting, the ICOC expressed concerns about this geographic gap and pointing out that this region is not served by any current clinical infrastructure. That represents a population of about 3 million Californians. And so the board directed the team to identify options to expand the reach of our clinical infrastructure into Northern California. And so we set about doing that. We've actually engaged with centers. We know there is capacity in that region. And because there is such capacity, what we're recommending is that we reopen the Community Care Center of Excellence request for applications. That would require some very minor technical amendments. We're asking the authorization of $9 million, which would fund 1 award. And that $9 million represents the uncommitted funds from the previous concept plan you approved last year. We would obviously have to modify our deadlines as we have to have new dates. And we would limit eligibility to applicant organizations with clinical operations in those northern counties, which were illustrated in the previous slide in the box. We would also propose a somewhat expedited timeline. So the launch, if there is an application that's recommended for funding and approved by this board, the startup would coincide as quickly as possible with the existing awards, which we've actually just finalized the contract of 2 of the programs you approved in October. And just as a sidebar note, I mean, the excitement and the enthusiasm is palpable. Those programs are just, they're so exciting, and it's just a pleasure to be a part of that. So we'd like any new award to be as close as possible to that program. So we would recommend, or we're recommending the program would close at the end of June, and it would come to the application review subcommittee in September. I'm going to have to take a class in Clicker. There we go. So the request is that the board reopen the Community Care Centers of Excellence program, which is Infra 8, and that the approved concept plan, which is part of the materials noticed for this meeting, that it includes those minor modifications, which I covered in the previous slide. So with that, I turn it back to
Thank you, Jeff.
the chair.
Vice Chair Bonneville?
I'd like to make a motion to reopen the Community Care Centers of Excellence funding opportunity with a modified concept plan.
Second.
Okay. So the motion on the table. Motion is on the table. We have the opportunity to discuss. Anne-Marie?
Yeah, just can you give us a little bit of a historical perspective? You make here, they reopened this CCCE. Were they open before? Did they close? What's the reopening?
Sorry, yeah, just to be clear. So this is a new program. We've just gone through the first funding cycle and approved 3 awards. And the reopening is to allow, when we approve those awards, one of the elements of the approval was to take the money off the table, so to speak, that 8 million. So the reopening, if you will, is a confusing way of saying, can we re-access that 8 million so we can allow the RFA to open and access those funds if there is a successful applicant pursuant to that RFA.
I want to thank the patient access team and also the board for directing that we explore more in this part of the state, because it was clear that we needed to go back out and assess whether or not there were community care centers in that region that could apply to expand our footprint. So I really appreciate it. And there's been a lot of work that's been done with outreach and visiting different sites. And so thank you. I appreciate it. And thanks to the board for understanding how important this is. Adriana.
Okay, and then Adriana.
Similarly, I want to thank you for the work. This is really exciting and it is important. I also want to thank the board for making the call that this was not serving the whole state. So I guess my question is, going forward, and I'm entirely supportive of really reopening it to ensure that we have geographic diversity here. But I guess my question is, going forward, when we have new initiatives, what will be the process that we will take so that from the get-go, we're looking at the state as a whole? I mean, I know that these things are iterative, but it just seems to me that now that you're showing the graph, it seems quite obvious that we had a gap. So what steps would we take in the future to ensure that it wouldn't take the board reacting to have something that is comprehensive and inclusive?
Yeah, thank you for that question. So while the gap could be interpreted as we were remiss and missed something, we have in the prior round, we did engage applicants, and I would note, and there were a number of awards that weren't recommended for funding. Let's say, had all the awards in the original round been recommended for funding, that map would have looked a bit different. So at the end of the day, we aim to fulfill the objectives of the program. But again, we're still dependent on the applicant to submit an application that is deemed meritorious. And so it wasn't a question of not recognizing the need. It was a question of applications that were ultimately funded. We were, when we, at the end of the day, we were left with that gap. So the process worked, and we're just trying to restart it again now with that more narrow set of conditions to ensure we achieve that outcome as best we can, provided there's a fundable application that comes through the process.
Thank you for the clarification. So essentially what you're saying is that it was the caliber of the applications you received that led to what appeared to be a gap in Northern California.
That's correct. And again,
And so what steps are we taking then, now that we're going to focus on them? I would imagine we still want to have high caliber applications. So does CIRM have an opportunity to maybe help build the capacity so that the applications that come forward are in fact strong enough that we were able to fund them?
So we always encourage applicants to contact the CIRM team and ask questions to strengthen their application. And in addition, there are within the network itself, and we saw this in the first round, and I have every expectation this would occur if you were to reopen this application, that the applicant organizations themselves will reach out to the existing CIRM network of alpha clinics, and in this case, community care centers, and engage with those centers and develop the application in such a way where it will be as competitive as possible. And we very much encourage and will facilitate those collaborations or communications during the process.
Thank you. That's one of my questions. I was going to ask, is CIRM going to be nurturing some of the organizations that had applied during the first round, and to assist them and to move into a more successful selection?
Yeah, again, we obviously do everything we can to make this opportunity visible to organizations that appear to be positioned to apply. We help facilitate the networking and communications, as I had just indicated, and as Co-Chair Bonneville indicated as well, we've been out into the region and recognize there are organizations that appear to be eligible for this program, and so again, showing that visibility, giving them information about the process, and again, I anticipate we will have quality applicants in this round. And so I think we have to be very careful. I think once we open an application that we're being fair and objective and not going beyond our remit as what we can do in terms of the applications themselves.
Only because these sites also have limited resources to pull from. They may not have the expertise to build a robust application process. And it would be incumbent on the major university institutions in Northern California to also realize that and reach out from this call of this new proposal.
Yeah, and again, I think that interest is there with the existing centers, and there's certainly headroom within our existing clinical network for those teams to, support some of these efforts. So I think that if the interest in the teams and the brain power is there, we just need to pull those applications through.
Good. Board Member Al-Masri.
So I understand that we missed a big part of the geography, but do we have at least rough estimate of what's the population of this area? Is it 5% or 10% of the state? Because obviously, yeah, we are after the humans, so I totally understand. Because also limiting the remaining fund to only small geographic area who may not have the expertise and the resources to conduct what we are trying to do can be also restrictive.
So maybe I'll just comment on a couple of realities we've surfaced during our needs assessment and leave it to the board to determine how compelling they are in terms of your decision. So again, as I indicated, we're roughly about 3 million residents. That area is two of the existing major medical centers in that region are under, as we understand it, tremendous strain largely because of federal funding cuts. So I won't call them out because I don't want to sort of represent them, but that area is experiencing substantial strain in terms of patient access to healthcare in general, and these are major medical facilities. So there is to the extent there's a need, it's amplified in that region given cuts to federal health funding. They're also, interestingly enough, looking at the sort of epidemiology of the region, there are comparatively high incidence of cancers throughout the northern part of the state. I actually had an earlier map of that. I'm sorry, I don't have it in the appendix, so I would show it. And those are conditions which are sort of uniquely aligned with regenerative medicine treatments. So in terms of the burden of disease, that region is somewhat unusual in terms of what we're seeing in the cancer registry in particular. So there's perhaps a unique opportunity to really help patients in that region. So within those numbers, having a larger proportion of patients that could potentially benefit is a real potential value proposition for this program. And I think finally, just overall, CIRM's, with the exception of a couple of centers, our footprint is somewhat limited in that region. And if we go back to the proposition and the mandate for sort of geographic diversity and access, our interpretation was that what we're bringing forward today does align with what the proposition is asking for us. So I hope that's helpful in terms of how you weigh the benefits or the value proposition of what we're proposing.
In light of what you just said, that the major institutions in the region are under such constraints, I wonder whether limiting this extended deadline to only that region might in fact achieve what we're looking for. Is there a way in which we might encourage collaboration between institutions in the central region, in the southern side of California, who might actually have the capacity to, and are not as distracted by the funding cuts, to support the applicants who are going to come from this region?
Yeah. So, again, just to give you a little bit of a sense of how we try to, through a needs assessment process, look at issues like feasibility, capacity, and in terms of then informing the timelines. So in discussions, and I'll acknowledge because there are known and in discussions, for example, with Fresno, who we've just funded in the first round, they were extremely open and motivated to engage applicants as a sort of peer-to-peer, because they're a center that they have a lot of characteristics of the type of applicant that would come into this program. So in terms of the collaborations you're alluding to, I think there's a very much a willingness amongst the existing centers to drive those collaborations, support those applicants. That they're fresh off of coming through in October. So our sense is that support will be there, but acknowledge that these centers can be have more limited capacities. And I think it goes back to the comment previously. But we, do try to match make and sort of provide those support structures because obviously our mission is to get, or our goal is to get the best applications possible. So it's striking that balance. Yeah.
Okay. Board Member Pat Levitt is next.
Yeah, I just wanted to say that
Unmute, please.
I am unmuted. Can you hear me? Hello? Hello?
There we go.
You hear me?
Yes, we can hear you.
All right, thank you. I just want to say that the community care centers of excellence are really critical in terms of their operations locally, and that right now that 3 million people in that part of the state, if they want to get access to the programs that would facilitate and improve or even referrals for what we're trying to do for clinical trials and for other kinds of treatments, we need them locally in that region. It's 3 million people. And I've talked to Jeff about this. There is a lot of collaboration already amongst those that have been funded and a willingness to share their strategies for how they improve access and engage with the communities. So I think this is a really positive move. I think the CIRM team has done a lot of due diligence in terms of understanding what the opportunities are in that region for funding really strong application. Thanks.
Thank you, Pat. Did you want to say something? No. You're still, okay. Yes. Kim.
Yeah, I just wanted to comment on what I see as the great importance of this proposal. As the academic medical center that is the furthest north in California, at UC Davis, we have a lot of interactions with medical facilities in this part of the state. There's certainly a huge amount of need, but also some very energetic and enthusiastic partners that we've had discussions with about other types of collaboration. But those conversations let us know how much this is needed. And when we're talking about all the way up to the border with Oregon, those people are way too far from the existing centers to have the ability to access.
Thank you, Kim. Anne Marie?
Yeah, just to say that I'm also very much in favor of this proposal, but maybe a more basic question for some of us who may not have been part of the board for a long time. Can you remind us the mandate of a community care center for excellence? What defines a great center? And for those centers that have been open for some time now, how have they helped the mission of the CIRM? What difference they have made? I'm sure they have. Would be great to have that.
Yeah, thank you for that. And so I just want to go back and as a reminder, so this program, this program is 3 days old. We have just, we've just signed our first contract. So this is a new program. Your October decision was the initial round of funding for this program. And so we can't give you outcomes at this time. So this is an experiment, but it's probably one of the coolest experiments in regenerative medicine right now. Everyone, again, I think 2 years of needs assessments, interacting both with commercial providers who have approved therapies and our clinical trialists who are trying to reach patients. Everyone comes back to the value and the importance of working outside of the academic centers to engage these patients. And so we think, based on that sort of collective wisdom, that we've really lined up a program that's going to be fit for purpose, but we just can't answer at this time. And then I do want to come back to the point that if we can get them all working at the same cadence, which is what we're trying to do here, is to allow this first cohort to move roughly the same cadence. We see a lot of value in that because there's a lot of co-learning and learning in that initial sort of uptake process, accreditation, clinical research, the clinical research aspects of it. If they can sort of help work with each other in a partnered environment, we think we can get that 1 + 1 = 3 and the value of the network. So, but like I say, this is one of those great CIRM experiments.
And I think part of that question was the mandate.
I've just put this. I've just come back to the slide to kind of remind us of how Prop 14 put forward this request or put forward this mandate in the proposition.
Yes, good. No other comments from board members or questions for Jeff? We'll open it up to members of the public. There are none. Okay, so we have a request to the board to put the request back up again to alter the Infrastructure 8 program with amendments that Jeff has gone through and approve the modified concept plan. So I think we can go to a vote.
Okay, so I'll take a voice vote in the room, and then I must pull members individually on the Zoom. So all those in the room in favor, say aye.
Aye.
Those opposed, nay. Any abstentions? And I'll pull the members on the Zoom. Monica Carson.
Aye.
Judy Chow.
Aye.
Isabel Derone.
Aye.
Mark
Yes.
Fisher Colbury. Thank you, Isabel.
Aye.
Judy Gasson.
Aye.
Jeff Golden.
Aye.
Rich Lahara.
Aye.
Pat Levitt.
Yes.
Hala Madna. Maybe out. Chris Miaskowski.
Yes.
Marv Southard.
Yes.
Carol Watson.
Yes.
Kevin Zhu.
Aye.
And Keith Yamamoto.
Yes.
Great, thank you very much. That motion carries.
That carries. Thank you, Scott. So good. Happy to move on to agenda item number 10, which will take us up to our lunch break. This is the second installment of a new and regularly occurring agenda item that features researchers and patients who have benefited from CIRM's activities. And to lead the discussion, our Director of Communications, Amy Adams.
Thank you, everyone. I'm excited to announce two speakers today. You're first going to hear from Dr. James DeClos, who is Distinguished Professor of Biotechnology and Biomanufacturing at Solano College, which is in Fairfield. He runs our COMPASS program in Fairfield, and they've done some incredibly innovative stuff there. Jim will introduce some students from the program who will also present. And I just want to point out, Jim is so dedicated to this program and talking about it with all of you. He took a red eye in this flight environment. He took a red eye to get here. So I'm just incredibly grateful for that. Following that presentation, you're going to hear from Diana Farmer. She is Surgeon-in-Chief at the Children's Hospital at the University of California, Davis, and Chief of Pediatric Surgery at Shriners Children's Northern California. She's going to present results from her work developing a stem cell treatment for spina bifida. CIRM has supported several stages of her work and is currently funding or supporting a clinical trial. So Jim, I'll turn it over to you. And your clicker is right here and your slide should be.
Thank you very much. I'm from Solano College, and we are about 40 miles west of here. As was mentioned, I took a red eye from Washington, D.C. I was invited by the Federation of American Scientists to lobby Congress to try to institute a series of recommendations that came out in a recent report that was commissioned by the Senate and carried out by the National Security Commission on Biotechnology. And let me just say that I'm happy that we have CIRM in California. So thanks. Oh, here it is. And to introduce Solano County, again, we are really 2 counties over from here. This is the poorest of the 9 Bay Area counties, although we straddle the fence between the Bay Area and the Central Valley. I put up data from the last census. We are formally, according to the U.S. Census, the most diverse county in the state of California. Also, we contain the city of Vallejo, which is the most diverse city in the United States. So we take our mission of diversifying the workforce in regenerative medicine very seriously. We have on the right, you can see we have a new $34 million biotech training facility. And we have multiple campuses. Our training facility is in Vacaville, which is even closer to here. Vacaville is the center of a biomanufacturing hub. In the community college system, what we can do something that our brethren at UC and CSU cannot. We can pass a school bond with the local voters, and our voters passed a third of $1 trillion, that's a third of $1 billion. I was in Washington. The numbers are, yeah. Okay, third of $1 billion school bond, and now the voters added $10 per year on the property tax. Yeah, that was a slip. And everything that you say about us can be said about our partners in northern San Diego County, Maricosta College. There is their brand new biotechnology training facility. Their voters passed a $500 million bond, and they have a parallel program to ours. So I wanted to talk about our journey here and how it is reflected in our journey in regenerative medicine. So we started our biotech program in 1994, or in response to a 1994 announcement by the pioneering company Genentech that they were going to build the largest cell culture manufacturing facility in the world in our community. So I went to them and I said, I'm going to start a biotech program, and my background is molecular biology. And they said, you're going about this entirely wrong. This is manufacturing. What you need to do is teach the skills and knowledge that are required to go into manufacturing, not into research. And there were biotech programs at multiple colleges and universities around the state and around the country, but no one had done that to focus on what's now called biomanufacturing. And there's a picture of Genentech Vacaville. It's a gigantic plant. They grow cells in tanks that are about the same size as the tanker truck that brings gasoline from the refinery to your local gas station. Each one of those tanks may have $30 million worth of product in it. At its heyday, that one building made more money per year than Disneyland and Disney World combined. That is, it makes antibodies, and antibodies are very lucrative. And so this is what workers look like when they work in the field. They're gowned up not to protect them from the product, they're gowned up to protect the product from them. And here are some of the protein pharmaceuticals. It was really built for Herceptin, which is an antibody that treats an especially aggressive type of breast cancer. Before Herceptin, women who were diagnosed with that type of breast cancer would not last the year, and many of those women now are lasting beyond the 5-year part, and there are improvements to that as well. So these are protein pharmaceuticals, and that's where we started. And you can see how lucrative protein pharmaceuticals are. These are the top pharmaceutical products, and most of the top pharmaceutical products are biologics. They're made not by mixing chemicals into a tank, but they're made by growing cells in a giant tank and having those genetically engineered cells produce the protein. The protein is then isolated to purity and injected into patients. Notice that the annual revenue there is greater than some of our states. And here's some pictures from Genentech, and again, gigantic chemical engineering challenges. And this is what a worker looks like. These are chromatography columns on the right, and it shows the scale of just how massive a plant like this is. Now, Genentech was acquired a year and a half ago by the Swiss pharmaceutical company Lonza. They not only acquired the company, but they acquired a working company with all of the quality control staff intact, all of the maintenance staff intact, all of the operators, and it's worked out very well for them. And we have students interviewing with Lonza this week. So our program started out as an associate's degree and a certificate. It was formerly career technical education, what we used to call VOCED. That is, we are gauged and measured by the number of students that get placed into the industry. And it isn't only the science, we also have a major component of it that teaches the students about FDA regulations, that teaches the students about the business part of the industry. And this provides a career that pays a livable wage to start with, and it has tremendous potential for upward mobility. So it's not a job, it's a career. The students often move up, and what a recent study showed is they move up even faster with a bachelor's degree. So when the state of California authorized community colleges to offer bachelor's degree, we applied, and the stipulation by the legislation was that you could not offer a bachelor's degree in a discipline that was already being offered by either the California State University system or the University of California. We made the argument that biomanufacturing was fundamentally different than the research-based biotechnology that they were teaching. And what that's allowed us to do is have a seamless transition between our associate's degree into the baccalaureate degree. And that creates a complete education pathway. We are teaching dual enrollment in the local high schools, and then we have stacked certificates, 3 stacked certificates, including a cell and gene therapy certificate. Then add general education. That's an associate's degree seamlessly into a bachelor's degree, but beginning to end under $10,000. And most students will pay nothing, especially those who are funded by CIRM will exit college without debt. The other thing that has occurred is we've negotiated with 15 different universities to pass our students into graduate programs. So this is a complete education pathway from high school through PhD, but what's important is there are multiple transcriptable exit points and easy reentry if life happens. What happens to a lot of students is they're in a 4-year degree, something happens, they exit the 4-year degree, and then they have a lot of classes and nothing on their transcript. And I've noticed that many universities now are thinking about offering an intermediate associate's degree in order to have something on their transcript if a family member gets sick or if there's a family emergency, if there is some reason to exit. And students have very complicated lives now. So we were, as I said, one of the first 15 community colleges, us and our partner, MiraCosta in San Diego. Since then, currently, California, although we got started late in community colleges offering baccalaureates, California is starting to catch up. We're now 4th in the country. 24 states have enabled their community colleges to offer bachelor's degree. The number 1 state that does this is Florida. Number 2 is Washington State. Number 3 is Texas. We're number 4. We have our eyes on Texas. So as of last week, there are now 63 bachelor's degrees at 46 community colleges. What's important on that chart is that there are now 3 additional biomanufacturing programs in the state. All 3 are in the LA area, and that gives us geographic diversity. Well, the first one after us was Moorpark College. They're across the street from Amgen, which is the largest independent biotech company in the world. They've had biomanufacturing associate degree and certificates for a long time. It was logical for them to go next. And then our partners at Los Angeles Mission College, what's important there is they have a very tight relationship with Cedars-Sinai and with their regenerative medicine group there, and they would be a good candidate for a CIRM program. And then LA Pierce College is starting next year. By the way, Moorpark Colleges, their first cohort will graduate in May. Our 8th cohort is graduating in May, and our 10th cohort, we're bringing in our 10th cohort. Now, these are places where the students can go after they graduate, and notice the dominance of CIRM in the graphics. So summer interns, we have a very, very close relationship with the Stanford Laboratory for Cell and Gene Medicine that I found out last week that 4 of their workers were trained at Solano College. But more than that, all 4 of those workers were supported by CIRM. All 4 of those workers worked at the Laboratory for Cell and Gene Medicine, and they're up in the upper left-hand corner is DeMario. He's the poster boy of their summer internship. He's from Solano College. And down below is Arlene. She graduated from Thomas Jefferson University in Philadelphia. I'm about to introduce Jen. There she is standing by her poster. And then we have a close relationship with several of the CIRM Bridges master's degrees, especially Sacramento State. We have a student there, Cal Poly, and others. We have a student who just completed the Stanford Cell and Gene GMP certificate program, and she's been hired by the Stanford Laboratory for Cell and Gene Medicine. So what we did when we were looking at proteins is I always overquote Wayne Gretzky, I skate to where the puck is going to be, not where the puck has been. And so what happened in my journey is across the street, many of our students who were working with traditional biotechnology, producing protein pharmaceuticals, were being recruited away by cell and gene therapy companies, and they were giving these gigantic signing bonuses. So I said, oh, you've got my attention. So we jumped into it, and with the help of the National Institute for Innovation in the Manufacture of Biologics, we did crash courses, and we now have a cell and gene therapy certificate. And we teach the students how to generate CAR T cells. We also teach the regulatory environment of it. Our students work under simulated good manufacturing practice, and they are well-versed in the laws and regulations that govern pharmaceutical manufacturing. And we also produce adeno-associated virus, which is the lead vector in gene therapy. So that brings us to, we applied for and received a COMPASS grant, and that would have been 4 years ago. And what's significant about that is that the COMPASS grant was for 4-year colleges. That is, only 4-year colleges were eligible. And so of the community college system, only Miracosta and Solano College were even eligible to apply. And we applied and received that, and it's been a game changer for us. And notice we're in very prestigious company there. Over on the right is a slide of our partner Miracosta, and those are their COMPASS scholars there. So these are our COMPASS scholars, and again, we're in our 4th cohort. They go on a summer internship between their junior and senior year, and they come back transformed, and you'll see that. That is not the same student that I sent to that internship. Down below is Jessica, and she is indicating that she just finished preparing CAR T cells, and those CAR T cells went into a patient the very next day. Our other project, which has been one of the engagement projects, we've been working with Be the Match, now named the National Bone Marrow Donor Association, and our students will do recruitment drives where students, you can see in the upper left-hand corner, students will take a cheek swab, and that'll be sent off to be sequenced, and then they will get a tissue type, and that will go into the national registry. And I just learned this morning that we are number 3 in the country in terms of recruiting students into the national registry. We have had 3 matches, And the match in the lower right-hand corner is Jose. Be the Match flew him to New York, where he donated stem cells. He donated bone marrow cells. And as far as we know, the 14-year-old who received them is still alive. That was the last that we heard. Our students participate in outreach. You can see them here with middle school students, but they also do K through 12, and they also volunteer with the local adult school. And we do this maybe every other week. And then our community turns the city of Vacaville into Bioville every year. Everyone is invited. It's right around DNA Day. And we have events for the community that are related. Now, related to this is we're very closely aligned with our local economic development folks. And whenever a biotech company is looking for a site, Vacaville, the city of Vacaville, always brings them by Solano College, where we guarantee we can provide them with a workforce. And that workforce is a major component of the site selection criteria. And we've been successful in attracting 4 companies in the last 3 years with a value of $4 billion, and the site is in the middle bottom. You can see the whole team is at the International Biotechnology Meeting. We sponsor events and go out and meet with potential companies who are looking for a new site. And again, the workforce is a major component of that. Economic development, we founded the California Biomanufacturing Center, and you can see how prominent Solano College is featured on the economic development literature for our community. And let me end with my own story. This actually is my family. So my son in the upper left-hand corner went through the Solano College Biotechnology program. He went and got a master's degree at the University of Edinburgh, and he's now in England because he also met a girl there. And now he's employed in the biotech field in England. My youngest son, and there he is at age 4, in the lower left-hand corner. When he was 4, he had he was deathly ill. He was about to die. He was going to die that day. He was airlifted from our local emergency room to Oakland Children's Hospital. And my father-in-law said, look, Jim, you've just got to face it. There's no surviving this. He had meningococcemia. And he had the fulminant form where every clotting factor in his body went off. And if you think of the deepest bruise you've ever had, he was that way over 100% of his body, except his fingers and toes were turning necrotic black. And they came to me and they said, we'd like to try something. We'd like to try a thrombolytic. And I said, a thrombolytic, that's what you need. He's got blood clots. Thrombolytic is an enzyme that digests blood clots, and you have to give him tissue plasminogen activator. And they said, you know what tissue plasminogen activator is? There's no drug I know better than that. That's a drug I worked on when I was on sabbatical at Genentech. You have to give them TPA. It's an off-label use. I have to sit you down, and I have to tell you what the potential side effects are. I have to tell you I can first give them my. And so anyway, to cut to the chase, they injected TPA. It digested all the blood clots, saved him from certain death, and there he is on the right, Jefferson DeClos, MD. So if you want an advocate for biotechnology, you can find no better advocate than me. If you want an advocate for CIRM, you can find no better advocate than me, because at the end of the day, what we are talking about is alleviating human suffering. The headlines are that the blind will see, the deaf will hear, the disabled will walk, and that isn't an empty promise. And again, I am a recipient of this technology, of investment by the state of California. In this case, the
citizens of California into research, and not only research, into development and into workforce development, to have the people that produce the medicines so they are available, and at the same time, those same people coming from modest backgrounds will have a career, not a job, but a career that provides a livable wage, and a career that provides opportunities for upward mobility. I'd like to introduce my students, and let me start with Jen.
Hello, can you guys hear me okay? Good morning. My name is Jen Mayo, and I'm a former student from Solano Community College, where I graduated in May 2025. I got my bachelor's in biomanufacturing as well as my cell and gene certificate from there as well during my time there. I'm going to start off with a little bit of. I'm going to click for you because we want to get to your slides. For sure. I'm going to start off with a little bit of background about myself. So I come from an immigrant household where my parents and my grandparents immigrated from the Philippines. It was around when my mom was at the age of 16 and her sister, which is my auntie, at the age of 12. But my family came from a poor part of the Philippines where they had to collect recycling and just save pennies and pinching pennies just to be able to come to America and live the American dream. And they were able to finally immigrate from there at my mom's age of 16. So they finally landed in California and resided in Fairfield. But within those 2 months that they came, they found out that my auntie had leukemia, and she was diagnosed with leukemia, and soon, shortly after that, she passed away from leukemia. So seeing that was really detrimental for my family, and that carried a lot of trauma for my family and still affects my mom to this day, losing her sister at such a young age and knowing that the hard work that my family was working towards to be able to live the American dream was kind of just taken away from her. So after that, my family started to establish themselves in America. And at the age of 26, my mom had me and actually named me after my auntie Jennifer. So yeah. And after that, I was born. I always knew that I wanted to get into researching cancer and just kind of why this happens, because I've seen the amount of pressure and trauma that it built in my family, and kind of like this weight that we just carried because we knew that that happened. And I wanted to give back to my auntie in a way where she would be able to live that American dream and actually get the opportunities that she had. So I knew that I wanted to get into research and being able to work towards something that ultimately put this big hurt in my family. So as I went through my middle school and high school, I was like, oh, what can I do? I can become like a scientist, et cetera, et cetera. And then my cousin actually went through Solano's program before it was actually a bachelor's and recommended me to it. He works at Genentech now. So I was like, oh my God, this is perfect. So I started my career at Solano at, what year is it? 2021, and was able to start that. And during that time too, my grandma, which is on the right, got diagnosed with B-cell lymphoma, and it felt like my weight was kind of just like pulled under me at that point, because it was like, oh my God, this is like happening again, and we don't really know why. So during that time, it just motivated me even more to be able to just push through education and being able to just work towards this. And fortunately, during my time at Solano in the year 2023, I was able to be part of the CIRM Compass grant, and I was able to also be placed at an internship at the UC Davis GMP facility. So during my time at my summer internship was in 2024, I worked for the summer at the UC Davis GMP facility, and it was amazing just being able to see the hands-on, boots-on work that people were doing to be able to tackle these illnesses and everything. And it was just kind of like a full circle moment for me, just being able to be like, wow, like I'm being able to be part of this and actually work on things that were putting pressure on my auntie and my grandma. And then after my summer internship, I stayed in touch with the GMP facility at Davis and was finishing my academic works and at Solano. And after graduating, I actually was able to be hired at the UC Davis GMP facility where I currently work right now. And it's just amazing to see that I'm able to kind of give back to my family in a way that I've always wanted to do. And it just really means a lot, especially with the support that CIRM has given me and Solano. So I want to say thank you.
And I misspoke, not my student, a graduate. Here's another graduate, Joel.
Hello, all. My name is Joel Sines. I was into the program around from 2022 to 2024. A little background about myself. I come from a pretty hefty family. I'm our second youngest out of 7 siblings, and I'm the first graduate out of the 7 siblings. We are first generation as well. So going into this program, I was a bit fearful. I didn't have guidance from really anybody in my family, but no, going into college, I pretty much already knew I wanted to do biotechnology. And I got introduced to the program, and just knowing that the support system that was there allowed me to kind of bridge my way to where I'm at today. So I really want to start off with thank you to CIRM, because without CIRM, I wouldn't be here today. It built me up to the person I am right now. It allowed me these opportunities that I would have never gotten otherwise. And I did my summer internship at the UC Davis GMP facility as well in the summer of 2023, I believe. And again, that allowed me the opportunity to kind of bridge my knowledge and bridge that to work industry as well. Not only did that kind of help me frame my confidence, again, similar to Jen, I stayed in touch with the director because I kind of found an environment that worked for me. I could see the benefits to other people. And again, with cancers and stuff, this stuff is very like debilitating to families just all across the nation. And I kind of figured it hurts to see all around. So I figured why can't I do something about it? So I took that initiative as well. And it really just helps knowing that now to where I'm at today is since I've been working at the GMP facility as a manufacturer for about 1.5 years, it has helped me knowing that I have saved dozens of lives in this 1.5 years. Whether it's someone's mom, dad, brother, sister, whoever it may be, I know that I am able to help with that. And if it wasn't for CIRM, I can't say that I would have that opportunity and be in the position that I'm in today. So again, I want to say thank you for that. Yeah, the experience alone is just it cemented everything. The knowledge I've gained is you can't take back. It's great. It's forever expanding. I went in there fresh out of the bachelor's degree program at Solano Community College, and as of today, I feel like I'm a completely new person just from the knowledge I take away from my coworkers, my managers, my supervisors. It's super reinforcing environment that you just can't, it's hard to replace. And again, it really wouldn't be a thing if it wasn't for the CIRM grant. And being that I was, I believe, one of the inaugural cohorts for the CIRM grant at Solano Community College, I felt a little bit more pressure because I wanted to set a reputation and hopefully build a pipeline from this internship to full-time jobs, or careers, I should say, because it's pretty much a career. And I would like to hope that me and the first cohort of this inaugural grant became a good stepping stone for the cohorts after me, such as Jen was right after me. I got accepted as a full-time manufacturer. Jen got accepted as a full-time manufacturer, and it's every day we are giving it our 100% effort to save a life, whether it's one at a time or dozens at a time. It's just one life matters to somebody. And I think that's just the principle that I will always represent. And again, I know I've probably said it a few times already, it doesn't happen without CIRM. And so knowing that I can make a difference, knowing that Jen can make a difference, it goes a long way. And a lot of thank you goes to the professors as well at the program too. They built me into this person I am as well. And without them and without CIRM, I don't get here at all. But I would just like to thank all of you for the opportunities that you've given me. And I think it's really important to keep pushing it forward.
Thank you. If you notice the pattern that they typically get a job at the institution where they were. So they were both interns here in downtown Sacramento, and now they're full-time employees there, making more money than I made when I started. And likewise, all 4 of our CIRM interns that interned at the Stanford Laboratory of Cell and Gene Medicine are now employed there. And I have a late addition. Let me introduce my colleague, Mike Silva. And what's important about Mike is he's a faculty member in our department, but Mike, during his schooling, was a CIRM scholar. And that's also true of Miracosta College, K.L. Wong at Miracosta College, who's now supervising CIRM scholars. He himself had been a CIRM scholar. So in a way, it's the next generation, and it's the culmination of a long-term effort and a long-term investment. And let me just ask Mike to say a few words.
Thank you. Ditto. I'm sorry. My name is Michael Silva. I actually attended my graduate degree at California State University. They had a CIRM Bridges program. And I've always felt that science has the ability to save multiple lives, not just to the products we serve, but for the workforce that it's actually able helping to put food on the table. I earned my doctorate degree. Is there any UCLA fans out there? Or alumni? I just want to say fight on, because I got my doctorate from USC last year and actually interviewed Joel as part of that. And it just it's just been pretty it's been pretty amazing and surreal from someone that came up with, honestly being trying to find police at one time really liked me, but for the different reasons. And back in the 90s, and just really being able to navigate those complexities in our society and wanted to do more, not for myself, but for my mom, who was struggling through a lot. And, and I ran into somebody that was a phenomenal teacher, Jim DeClo at the time, and just kept believing in myself, helping other people came around to help me go along. And then I got this $40,000 scholarship to do stem cell research the second year of my master's degree. I did that at City of Hope. I got hired on as well to finish that up, published. Some was off of my mom, came back home up here to NorCal, and she ended up having Alzheimer's. She passed away a couple years ago. But the ability for me to be able to give back where I came from, really involved in the community right now, full-time faculty member at Solano Community College. And one of the things that we do that was, I believe, mentioned already, that with the CIRM support, we're able to bring our students together to find ways to bring and introduce science back into our communities where it counts most. That's our most underrepresented youth, from elementary school all the way to high school. And our goal is to make sure that every kid knows that if they want to pursue something more, here's a great opportunity for them to do it. And the lives that you save won't just be the patients that you serve, it'll be through the revenue that you generate, so you can be like families like mine who don't have to worry about how they're going to put food on the table for the first time in their life. So I appreciate your guys' support, and thank you all.
And let me also say that Mike felt so empowered to contribute to his community. He's now vice mayor of the city of Vacaville. And so I've been told that there might be questions, although we may have run out of time. Go ahead.
May I? Yes. So thank you all so much. I'm Marguerite Casillas. I'm a patient advocate here at CIRM. Pretty new here. I've lived with MS for 23 years now, and for 21 years, I injected myself with a biologic every other day. didn't necessarily think about, what it took to put that together. When that box arrived at my doorstep packed in ice every month. So this was just such a terrific opportunity. I know you thanked us. I want to thank you all as well for what you're doing. And also to the communications team for continuing to bring us these amazing opportunities to hear from folks who are benefiting.
Yeah, thank you, Amy.
I just want to congratulate you, all of you. This is an amazing thing, and I'm really glad. Thank you for putting this presentation together and reconfirming for us the import of this program and this initiative. It's a testament to the state of California, to what it can do.
And I just want to note for the rest of the board members, we have one more presentation to come, and then we'll break for lunch. We've invited both presenters to stay around and enjoy a bite or visit with you. So if you do have more questions outside of the time available, please take advantage of that time.
And thank you. And you could sense the gratitude that all of us have
Yes.
toward CIRM and toward all of the work that you do. Thanks once again, and thanks for inviting us. Thank you, Amy.
So thank you. I'm Diana Farmer, and I have to say that's a really hard act to follow. I also want to say that I'm really looking forward to paying my taxes this year because I'm reminded by listening to that previous presentation of how broad the reach is of this amazing program. I've been a beneficiary for the last decade now. I feel like I'm a partner with many of you in the room. I've worked with you over the years. But it really, the previous presentation just pointed out the breadth of the impact of this program on our region and our state. So it's really a privilege. So I'll do my best. So I'm going to just share with you my story from the barn to the bedside and the journey that I've had the privilege of being on with all of you that started essentially with a phone call from an NIH program officer almost a decade ago, who said, withdraw your grant. I'm like, what are you talking about? The word fetus is in there. What are you talking about? I'm trying to help fetuses. He said, nope, not right now. Withdraw your grant. Good luck to you. And that's how I got introduced to CIRM. So let me just set a little context. So I am a fetal surgeon. I operate on babies in the womb. This actually has been going on for 40 years now. It's not brand new. It sounds amazing. It started at UCSF when I was initially a trainee. And it fortunately started out pretty slowly and carefully. But if you think back to when your grandparents had children, what did people do? They counted fingers and toes and kind of hoped everything was okay, and that's all we could do. But then along came a disruptive technology, ultrasound, which allowed us to look inside and see the fetus for the first time. And originally, we just used it to help identify whether or not a baby should be delivered by C-section because they were breech. Maybe try to figure out if it was a boy or girl. That was not very reliable in the early days. You shouldn't have decorated with pink or blue. But then we started to occasionally see things that were abnormalities, something that was unusual. Again, not much that could be offered other than giving advice to families on how to deliver. But this ultimately led, after many years of research, to asking very simple questions. If we did the same operation that we did after birth, if we did it before birth, could we change the developmental physiology? Could we change the science and the biology and change the outcome for children? So the first decade or so was spent figuring out how to do fetal surgery, how to make it safe for the mother, because it seemed like a crazy idea. But ultimately, we were successful. And that led to learning how to do this more safely for women, and being able to do non-fatal fetal defects. The original ones were all just fetuses who are either going to have late miscarriages or fetal demise at birth. But ultimately, we were able to think about non-fatal fetal defects. And that's where I came in and got involved in what was not a fatal defect, but in many respects, a very neglected birth defect, but relatively common. So 4 children are born in the United States every day, and more in our low and middle income countries. If you've ever traveled in places and seen kids in little red wagons with big heads, those are likely children with spina bifida, not very well treated. It is a lifelong defect. These children are paralyzed from wherever the defect was. They have bowel and bladder dysfunction. They, frankly, ultimately often die now of renal failure. So huge, huge annual cost, the care of these children, multiple operations over the course of their lifetime. And somewhat neglected just because no discipline owns it. You know, the neurosurgeons didn't go into neurosurgery to take care of spina bifida. They want to treat brain tumors. The orthopedic surgeons didn't go into orthopedic surgery to take care of club feet. So it kind of fell between the cracks, and really no work was done. If you look at the history of research from like the 1930s to about the 1980s, when we first started thinking about spina bifida. So what we know about it, just a little bit of science, is that fundamentally it's a 2-hit hypothesis. The first is that failure of the neurotube to close during development. That happens at about 6 weeks gestational age. All of that early developmental work occurs. And what that does is it leaves a portion of the spine, the spinal cord, not covered in a bone. Put your fingers in your back for a minute, and there's a bone, there's a backbone. You can't just reach your spinal cord. The rest of our organs kind of hang off our spine. So we wanted to attack the secondary damage, the intrauterine damage that occurs from both chemical toxicity of the amniotic fluid as well as just mechanical trauma of that spinal cord rubbing up against the uterine wall. And that was initially done by asking the very same simple question. If we close that defect before birth, if we did the same operation before birth that we do after birth, could we get an improvement in the outcome for these children? And that led to what was an NIH-sponsored clinical trial, started in 2000. This is when my journey of all of this started. This was a randomized controlled trial. Parents had to pull a number out of a hat to decide whether or not they'd have surgery before birth or after birth. It took 9 years to get this study done. It was done in 3 centers around the United States, but it was stopped early for efficacy. As you all know, that's pretty unusual. They stop a trial early because it works, because they think it's unethical to not allow this therapy for other people. That's what happened, because it reduced the need for ventricular peritoneal shunting by half. But what we also noticed was that it improved the motor function a little bit. No one really thought you could do anything for paralysis. However, still the majority of kids were paralyzed and couldn't walk. So that is what took me back to the lab. Hey, if there's a little bit of room for improvement, if there's some plasticity in that fetal spinal cord, maybe we can get more. And this was at the early days of stem cell therapy. That work was published in the New England Journal. And interestingly, we talked about funding at a CIRM event last week. This became paid for by insurance companies overnight because the impact was so impressive. It also didn't cost very much, right? They had to have the operation anyway. So the only thing was different was they were having it a little bit earlier. Separate topic. But again, took us back to the lab to ask the question, could we do better? And we hypothesized that we could further augment that already, if you think about it, the fetus is already not even regenerating, it's generating all of its cells and organs and fingernails and eyeballs and all of that. Could we augment that system and make it even better by incorporating some kind of biologically active stem cell at the time of doing that fetal repair to really focus on improving that paralysis part of the story and the bowel and bladder function. We didn't know exactly what kind of stem cell. We looked at iPS cells. We spent about 5 years getting it wrong. And finally, we identified, partly because I thought we would need an autologous solution, that no mother would let somebody else's stem cells into their baby, into their fetus. It's interesting how society has changed in the 10 years since this started to now. No one even thinks stem cells are weird anymore. But a decade ago, there was a lot of confusion, and when CIRM was started, a lot of fear about stem cells. I'm happy to say that society has moved along, in no small part, I think, to the way California led the way. But nonetheless, as a fetal surgeon, I was always interested in the placenta. Here's an organ that grows and becomes senescent in just 9 months. It kind of knows how to stop. And we identified some unique cells in the placenta that, if grown in a unique way, had some unusual effects. We discovered one, that they grew a lot faster. Their doubling time was much greater than a typical bone marrow cell, so they were different from typical bone marrow stem cells. They additionally had some multipotency. They were able to grow into 2 lineages, the ectodermal and mesodermal, a little bit different, again, than mature adult bone marrow mesenchymal stem cells. So unique characteristics. And particularly when sort of engineered and cultured down a neurogenic pathway, we could identify that they had a unique cytokine array. They had the ability to rescue stem cells that were growing in a test tube from apoptosis, from damage. Our stem cells could rescue that damage when we tried to poison cells in a test tube, neurons. And again, it secreted, I like to call it the magic stem cell juice, but particular factors that are known to be important in nerve function and nerve development. So BDNF, brain-derived neurotrophic factor, is one, and human growth factor is another. So with that sort of in the laboratory work, we went back to what has always been the way we could study the biology of fetal diseases, which was in fetal sheep. To be honest, that's what got me to move from San Francisco to Davis. The availability of the vet school, the large animal facility, and also at the time, it happened to be the only university that had a GMP facility, again, thanks to CIRM, in Northern California. So what you can see is on the left there is a picture of a human fetus with spina bifida. So in utero, but you can see the little back of the fetus, and you can see that portion that isn't closed, and that's the spinal cord, that little thing that looks like kissy lips in the middle, that exposed spinal cord. And the other one is the sheep. This is a model that we create, and it's very similar in its appearance and very similar in sort of what happens in utero to that. So we took the same repair that we did in the mom's trial, just added stem cells to it. And what you'll see is that we improved paralysis. We ultimately were able to test different concentrations of cells. And I'm hoping my videos work. We have a fancy double click technique here. Aha. By the way, your IT guys are brilliant, just for the record. So here you can see these were twin lambs in the same mother, the same ewe. The only difference is the one that's walking got stem cells, and the one that can't even bear weight, you can see their little scar from when they were that operation before birth. You can see it on their back. Can't even bear weight, which breaks everyone's heart. I am convinced that if human babies needed to walk at birth, someone would have solved this disease decades ago. Again, this is the lamb almost normally walking that received the stem cells. That was the only difference. Same mother, same uterus, same environment. Like, we couldn't have had a better first experiment. Plus, it's really great to have your first experiment be successful after 5 years of things that didn't work. So this is, in addition, by the way, we had one of the students in my lab, by the way, we've had several great students from Solano Community College in our lab, so thank you very much. Great CIRM Bridges students. I felt very much like a failure. He came in as a pre-med. He left wanting to be a veterinarian. I thought, oh, I guess I'm not a very good role model. But nonetheless, turns out he found out that bulldogs are born with a naturally occurring kind of spina bifida. The breeders don't want you to know necessarily because most of those dogs are euthanized, but nonetheless, there are rescue farms where there are little baby bulldogs in little bulldog diapers and little bulldog wheelchairs that are made, and they are alive and well. C, that picture there, is an MRI of a human skin-covered spina bifida-like defect, and D is the dog, the canine defect. So just as a proof of concept, you can't do fetal surgery on bulldogs. They don't have a prenatal diagnosis system. But after birth, we took some dog placental-derived stem cells. We cultured them in our same sort of proprietary system. And we're able to discover that, and we're going to try this again. You can see this first bulldog walks on his hocks. I had to learn a lot about how to evaluate the ambulatory status of dogs, and somewhat discoordinated there. But 8 weeks post-op, so this is postnatal, you can see this is Spanky. He's more on the pads on his toes. Usually as these dogs get bigger with more weight, they become more disabled. So armed with this sort of proof of concept, we came back to CIRM, showed them our preclinical data, and got the next set of funding after our early investigatory funding to go ahead with an IND and do all of the preclinical work that was necessary for an IND submission, which we ultimately were able to get investigational new drug approval from the FDA to do a clinical trial. So in addition, actually, CIRM was even stricter than the FDA and wanted to know that the exact cells that we now made in a clinical grade environment. So we made them according to the stringent manufacturing that you heard about earlier by our great Solano students in our GMP facility, that those exact drugs, the same stem cells that we were intending to give to the humans, worked in our sheep, which they did. As I say, CIRM was even more strict than the FDA, which is good training, by the way, for talking to the FDA. So again, the animals that received the stem cells were able to have this is the sheep locomotor rating score. I have to admit, I think this might be my most cited paper. I never thought I would be famous for how sheep walk, but you take what you can. So again, we submitted an IND, got FDA approval, and thank you very much to CIRM, got a CLIN 2 award to do a combination phase 1/2A. Again, because it's a rare disease and because it's fetal surgery, you would never do this on healthy volunteers. So it's a combination phase 1 and 2A study together. We also designed it, just as a side note, because I've always thought that maybe we could move the FDA to be more liberal about rare diseases. So we designed our phase 2A with the statistical power to potentially be switched into a pivotal trial to show a significant difference. And I think I learned from the first time around that if the impact is powerful enough, you can be persuasive. Now, we still may not be able to talk the FDA into it, but that's a side note. So nonetheless, we went on, and I have to say that my scientific partner, Aijin Wang, who's pictured there, none of this work could have happened without him. He was a postdoc at Berkeley when I first met him, and he's been my partner in this forever. We've called all good trials need an acronym. We call this the Cellular Therapy for In Utero Repair, the CURE trial. We're hoping that there are more stem cell trials, not just for myelomeningocele, but other birth defects in the future. The first phase of it, the first 6 patients were considered safety primarily because the FDA insisted that all of the patients be delivered before we could treat the next patient, to make sure that we were not creating little tumors, little alien things in these fetuses, because there had never been a placental-derived stem cell put in a fetus in the central nervous system before. So the FDA was appropriately concerned about safety, but we will continue to watch those patients, and we will use their data for the efficacy evaluations. So 35 total patients will give us statistical power to show a 20% improvement in the outcomes. And this is just kind of for fun. Lots of tricky things when you're doing something new. The OR wouldn't let us store a refrigerator in the OR area, so we had to push a refrigerator across from the GMP facility to the OR every single time we treated a patient, so that we could keep the cells and sort of even temperature control along the way. And of course, since we are in Northern California, our cell transport mechanisms, if you look carefully at those black bags there, anybody from Napa recognize what those are? Those are wine bags. So, but they did just fine. We also tested the temperature along the way. We'd make 3 batches just in case somebody dropped one in the operating room or something. We'd have a backup, which is why we wanted the refrigerator there to keep the cells. Just last week, so the timing of this was perfect. Thank you very much. The Lancet published our safety data for the first 6 patients, reporting essentially no safety concerns related to the cells at all. We were a little concerned that the anti-inflammatory effects, some of the things we know about, might interfere with wound healing or some other things. We had some concerns. We had no problems, knock on wood, on the first 6 patients. So I just want to thank the team. As I say, this couldn't have been done without moving to Davis at the time. My friends from San Francisco, I think, thought I was a little crazy. They may still think I am. But the ability of having the resources of the veterinary school, the large animal facilities, the GMP facility at the time really allowed this work to continue in addition to CIRM. We had to teach the operating room how to do this work, develop the fetal treatment program. It really has been great. I do want to share just one photo and remind everyone this is not validated data. We really aren't going to know, unfortunately, the outcome, the true outcome for these children. Do the cells make a difference until they're about 2.5? Now, why 2.5? Yeah, how many of your kids, right? So kids walk anywhere between 1 and 2, usually. In fact, 99% is of all children will reach the walking milestone by 30 months, by 2.5 years of age. So that is the sort of standard developmental biology marker that's there. And toilet training. Most kids aren't toilet trained in this country until they're about 2 years old. So again, it's going to take us time to know the definitive answer. But I will say that's okay. I had some extra slides. I don't know if you want to. The baby, that might help you. Well, all I can say is that, and again, I'm a little nervous because I don't like sort of trafficking in unofficial data, but the babies have been born. You saw that previous picture of the baby kind of with their legs folded underneath. All the babies have been born being able to wiggle their toes, move their ankles. Now, again, there's no correlative data. We don't know that wiggling your toes when you're born means you're going to walk when you're 2.5, or that the level that was expected is going to be improved over the level that the functional level is greater than the anatomic level. So it's going to take time. I keep telling the FDA to be patient. I keep telling CIRM to be patient. We're going as fast as we can. But we are incredibly optimistic that we might be able to get all these kids out of a wheelchair. And that would be pretty exciting. So thank you. Did you find it? Do you want to pop it up there just to, I will say we were all pretty excited. And our CIRM, actually usually it's CIRM Bridges students that, let's see if there you go. So way different than we have seen other kids with spina bifida. Unfortunately, there's no data on what newborns are like spina bifida kids because no one thought there was really anything you could do for these children. So we too are now sort of creating some of these standards just to evaluate these kids. But we are excited about the preliminary data. Some of our early patients are reaching the 2.5 and 3 year mark. And so we're collecting these data and looking forward to getting the rest of the patients enrolled. So thank you again, and happy to take some questions if there are any. I think we have a minute or two. Yes, sir.
Dr. Former, hi. I'm curious, a couple of things. How often are you asking the children to come in to be evaluated after? And secondly, do you have any, I'm speaking as a urologic surgeon, do you have any solid hopes for not just mobility improvement, but for their bowel and bladder function? Because that's what most of the other physicians, once your work is done, we deal with them for the rest of their life.
Absolutely. So thank you very much for that question. It was not a plant, I promise. We bring the patients back at 3 months, 1 year, and 2.5 years, specifically for urodynamics. So we're getting urodynamics in all of these children. Thank you very much. Actually, the biggest cause of death now that the Chiari malformation has been largely taken care of, that's the head part of the problem for these kids, is renal failure. So protecting their bladders is really important. I didn't show the picture, but we have great data in the sheep of the sheep peeing and pooping. So I had a whole army of volunteers out there at the barn recording measurements of how they were standing when they were peeing and what their poop looked like. So I will say, thankfully, my most cited paper is not the sheep pooping score, because I might have to retire. There's promising data in the sheep. I have tried to, I don't want to promise things I have no idea about. We did not see an improvement in the bladder function in the original MOMS trial. So just doing the regular surgery did not show an improvement. I would love to see an improvement in the bladder. I think we can get there, whether it's this version 1.0 of stem cells or whether it's going to take the next iteration. As all science, we grow on the backs of the people who went before us. But I think this can be solved. I think we can restore bowel and bladder function as well eventually.
Vice Chair, and then Joyce?
Two things.
And Chris on the phone. Sorry, just wanted to insert that.
Two things. I remember it was either 2018 or 2019. We went up to Davis. I was with Dr. Milan at the time, and we saw a presentation that you gave. And I remember walking away and thinking, if we don't get refunded, I don't know what happens to this. And seeing those videos and just thinking, this, CIRM makes all of this possible. So thank you. And thanks to the voters of California, which is where I go next. How long till we see this,
Yeah, so it's very interesting. Our initial enrollment, our first 6 patients was very fast, right on time. I'm embarrassed to say it's the first time in a decade that we have been late on a milestone with CIRM. For all those other little grants, I was so proud we were like ahead of time and under budget and really did well. And now we have 4 pregnant patients in a row. We're trying to figure out how to get in. So I cannot predict when people get pregnant. It seems to happen in batches for reasons I don't understand. I look back and I think, what happened 9 months ago? So it's complicated and difficult, and it's hard for me to be patient. I'm getting impatient. But I think we're back on track. We've now, the 20th patient has been operated on, and she's due to deliver in a few months. So we have 15 more to go. So I'm hoping that within 3 to 4 years, we'll actually have the definitive data.
I have 3 speakers now. It's going to be Joyce and then Kim and then Christine.
Thank you for an amazing presentation and inspired work. I guess I have 2 questions. First is, can you comment on any possible complications in the mother in terms of the in utero fetal surgery? And I would imagine the stem cell administration may have less of deleterious effect because it's coming from the placenta. I'm assuming, is it coming from?
So great question. First is the effect on the mother. So the issues related to the complications related to fetal surgery are all related to the fetal surgery and adding the cells to the back of the baby. So we topically apply a patch to the fetus, then we close the fetal dura, then we close the fetal skin. So the mother actually doesn't get any direct contact with the cells. We did, however, in the sheep do some safety experiments to just drop cells in the uterus just in case that happened. And was no evidence of any human DNA found in those sheep, those pregnant sheep when they delivered. So we were worried about that as well. I don't think there's a particular advantage to the placenta to the woman. And we now use off the shelf someone else's placenta rather than adding the additional risk of having to do it at the time would have been CVS sampling, go into the mother, take a bite of the placenta to use those cells. That was why there was a prohibition against fetal research, that we were using fetal tissues to do something. And that's why the original NIH reviewer at the time, which was about 8 years ago, cautioned me against submitting anything that appeared as though it could be fetal research. It was, again, so CIRM, it wasn't embryonic stem cells, but also the federal government wasn't funding fetal work either. So CIRM really stepped in and allowed this work to continue that would not have been able to go forward without CIRM. The NIH was not willing to even look at this kind of work. But the risks to the mother are all related to the operation itself and are pretty well defined, and I'd be happy to talk afterwards about it.
Sure. And I have a quick follow-up question, and I hesitate to even ask this question of a surgeon, but please permit me, I'm a primary care physician, so I always think about prevention. So given the data that preconception folic acid intake can actually reduce neurotube defects. I'm sorry. Given the fact that we have data that preconception neurotube, intake of folic acid actually reduces the incidence of neurotube defects, what role, if any, do you think CIRM has in expanding access to that knowledge about preconception care?
So great question. So the question again was about folic acid. So it's well known that folic acid supplementation, I have a great slide on this, reduced the incidence of spina bifida tremendously from, by I would say 80%. However, it never got it to 0. And we also know that even women who've had one child with spina bifida and who are on prenatal vitamins religiously for their next pregnancy and well before can get spina bifida. So it doesn't completely eliminate it. It just reduces it a lot. There's a lot of good science to understand why that happens, and we don't know yet. But that would be great. That would be ideal. We are involved in all of our outreach efforts. We are involved with just spreading the word about folic acid. It's interesting, the way folate was introduced to the public in the United States was by adding it to flour. So a lot of flour is fortified with folate. However, if you are from a rice-based or a corn-based culture, you're not going to get fortified flour. And plus, since Wonder Bread went out of style and fashion, I'm not sure any of us are getting the folate we would have gotten from fortified bread anymore. But it doesn't, the answer is folate has helped, but it has not prevented the disease entirely.
Kim Barrett. I think you two know each other, right?
Diana, thank you so much for your presentation. And I just want to go on the record as saying how wonderful it is to have a spectacular colleague like you. Can you perhaps, though, for the board, reflect on the importance of team science to accomplish the really impressive outcomes that you've had so far?
Yes, I should have lingered longer on the slide. Our lab now has about 70 people in it. There's a whole army of folks that we train. Most of them have been CIRM Bridges graduates who then stay on with us, and we train in the GMP manufacturing component of it. And when they are not preparing cells for a fetus, they are helping us do the science that's related to how the manufacturing would need to be if this were done in a commercial GMP level. We realized when we started, we never thought about the end game, really. We thought a lot about the cells, what would make the cells the happiest. And that was 2 days in a culture plate. This was a 4-day process when we started, and you had to do it at midnight. And so our technicians were sleeping in the lab. I mean, it just wasn't ideal and not practical. So they, in the off times when we weren't treating patients, have helped with some of the research on getting that down to a 2-day process. We hope to really get it to 36 hours so that it could be used for spinal cord injury or other things that we think there may be an application for. So that group is involved in it. Then all of the CRCs, the clinical research people that help and get to know the families, to follow up and keep families coming back for the follow-up. Once they get the magic cells, what's their incentive to come talk to us? So developing that relationship is really important with the families. And then just the fetal treatment team and the OR team and the people who have to counsel the families and all the work that has to be done. So it sounds like it's a complicated effort, although a lot of it is just the fetal surgery part of it, which is already happening. The stem cells shouldn't be, and that was part of what also was important in the paper, that not only was it safe, but feasible. It wasn't that difficult for sort of an ordinary neurosurgeon to figure out how to add cells to the regular repair. So thank you, Kim.
Thank you. And Board Member Miaskowski is our next.
Hi, good afternoon. Thank you very much for your presentation. This is not my area at all. And I was curious, how early in the pregnancy can this be diagnosed? And do you do the surgery as soon as it's diagnosed? And then the other question I had was, I'm wondering how easy it is for moms and dads to agree to do the surgery. Do you have challenges with refusals?
Great question. The first is about timing. So the diagnosis is usually made around 16 weeks. So often the first hint is in the state of California is mandatory alpha fetoprotein testing, which is usually looking for signs of Down syndrome and other disorders, but that's the first hint. If that's positive, that generates an ultrasound, a focused ultrasound. Otherwise, sometimes it can be picked up on the screening ultrasound that is usually done around 16 weeks, 12 to 16 weeks. And that then generates usually a referral to a maternal fetal specialist who can do some advanced diagnosis. The treatment, the surgery, happens between 22 and 25 weeks gestation most of the time. So mid to late mid-trimester. There's reasons for that. Ideally, you'd think, gosh, wouldn't it be great to start sooner, right? You could prevent more of that damage. And we've seen with electron microscopy in mice that the cellularity gets lost. The cells get degraded as over time in gestation. So that the earlier you could get there, the better. The problem is the technical difficulty of doing an operation that early in the pregnancy. We can't do it safely. We can't fool the uterus into thinking nothing happened and keep the mom pregnant. There's a lot of science that had to be worked out for that. Also, it has now, there was sort of a debate, if we did the operation at 22 weeks and something happened, would you resuscitate the fetus? Would you not? You know, this is right on the border of fetal viability. So these were complicated conversations. So I would say most of them happen around 24 weeks now. And most of the time, if there were ever an intraoperative problem, which again, we have not had, knock on wood, but in the early days of fetal surgery, it was more common. And for other fetal defects like fetal tumors, where the fetus themselves are ill, this fetus is not physiologically ill, just has a defect on its back. So I think that's why we've had more success in terms of preterm labor, et cetera, with these fetuses. So 22 weeks gestation, and we'd like to be able to operate sooner, but we haven't figured out how to do that yet.
And difficulty of parental consent, I think.
Oh, yes, and the difficulty of parental consent. I mean, the hard part is, frankly, and I'm talking to a mom later this afternoon, you are not, no pregnant woman is expecting this diagnosis. No, you know, no couple thinks, oh gosh, let's get pregnant and worry about birth defects. You know, everybody's expecting a perfect baby and it's a really happy thing. And, you know, they've sent the ultrasounds out to their families. And then, oh my gosh, they have a diagnosis they've never even heard of. And they have to make a decision in, you know, 2 to 8 weeks period of time. What are they going to do? Are they going to terminate the pregnancy? Are they going to, is there anything that can be done? Are they eligible? How do they find a fetal center? How do they find the CIRM trial? We've done a lot to how to get to families to get this information, because you've got to make hard decisions in a short period of time. So interestingly, pregnant women are also protected. There's special status, a vulnerable status in terms of informed consent. And boy, is that true. If I said to a pregnant woman that cutting off her right arm would make her baby walk, she would give me both her arms. I mean, it's just really, you know, a vulnerable population beyond imagination. They are hormonally wired to do whatever they can for their fetus. So it's tough, but it's a privilege to be part of their journeys and to give them hope. That's what CIRM gives, is even more hope.
Dr. Farmer, thank you. I know there are probably lots of you that have more questions, but remember that our Solano College guests and Dr. Farmer are guests of ours at lunch, which is now about to start, correct? So we're going to break for lunch then. Okay.
Thank you
Dr.
again.
Farmer, thank you so much.
So wait a minute. Okay. For members of the board on the phone, we will be returning at 1 o'clock. CIRM is about accelerating and lunch is no exception. So what we would ask is that we allow the board members in line first so they can get their lunch and get seated, then the CIRM team and speakers. And for members of the public, there is a restaurant right at the entrance of the hotel. So once again, we'll be resuming at 1 o'clock. Good afternoon, members. We are going to get started again in about 2 minutes.
We are coming back into live session here. Thank you, everyone. I hope you enjoyed your lunch. We are going to just jump ahead temporarily to agenda item number 12. We'll come back to 11 in a minute. This is the consideration of an amendment to our warrant co-funding program. The discussion will be led by Chief Counsel Rafael Aguirre-Sacasa. Rafael.
Thank you, Dr. Mbashani. Good afternoon, everyone. My name is Rafael Aguirre-Sacasa, and I'm the General Counsel, and I'm here to update you on the warrant program. I'd also like to thank Dr. Sean Patel and JT, who've also been helping me with the structure and the updates here to this program. What we're talking about today is a targeted refinement to the CIRM warrant program, which was approved by the ICOC in December of 2023. Before we get to the slides, I want to give you a quick update on the status of the program. At a high level, the program is working as it's intended. It permits companies to satisfy their co-funding obligations through warrants instead of cash, and this allows CIRM to participate in the upside of any successful programs. Where we've encountered challenges is with early stage companies that have not yet completed a priced equity financing. In those cases, the current structure uses an assigned placeholder valuation of $1, which we found can create distortions and make participation in the warrant program more difficult. So what we're proposing today is a narrow, practical fix by introducing a safe warrant hybrid. And a safe we'll talk about a safe in a minute. And that defers valuation until there's a real market financing, which preserves the same upside for CIRM in the future. Okay, now to the slides. Our original co-funding warrant structure, applicants could, again, satisfy the co-funding requirement by issuing warrants to CIRM in lieu of cash. As a point of reference, under state law, we, CIRM, cannot hold equity, and that's why we structured this as a warrant program. The core structure is that CIRM receives a stock warrant equal to the waived co-funding amount, and the shares are determined by dividing the co-funding amount by a reference price. For the reference price determination, it depends on the capitalization and structure of the applicant company. If preferred financing exists, we receive a preferred warrant with a reference price equal to the price per shares of the most recent preferred stock financing. For public companies, pretty straightforward. Common warrant, reference price is the average of the closing bid-ask prices or the closing price for the 10-day trading period ending 5 trading days before the issue date. In both those cases, it's easy to determine the reference price. In the last situation, where there is no preferred financing, under the current structure, we receive a common warrant, and the reference price is a default $1 per share, subject to adjustment and negotiation. The other key warrant terms are an exercise price of penny per share, 10-year term for the warrant, anti-dilution protections, and automatic exercises upon IPO and acquisition, which are pretty standard for warrant terms. As I mentioned, the implementation challenge has arisen when companies have not yet completed a priced equity financing, and that makes it hard to determine the right reference price under the original warrant structure. We initially used a $1 default reference price, which we have found, and we've heard from our applicants, so this is based on feedback that we have received, that it raises arbitrary evaluation assumptions, which can create potential distortions to ownership percentages, and obviously can create uncertainty for future investors. To address these valuation challenges, we are proposing a warrant program that introduces the safe warrant hybrid structure, which we'll talk about in a minute. And again, this would only apply to companies where a public market does not exist for its common stock and no shares of preferred stock have been issued. Any questions so far? Okay, we'll continue with the securities law update. Not moving here, folks. There we go. All right. The updated structure, to address these valuation challenges, we're proposing the warrant program introduce a safe warrant structure. The SAFE stands for a simple agreement for future equity. How this works is that we would receive a SAFE warrant, we would enter into a SAFE agreement with the applicant. The SAFE would be convertible into warrants equal to the waived co-funding amount, so similar to what we had before. The SAFE would convert into preferred stock warrant. Now, the difference here is that the warrant shares would be determined on the preferred financing round, whenever that happens at a future date. Part of that is that we, CIRM, would get an economic equivalent of a 20% discount to new investors on the sales price, giving us 20% more return on the shares, obviously. There would also be a side letter providing for information rights to make sure that CIRM is apprised of all situations and information that apply to the applicant. And there's also a most favored nation clause that means that we would get the same terms as other investors if there are better terms than what happened at the most recent financing. So we are protected on it moving forward. The key advantages is that allows startups without a priced equity round to participate in the program. We've received direct feedback from some applicants that they would not be able to participate under the current structure because they feel that it would cause them problems in the investor community. Using the safe price mechanics, which are widely used in the Valley and across the country, this aligns with venture market practices. Of course, it still preserves our upside protection as we would have had with the original warrant structure. And most importantly, it avoids a lot of friction in the whole process because it avoids the need to assign an arbitrary evaluation to early stage companies, which can create those distortions I've talked about. So to summarize, the updated structure maintains our ability to capture value. It preserves our warrant-based upside participation, simplifies the participation for early stage companies by deferring valuation until a real financing occurs. And finally, it aligns the program with current venture financing practices. To talk about, whoops, that went backwards. That's not supposed to go like that. And then the request is that the IC approve the proposed safe warrant structure for use in applicable funding programs where a company has not yet completed a priced equity financing. And I'm happy to answer any questions at this time.
Thank you, Raphael. I'm going to move that the board accept the request that we approve this change to the safe warrant structure.
So you're making a motion?
I'm making a motion, yes.
Second.
So that will open the floor to discussion. Thanks for that lucid presentation. I know this is dense material. Anyone have any questions of the JT?
I want to thank Raphael and Sean for putting this together and for sorting out that this is something that we should be doing. This is just for members of the board. This is an extremely conventional instrument in the financing space for early stage companies, and something that, so we're not reinventing anything here. We're just sort of taking things more into the mainstream. So strongly endorse this.
Thank you, JT. Scott?
And also, not sure where the feedback is coming from. The team had the opportunity to present this to co-chairs Kim Barrett's alternate Don Taylor and Hala Madnat, and walked through it as well. And they endorsed the approach that you see here.
Oh, okay, that's important news. Thank you.
Hala has her hand raised.
Hala has.
I
Yes,
had
I
my
said,
hand
hi, Hala.
raised.
Thank you.
Hi.
The floor is yours.
It was actually just to say,
But you're still on mute. You're on mute.
I'm not. I'm not on mute. Oh, Can you hear me? now, Hala. Yes, we Okay. can hear you. I was Thank like, you. No problem. I was like, I'm not on mute. No, I was going to say exactly the same thing, that we met and both Don and I agreed that this was really a good idea. So that's all I was going to say. That's why I lowered my hand.
Thank you, Hala.
Questions or comments from anyone else? Okay. Motion, comment, anyone, any comments from the public? Either in the audience or remote? You don't see any. All right. Scott, we can proceed to a vote. Thanks, Raphael.
All those in the room in favor say aye.
Aye.
Those opposed say nay. Any abstentions? And I'll poll the members on the Zoom. Monica Carson,
Aye.
Judy Chow,
Aye.
Isabel Larone,
Yes.
Mark Fisher Colbury,
Yes.
Judy Gasson,
Yes.
Jeff Golden,
Yes.
Rich Lahara,
Yes.
Pat Levitt,
Yes.
Hala Madnat,
Yes.
Chris Miaskowski,
Yes.
Marv Southard, Carol Watson,
Yes.
Kevin Zhu,
Aye.
and Keith Yamamoto.
Yes.
Great, thank you very much. That motion carries. Chair.
Thank you, board.
Thank you, Raphael. We're going now back to agenda item number 11. We've dedicated a substantial amount of time on the agenda for this lengthy discussion here, the consideration of program preferences and guiding principles. And I would like to invite JT, you want to
I think
lead
we're
this?
going to Mark first and then to me. Mark wanted to give some opening comments.
Okay.
Sure. Yeah, thank you, JT. Just as a reminder to set the stage a little bit, and I'm sure JT will amplify this as well, we're in an environment where due to lack of funding within biotech and with the impact of what's going on at NIH and other funding sources, there has continued to be a massive increase in the number of applications submitted to CIRM across the board for advancing research. That's point number one. Point number two is that as CIRM has continued its goals of improvement and acceleration, the strategic allocation framework that's been implemented that is been such an incredibly strong process because of the fact that it is derived on deep data, deep thought, and elements that a lot of people have not been able to consider in the past. It's actually, in my view, worthy of publication as a roadmap for other organizations for consideration. With those two activities having kicked off the allocation of funding in mind, that's then a condition where the mechanisms for determining, well, how do you make the proper decisions related to both the strategic allocation framework and the volume of applications coming in. And our first attempt at that was clearly based on doing the best job we could to create completely objective criteria that we called preferences that had a numerical score, associated with that. And whereas that had some effect in terms of ensuring that that was not subject to debate about, well, what do those numbers mean and how does that work in terms of the criteria that went into that. It caused a number of issues that a lot of people then responded to and said, hey, we need to make changes in terms of how we make that triage decision. With that in mind then, you'll see on the presentation two primary topic areas around that. And the first has to do with what we call the volume control, which will get covered in detail as to how to make those decisions with a full scientific review. So that is a review cycle that is a modification of what the GWG does as an initial step. It is loosely called positive selection, but for the deep inside baseball people, it's not the same as the positive selection is what's been done previously in other areas. In the perspective that there are a dedicated number of people with scientific background who have to review all the applications. You'll hear more about that in the decision process. So there's a mechanism then around volume control. The other one is the removal of the preferences, scoring and criteria, but rather a shift to what is called guiding principles. And that is helping to frame the objectives of the strategic allocation framework with directions about how to look at things. And so the key elements that people should be taking into consideration as the triaging is being done are transformational elements and other characteristics, along with a reframing of the communication of the word, affordability, in the context that caused a lot of problems for people, because the presumption was affordability just meant low cost. And there's a update on the framing for that discussion around access affordability, where the focus is on how it can access be achieved with the view of looking at access as what are the characteristics that are going to be helpful for somebody looking at a clinical trial design and other elements to be able to accelerate and enable commercialization of the clinical trial work as positive results come in. So with that in mind, you'll hear the specifics and the details about how the guiding principles and how the volume control is being managed. We've had 4 to 5 hours of discussion on the scientific committee, March 5th and March 19th around this. There's a very strong consensus that there is a real need for changing our volume control system and process, and there is a strong consensus that the new mechanisms they proposed are fully supported by the science subcommittee. So I want to make sure to get that out there front and center, that we're all in behind what the proposals are related to this. There were a couple of open issues that we felt were relatively small in scale for further consideration. But for example, one would be there's a numerical element to the scientific review for the first pass before leading to the full grant working group review on, do you look at a true scoring system, or do you look at a stack ranking of the proposals that those scientists are reviewing? And you'll see some information about that as well. So we didn't come to a final determination on that, but we felt that was very much on the margin related to what the underlying proposals are. So basically, the science committee then is fully in favor of this. There are other nuances of, clarification of the patient advocate role in this element and other things that you'll hear in the presentation, but science subcommittee is in support of the overall proposal, and we just wanted to give some characterization of that so that we didn't necessarily want to get into a long extended debate about, a particular wording or other things. But I want to ensure that there's adequate discussion for the board to make sure that the big moving parts and how this is going to work is understood. So with that, I'll turn it over to JT, and I know the team will do a great job, much better than I can, of presenting the information going forward here. So JT?
Thanks very much, Mark. For that was a very good, concise summary of many things that you all are going to hear about today. And I don't have any introductory remarks that could do better than that, other than to note that the run of show that you're going to have, Rosa is going to start with the presentation on the switch from preferences to guiding principles. Gil will follow that with a number of remarks about volume control. Dr. Kelly Shepard will after Gil, Rosa will come back and talk about how this shift into guiding principles will inform programmatic discussion at the ARS. And then after that will be Dr. Kelly Shepard talking briefly about the issue of the focus of the next DISC 4. And then finally, we'll close with Dr. Shyam Patel talking about the question of going from 2 PDEV rounds a year to 1. So that's what you're going to hear. All of this, of course, keep in mind, is in our never-ending quest to produce the best possible product to benefit patients in the state of California. And we think that all of what you're about to hear is a major step forward with respect to all of the topics to be discussed as we proceed. So without any further ado, Rosa.
Thank you, JT. And thank you, Mark, for the very good summary of, and one point that you've made it several times, I want to acknowledge it, is that we should publish the strategic allocation framework, the process and the outcomes and the learnings. This is part of the learnings. And I promise you, we'll do this. So as JT mentioned, as Mark mentioned, this item deals with the response that the CIRM team has around the discussion that we had about the preferences that started already in December. Before I go into this, I have to thank a lot of people. So I would like to start with thanking the full science subcommittee for all their time, support, the discussions, the extra hours back and forth that has been very important. Secondly, I want to thank my colleagues, especially Dr. Shyam Patel. I don't know, Shyam, you can say hi. You're going to say hi later. But Shyam stepped up to present during both science subcommittees when I was not available. And he's been a great partner in crime in developing this. I also want to thank the 2 other leads of teams, Dr. Kelly Shepard, who leads discovery and education, and Dr. Joe Gold, that leads the clinical development team, for their joint forces in developing these answers and going through the guiding principles, etc. Dr. Sarah Taylor and Dr. Liz Noblin, as you can see, there's a 50-page summary memo that requires a lot of coordination, and I want to thank them for wrangling everybody as well. My partner in crime is Gil, Dr. Gil Zambrano, who we are going to
tag team in this presentation, but him and his team also. I want to thank my colleagues at the executive team and really everybody else, and obviously JT for his leadership. Am I missing anybody? Hopefully not. So without less further ado, I'm just going to get started. The first thing I want to start mentioning is that we are not stepping away from, as Mark said, from the strategic allocation framework. We are actually providing a refinement of how we implement this. As JT noted, in the last few months, the board has provided extensive feedback around the preferences and the preferences-based evaluation process. Some of the things that we've heard is that we cannot fund all projects and that the use of preferences can filter and exclude some modalities, classes, or innovative projects, and that applications, as a consequence, are being triaged without any scientific evaluation. These are some of the things that we've heard. We've also gotten feedback that we should tailor the guiding principles for each program. And one of the things that we heard was at the beginning, in January, we presented the start of the guiding principles. And in the work that we've done in partnership with the Science Subcommittee, what we've done is we've been refining them to adapt to each one of the programs. And specifically, what Mark was mentioning about accessibility and affordability, one of the items that came out of the discussion on March 5th that went into March 19th was the focus on PDEP and CLIN 2, that there was a terminology issue that we should focus on access rather than affordability, with clear expectations for applicants, stage-appropriate expectations, right? And then there were concerns around the guiding principles and preferences for discovery programs. The message was that we need to be specific to the discovery, and as you will see, we've been specific. And then, as you can see, more transparency and stronger communication of preferences needed. My colleague, Amy Adams, has been at the helm now for the past, is it 6 months, Amy? Yeah, and she's like breaking ground. And as you can see with the memo and all the letters and everything, there is actually communications plan as well. And we will be, depending on what comes out of today, there will be communication to the applicants and potential applicants in the upcoming changes. And then the request for data to inform the discussion. The memo has appendices that contain the data of how the preferences apply to the programs, what were the outcomes of applying the preferences, and there's also portfolio analysis in the memo. So, as a recap of the preferences flowed from the guiding principles, and the guiding principles are basically how they delineate how we will fund, what our programs will fund. Basically, a guiding principle will tell us whether we are going to, how are we going to get to accelerate later stage clinical trials in our portfolio, or to discover novel mechanisms. The way that we do that is through shaping our programs through these guiding principles, right? So when we presented the preferences, they had two goals. There were two separate objectives. The first one is that they were going to help us to prioritize what we fund. And the second was that they were going to help us control volume as well. However, it kind of backfired because the volume control, it led to some limitations, as we presented in what we heard. By applying prior to scientific review, there was some unwanted filtering that happened, and it's reflected in the analysis of the data that we got through applying the preferences. And then, by having preferences, we were also going to require iterative cycle-to-cycle changes, which was not going to be feasible in the long term. So what we propose is to remove the preferences themselves and also as they apply to volume control measure. Instead, the intent is to go back to the guiding principles, which we have always been in the background shaping our strategy. We just never like specifically spoke about them in the way that we are talking about today. So the goal today is to ensure that the board has agreement on how these principles shape what we fund for each of the programs and how do they apply at each step of the application life cycle. Throughout the presentation today, with Gil and I, we are going to reference to the steps in this slide, the volume control, the merit review, and the funding decisions. The guiding principles are embedded in each one of the steps. And the presentation is really about how are we applying these guiding principles in each step. So just for a reference in terms of the terminology, I want to go through each one of the steps. The volume control step focuses as well as reduces the number of applications to allow meaningful scientific review for each one of these programs, that is for the PDEP and the CLIN2. The second step, the merit review, is that once those applications have been forwarded to scientific review, the grants working group will review those applications and will provide recommendations to the board. And the third step is how the board makes those funding decisions based on the recommendations from the grants working group, but also from programmatic factors that will be provided to the board. And other information like budget available, et cetera. As you've seen today in the DIS4, we had to make some tough decisions, right? So today we are going to go through each one of the steps and discuss how the guiding principles help us shape what we fund. We are seeking today from the board two things. The first one is feedback as well as agreement on what we should fund for each one of these three programs, that is for the PDEP and the CLIN2. Effectively, what are the guiding principles underlying these programs? And we are going to present them for each one of the programs. Secondly, we are seeking board feedback on ensuring that the guiding principles are applied by the right party with the right information and at the right time. And then, as JT mentioned, we are going to have some updates that are separate from this guiding principles discussion with regards to the DIS4 and the PDEP program that will be presented by our colleagues. So I'm going to stop in the middle as we are presenting. If there are questions, please feel free to interrupt me as I'm presenting. Before we go and jump right into the guiding principles for PDEP and CLIN2, which is the next slide, we are just going to jump right into it. Those programs are the ones that move therapies from preclinical up to clinical development and hopefully commercialization eventually. But before walking through them, just a little bit of framing. The cell and gene therapy field has made real progress with the first generation of cell and gene therapies out in the market. We have CAR-T therapies, we have AAV therapies like Zolgensma and Luxturna, and we also have gene-modified cell therapies for sickle cell disease, right? And one of the things that we've learned, and Dr. Farmer was also talking about that, is that these therapies, we are learning now there are issues with access in terms of manufacturability and other issues that are precluding sometimes from maximizing the access to these therapies from patients. So we do need to take this into account, and that will be reflected in the guiding principles. And this comment came from the March 19 discussion in terms of making sure we don't conflate access and affordability. The access is very necessary, and we have to take it into account. So given what I just mentioned about the overall landscape of the cell and gene therapies, the guiding principles for the PDEP portfolio and the CLIN2 portfolio are very similar in scope, as you can see. We believe that overall, and that's what I was mentioning this morning, for the PDEP portfolio and the CLIN2, the therapies that we fund should offer should offer potential for transformative patient outcomes. And specifically on the PDEP side, they should be leveraging cutting-edge therapeutic technologies to serve that purpose of transforming these patient outcomes. Across the portfolio, we expect also that projects to also learn from this first generation of CGTs by taking practical stage-appropriate steps to address patient access barriers. That's the second guiding principles. And that means that they need to take deliberate stage-appropriate steps to address those barriers rather than treating them as an afterthought. Consistent with our patient access framework and review processes, this also includes choices for therapeutic design, development strategy, and clinical planning that may affect the ability of patients to ultimately receive these therapies. And lastly, the therapies, as the mandate of our proposition mandate makes it, they need to broadly address both prevalent and rare diseases affecting Californians. I'm going to stop here for a second in case that there are any comments or questions with regards to PDEP CLIN2. And I don't know if Mark, if you want to make any comments. Okay.
Yeah, please continue. That's excellent.
Okay.
Keith Yamamoto has a question.
Keith, Dr. Yamamoto.
You're on mute, Keith.
Sorry. On the third one, I wonder if you want to say broadly address prevalent or rare diseases affecting Californians. I don't know if you want to ask the applicants to come forward with ideas that would address both prevalent and rare.
Thank you, Dr. Yamamoto. Actually, the guiding principle refers to the portfolio. It's not so much on an individual application. So what the guiding principle is referring to is that CIRM, what we want to fund, we do not remove either or. We will fund both types of diseases because both types, rare and prevalent, do affect Californian population. And maybe I wasn't explicit. Sorry about that.
That's good. Thanks.
Oh, Dr. Dulesh.
Yeah, first of all, thank you. It's actually very interesting to have this dialogue, and thank you for all the work that went into that. Maybe can you outline what's different in what you're proposing versus the underlying philosophy that the CIRM always has had to fund cutting-edge science to improve access to patients? So what's specifically new in these principles?
I think it's the way that we are applying and implementing this. The philosophy was there, but I believe that when the rubber hits the road is in the way that we implement it through our program. So what's specific now is that, for example, at the volume control, as we will discuss, as Dr. Zambrano will explain, we have developed a selection criteria that derive directly from these guiding principles, and they will help the grants working group review delineate what will make it through to review, right, of all the awards. For the review criteria, we've always had the review criteria based on those guiding principles, and they haven't changed. We've refined a little bit because we are not adding, there was the whole nomenclature terminology between conflating access and affordability, and now it's been refined. And then at the ARS, and I mean, you just asked the key question, Anne-Marie, because it's what we are going to go through. But at the ARS, we are going to be more explicit and transparent in the way that we provide the recommendations to the ARS that hit those guiding principles. Is that, yeah? Does that answer your question? Okay. So I will go now to these four
So this 4 operates under a different structural logic. It is part of our discovery engine, which leads to elucidating disease mechanisms and seeding the field with new pathways, discovering new targets and biomarkers. So the guiding principles reflect that design. The DIS4 portfolio is intended to create multidisciplinary research approaches that integrate diverse sources of evidence, different, like computational biology, clinical data, animal model data. That's what we mean by that. That they have an innovative, that they innovate through collaboration, network synergy, and data leverage. And ultimately, the guiding principle also reflects the mandate of our proposition that has been materialized through the Neuro Task Force, and as well as other organizational priorities that we might find, and we left it as a more open thing there. These 4 principles are more about catalyzing the ecosystem level impact, which is a different type of approach. I will stop here because we are going to be switching gears a little bit to focus on ensuring that the guiding principles are applied to the right party with the right information and at the right time. So before I get there, any other questions? Am I going too slow? Feel free to tell me. Okay. Because it's the postprandial time and we all, I just want to make sure we all keep the conversation. Okay.
Just
so,
one thing about this. Will you at some point, or Gail, talk about removing the point-based preferences? Will you expand on this?
Well, slide number, can we, Harris, we've removed them. They are removed.
Okay.
Yes, so the slide number 3, I believe it was. Can we go back to, let me see, I'll just go here. So the slide says we propose removing point-based preferences. So what all this is about is instead of the preferences, we are going to utilize the guiding principles. And I think I probably went down too much into detail of what these guiding principles are. But now we are going to talk about how are those guiding principles applied at those 3 different steps. For the merit review and the funding decisions, they've always been applied. And we will revisit that, and Gil will go into detail of how we do this. For the volume control, I'm going to show it in a minute, how it changes. But thank you. I mean, you are on top of it, which is helpful because it makes me then have the discussion and yeah.
One question about, you mentioned the portfolio review. It might be helpful for us as a board over time to see what that looks like in 5 years, just something to think about,
Excellent. Yeah, that's an excellent question. And we are coming, thank you for asking that. I think it could be very good for us to come in June, as we were planning, and to frame it in the context of, if other members agree, to frame it in the context of this is what we have now, this is how we see it evolving in 5 years. And then the board can entertain like, well, maybe this means that we need to push something else or we should stop. It will help with framing things. So thank you for the question. To frame the proposal, we've clarified roles across 3 stages, as I was saying, volume control, merit review, and funding decisions, which we are going to be showing one by one. On this left column, we have who is going to apply the guiding principles, and hopefully this will answer Dr. Dulesh's question. How these guiding principles are applied through selection criteria, through merit review, or through recommendations, and what happens when we do that. So I'm going to go step by step. For the first one, the volume control, we are proposing a change from the preference-based approach that's currently driven by CIRM staff to one that's going to be led by the grants working group, who would select applications not based on preferences, but based on the selection criteria that are defined, and actually they are in the memo. Something very important that's reflected in the memo is how these scientific criteria are being developed, and they are derived from the guiding principles. And we can go in depth, and we could pull the memo if you want. At the next step, we have the merit review, which is the responsibility of the grants working group and has always been grounded in established review criteria aligned with the guiding principles. And finally, as I was mentioning, the final funding decisions continue to rest, as always, in the application review subcommittee of the ICOC. And as you can see, the guiding principles are applied directly into the criteria used at each one of the steps: selection criteria, review criteria, that has always been there, and the programmatic factors, which has been there, but it hasn't been streamlined, and it had not been as transparent as we are going to make it happen moving forward. Under all of this is the role of the CIRM team, which our role is to design and support the process, defining the criteria, assembling the right expertise, and making sure that each group has the information needed to make sound decisions. And this includes providing more transparent and structured information package to the board, including how each application aligns with the guiding principles, alongside with the grants working group recommendation. And with that overview, I will turn it over to my colleague, Dr. Zambrano, to walk through the details, particularly of the volume control.
Okay, thank you very much. And I also want to extend my thanks to the Science Subcommittee and board members that we have had conversations about this process. I think it has been very helpful in our thinking and in what we're proposing. Oh, thank you. Essential piece of equipment here. And so what I want to speak about today is the process that we would like to utilize for the volume control step. And as Rosa mentioned, the volume control step comes in immediately after application submission, but before the full grants working group review. And now this happens, and the reason we need to have it has also already been mentioned. We do get a large flood of applications across the many programs, and that has continued to increase over time. And over the years, we have utilized different mechanisms. So this is not the first time that we are dealing with this issue. We have dealt with it for many years. We've used pre-applications. We have even limited the number of applications that could be submitted by an institution, which of course did not work well. I think there are many reasons why that doesn't work. Nevertheless, we've tried different things. The latest was the use of the preferences as a method by which we could do the volume control. And so, as was also mentioned, we want to step away from those preferences and propose something different. In the end, the purpose of volume control ideally is to remove out of a pool of applications those that clearly are not going to be competitive, to enrich the pool for those that best align with our goals, and to allow a more rigorous review of that group that is most competitive. All right, so in going into what the proposed process looks like and the goals that we set out to achieve, the very first one is to refocus the selection of applications on the science rather than on strict preferences. We learned that the strict preferences did not allow for a nuanced assessment of the scientific value of applications, and so we really want to move away from those and back to a more scientific-based review. We want to align the programs across the board so that they are more consistent in terms of how the volume control process is implemented. This allows a better, an easier way of communicating it and understanding it from the perspective of applicants. And then we also want to leverage, and as you will see,
longstanding learnings from our positive selection process that was implemented in 2015, again, for the same purpose of volume control. And so what we propose is a modification of this. One of the things that we have done with positive selection is that the process has evolved. We have a continued collection of data and analysis to ensure that the process is working as we think it should. And where it's not, we try to make tweaks and improvements along the way. And as a result, I think having a unified process allows increased efficiency in that opportunity to learn and implement those improvements. All right, so this table is a just a depiction of what, for the 4 programs shown in this table, DISC 5, DISC 4, PDEV, and CLIN 2, what the current process is. And as you see, it varies. It's positive selection in one case, pre-submission in a couple, and what we call qualification in CLIN 2. And then also, importantly, who makes the selection is also variable. So in some cases, it's the grants working group. In some cases, it's CIRM or a combination of. And then lastly, the basis for that selection has been, in many cases, the objective preferences, but in other cases, it has been more the what we call scientific value or value proposition. Now, the variability makes it, as mentioned, hard to track and understand by applicants, and also very inefficient.
Yep.
So,
I'm sorry, Isabel Duron has her hand raised.
oh, okay. Sorry, Isabel.
Let me make sure yeah. Thanks, Gil. And should have perhaps asked Rosa earlier, because what I am not seeing in any way, shape, or form is any, when you talk about scientific value versus strict preferences, I am very concerned about where we have buried DEI. I know how hard you worked on that. I know how much we worked around this whole issue. And I'm very fearful that if we play language games here, that we're going to miss the total intent of making sure that there is representation from across this state within the science and within, the clinical trials, et cetera, et cetera. I know we talk, but part of me needs to see it on paper expressed somewhere, somehow, so that the scientific applicants themselves also know we are still very committed to that principle. And I'm not hearing it or seeing it. And I think as people keep praising CIRM for being the one good go-to place, I'm hoping we're not losing one of the most important criteria, and that was the turn towards DEI. So can you tell me where within all of this we are keeping our eye on the prize of DEI when points have been taken away, not being considered, et cetera, et cetera?
Right. So thank you, Isabel, for your question. So the elements of DEI continue to exist in what we call population impact. And so that is still a core criterion that exists throughout our different programs, and something that is evaluated by the grants working group as part of the score that they derive for the applications before it comes to the board. So those key elements are there. They will continue to be there. And so that, at least in this whole process, is where we currently have them.
I'll just say, Gil, I appreciate everybody's best intentions. I don't know that I feel better about it right now. Thank you.
Okay, understood. Thank you. So let me transition from this table that describes our current process to what we intend to do for the volume control step. We propose what we call GWG selection, just to distinguish it from the previously known positive selection, because it does deviate in terms of the methodology a little bit. and unifying that around who is making these selections. So it's the grants working group members that do the selection. And in all cases, it's based on what we're calling the scientific value. So that scientific value or the selection criteria, as Rosa mentioned, are defined in more detail in the memo because they differ from program to program. But they are aligned with the guiding principles, particularly the first often bullet of the guiding principles that is looking for the most impactful transformative projects, particularly for PDEV and CLIN 2, and those that offer a meaningful advance, collaboration and innovation for projects like DISC 5 and DISC 4. And so with that, I'm going to go over the process so that you get a sense of what this would look like. And so this is a process that we would apply to all programs that require volume control. And it all begins with establishing a target number. So for each of the different programs, we target a number of applications that allows us to have a full, robust discussion with the grants working group panel and ensure that each application is very well considered and we can bring therefore appropriate recommendations to the board. So the target number and then of course the number of submissions we get will determine the necessary parameters that we have to set up for how that selection occurs. Now, of course, if the target number is the same as the number of submissions we get, then there's no need for volume control. But of course, that is rarely the case these days. So in that process, which I will describe, scientific reviewers are going to do two things. They're going to select and rank applications based on the assessment of the scientific value that is appropriate for that specific program. And this is just a table that gives you a sense across these 4 different programs what the relative application pool might look like. So something like DISC 5 will often get, in the order of about 200, sometimes more applications. We did have a recent DISC 0, I believe, that had 350 submissions. So it is variable, but, in the range of the couple hundreds. PDEV may be around 75. We used only pre-submissions, so this is still a little bit of a guess. We had, in terms of pre-submissions, about 130 or so that we got. So for applications, it might be a little less. So when you look at the pools and the target that we would take to a full panel review, that helps us set up then the number of reviewers that we may need, the number of applications each reviewer needs to see in this selection process to ensure that each gets enough views, and the number of reviews that each application is going to receive. And so we're going to follow one of these. So we're going to take the PDEV as an example to follow this through. So let's imagine an application pool of about 75 applications where we're going to use 20 reviewers and we're targeting 30 to advance. And so in that scenario, we start with our pool of 75 applications. And so the first thing we do is we take the pool and split it into 20 overlapping batches of 15 each. And so with those 20 reviewers, each reviewer gets a batch of 15. And we're going to follow what happens to one application. So that application that's in red is going to appear in 4 different batches that 4 different reviewers will look at. And so each of those reviewers will look through their batch of 15 and they will select what they believe are their best 5 applications and then rank them. And so in this example, reviewer number 1 selected the application and ranked it number 1. The second reviewer also selected it and ranked it number 2. Reviewer 3 did not select it, so that one is still sitting in their pile between 6 and 15. And then reviewer 4 also picked it and ranked it number 1. All right, and so if we move forward with that, we take the values of selection and rank in order to create a composite score for that specific application. And so what the composite score does is that each application combines two measurements, the selection and the rank for that application. And we weight them a little differently. So let me first tell you what the selection and rank is measuring. So the selection ranks what fraction of the assigned reviewers pick the applications, and meaning, is it that multiple independent reviewers agree this application should be advanced? The rank measures the intensity. How much among the applications that they saw did they like this application compared to their other selections? And so we weight them 60-40%. We want to prioritize selection over rank because selection is more predictive of applications that do well in review over rank. So we create an equation from that. And then on the next slide, I will show you how that calculation happens. So for our example application that I showed you that had those 4 reviewers, based on their selection and rank, reviewer 1 selected it, and so the application gets a point for that selection. If it's not selected, like reviewer 3, then there is no point. So this is a binary calculation. They either get 1 point or no points for 4 reviewers. And then based on the rank, so the number 1 rank gets converted to points. Number 1 rank gets 5 points, number 2 gets 4, number 3 gets 3, and so on. And so we plug those into the equation so that we can get a composite score that's balanced 60-40%. And that if you do that calculation, that gives you a composite number of 0.73. And so what does that mean? For a composite score, the best possible composite score, meaning every reviewer selected it and every reviewer ranked it number 1, the best possible score is a 1. The worst possible score, nobody selected, therefore it didn't get ranked, is a 0. So the composite score will always be somewhere between 0 and 1. And so each application then gets its composite score. And if you look at the distribution of the composite scores from 0 to 1 across the example 75 applications, this is what it would look like. And so the distribution here and the examples that I'm showing you, we ran these through Monte Carlo simulations that allowed us to do many iterations of this. So about 100 runs for each of the different programs that we have, and in order to get the averages of where the cutoff would generally fall, and which fraction would advance and which ones are filtered out. And as you see the distribution, because what the reviewers are doing first as their step is a selection. They're first determining, out of these 15 I have, what are my top 5? So they're first picking their top third. And in doing so, the large fraction, the lower 2/3 is filtered out. And then they are ranking their then top selections in order to get a spread and establish a cutoff. And so that's why you see the distribution as it shows here.
Before you go on, can I just ask a quick
question?
I just wanted to know, why was it 60-40 the weight picked versus like 50-50? I'm just curious.
Yeah, so it is simply because we want to prioritize selection over the rank, because the selection is across different reviewers. So the more an application is selected, the more predictive it is of its success. The rank is really now limited only to that top fraction, and it's giving you more limited information. It's telling you only within the ones that they like, did it happen to be their number one, their number two, and number three. So it's really prioritizing the selection over the very specific rank.
So why not 75, 25? I mean, I'm just.
It could be.
Yeah, so
right.
this was just where to start.
So it's where we started, but it's also what the model suggested might be the best, but that can be tweaked. So, we could do a 75, 25, and try that out. Kim.
Gil, if we've stopped to take questions
Yeah,
briefly,
okay.
I also wondered how you selected the number of reviewers for the different programs. Was that also arbitrary or?
Well, so the number of reviewers that we select for the overall pool is not necessarily arbitrary. You can work within a range. You have to have enough reviewers where you get coverage of the entire pool, and that every application gets at least the 3, 4, or 5 views from those reviewers. What starts to happen if you have too many reviewers is that they don't get enough representation of the pool. So you want to balance it so that each reviewer has enough pool representation within their batches, but at the same time, you have enough reviewers that you have good overlap and are able to get sufficient number of reviews for each application.
But to get 5 people to look at each of, I can't remember which program, the CLIN
Yes.
program, right? You're talking about a lot of extra workload. Is that feasible?
Yes, because for that one, the pool number is not as big. And so the number, I mean, we want to start with a minimum of 15 reviewers. And so with that's easily achievable for CLIN.
But this is adding additional workload to the reviewers to do this upfront selection, right?
Yes, in some of the programs, but this is something that they've been doing for over 10 years in some of our other programs that have used positive selection.
Okay, thanks.
Mm-hmm. Yes.
Thank you for this overview, and I like the exercise you just walked us through, picking one application and walking it through. I guess my question has to do with what happens upstream to this stage. It strikes me that we are assuming that, I mean, I like the fact of being principled, but we're assuming that the 4 reviewers or 5 reviewers are interpreting the guiding principles the same way in terms of how they are ascribing scientific value. And it brings to me the question that Isabel had before, because when you described where DEI has landed, you actually use language that I think there may be reviewers who may not necessarily interpret scientific value as including population impact. So I'm wondering if maybe there needs to be something more explicit to ensure that we know that those reviewers are in fact applying the principles similarly. Do you see what I'm trying to say?
Yes. Yes, and those are things we worry about too. And so part of, addressing that and mitigating that is that, we have a lot of meetings with reviewers where we, do what is essentially training. We go over the review criteria, address questions to ensure that they understand them in similar ways, and know that there are 2 different tasks, right? So they're going to have the task of doing the selection and what the selection criteria are, how they differ from the review criteria that they would utilize in a full discussion and in the panel, and how to apply them. So that is an important element, because you want to make sure, to the extent possible, that all reviewers are looking at the applications in the same way in order to ensure that we're on the same page of why they're selecting them. because especially when you have so many applications that in general do have merit. From a scientific perspective, they may be interesting, they are good ideas, but not all good ideas are the ones that we want to have within our program. So it's how we define that very specific value to CIRM that we want to make sure they understand. So I think you're very right that that is a key element.
Gil, we have Board Member Yamamoto, followed by Dr. Levitt, and then Anne Marie. Gil, thank you for this. Can I ask you to go back to the table where you talked about the different mechanisms and the number of applications that come in and the goal to get down to a certain number? Is that, I'm sorry, yeah, this one. So first of all, you said that, so I have two questions about this. The first is that you told us that you would apply the volume control to the mechanisms that require it. That is where the number of the applicant pool is so large that you really need to cut down to some target number, which you have here. But now you've shown us 4 mechanisms in which the application pool ranges over an order of magnitude. But for each of them, it looks like you're still applying this volume control mechanism, if I'm understanding the second line, the target for advancement. Is that true? Is the goal to actually apply the volume control mechanisms to every mechanism, no matter what the number of applications that comes in? I mean, if you have 20 applications coming in, why not just review them then?
Right. So if for example, if we continue to follow the PDEV, if the target is 30,
yes,
and the application pool that comes in is 30, then there is no volume control. Then there's no need to go through this step. But if it exceeds that, if it's
right,
so like in this example, 75, then yes, then we would implement it.
But what determines the target for advancement? I thought it was the capacity of reviewers that you have to.
It is how we conduct the review. And so why is it so different, say, between CLIN 2 and DISC 5? Well, it's because the nature of the review and the number of reviewers that we assign to applications in the full review is very different. The content of the applications differs greatly. And for example, with CLIN 2, we bring disease area experts, regulatory experts, CMC. We have the access and affordability reviews. So the reviews are much more involved and take more time. And so in order to allow for a full rigorous discussion, the target number of about 10 is what we think allows for a robust discussion. Whereas I think for a DISC 5, we can do as many as 50 with that type of application.
Okay, good, thank you. So my second question, so thanks for that. My second question is, how is the composite number used to arrive at the target, or to reach the target for advancement? So
yeah, so I have more slides in which I can show you some of that, but I mean, in brief, so the way we did these simulations, they include that target number in terms of what we would want, where we would want to do the cutoff. So in most cases, the composite number ends up being somewhere between 0.3 and 0.4, where this distribution lands where the 30th application falls in that gray zone area. And so I will show you a table that shows you the rank order of how then we would determine that cutoff.
Okay, so the goal of when you talk about volume control, what you're talking about is arriving at a reasonable number of applications that will be discussed, not arriving at a reasonable number of applications that will be reviewed. So all of the 200 applications get a full review, but
No.
only a certain number of them will be discussed. Is that correct?
No. So the selection process is not the same as a full grants working group review. It is a triage step, basically, that where you look at the applications only at a very high level. You're looking at them from what are the selection criteria, not the review criteria. So the selection criteria in these types of programs are going to be, is this a transformative project that is going to have a meaningful impact to patients? And so looking at that, not looking at what is their plan and design, not looking at, do they have the preliminary data and appropriate rationale, not looking at those details. It is looking at, what we feel is the most important and aligned with the guiding principles. Is this something that's worth funding to start off with before you dive into, the detail? And if it is, then we can, dive in deeper and discuss, does the plan and design, does the team and all that support this idea that we determined was a valuable one?
So are the applications set up in such a way that the reviewers can do a scan of some summary level thing that focuses on the guiding principles and make some assessment? I mean, if I was a reviewer, I would feel like it would be my responsibility to look at these things pretty carefully, and knowing what the consequences of my decision is. So I'm just wondering, it just seems like you may be asking the reviewers, unless you provide them with some page that focuses on the guiding principles that the reviewer can look at without plowing through the whole application. Then I would think many reviewers would feel it's their responsibility, in fairness to the applicant, to look at the application very carefully, and then you'd be in a situation where every application is being reviewed, where that doesn't really fit with the volume control principle.
Right. So there are the applications, as mentioned, are each kind of tailored to the different type of program. For the DISC 5, because we have had a long-standing use of the positive selection process, include a preview page, which is a summary of their overall aims, what the goals are, and what the ultimate impact of their project, if successful, would achieve. And so there is sort of that kind of front page preview that that reviewers can focus on, but have the full application available, and which they often utilize in order to confirm or to reference any of the elements that they feel are important to assessing whether this presents, a good scientific value. For the other applications, like the clinical, there are clear sections that are focused specifically on the value proposition of the proposal, that summarizes, the key elements of what this proposed therapy intends to do, what the completion of the trial would achieve, and of course the long-term impact of the proposed therapy should it be successful. So those elements are, easily pointed to in the application. And we do that as part of our training with reviewers in terms of advising of where to find the information and how to best access it. But we also don't want to necessarily limit where they look, because some of that information can be found in different parts or elements of the application, as some reviewers have pointed out to us. So it is something where, providing the application as a whole gives them the opportunity to look where necessary to feel comfortable that they are making an appropriate selection.
Okay. Well, we can talk about this more as you go through this, but I have concerns, and we'll get back to that.
Okay.
Whether the capacity to cut down 200 applications to 50 based on a relatively cursory assessment, where you're making a claim that you're making judgments based on scientific merit. So, but let's go ahead. We can get back to that. Thanks, Keith. Pat's next, followed by Anne Marie and then Christine.
Yeah. So we've had lots of discussions about this, hours. First, I would say having a single page summary addressing the guiding principles is not going to be any better than a specific aims page that has that always has to have a rationale section for the specific aims that are being proposed. The second is that the guiding principles don't go away with the review. They're still part of the review, which includes access and accessibility, which is really now emphasized. When it was combined with affordability, at least from my perspective, it got diluted. Access is really something that the investigators can address in relevant ways that include the diversity and equitability of access. We've had discussions about how it was so difficult for a stem cell biologist doing transplants into spina bifida physician would be able to address affordability when policymakers and health economists can't do it well. So access is a key to the principle. And if I understand what's been discussed, these are going to be communicated very clearly to applicants. And if they understand that the guiding principles are being used to make selections about finalists, and it's still going to be part of the application review process. If they don't address the guiding principles within the full application itself, it's not going to do well. So I don't see it going away and vanishing. The other is that it's not a terrible burden to read a specific aims page and abstract, which are all included in a full application, as well as a significance section, which really gives the reviewer a complete picture of why this is so important for CIRM to consider, independent of the details in the method section and the statistics and other things that are going to be evaluated in the full application. So I would argue that a single page, in addition to what they're already doing, or just having a single page, is not going to satisfy those who complain that there wasn't scientific merit for the initial process that was used for making selection. Finally, something that I learned along the way, even when I didn't know this when I joined the board, there's a limit to the number of reviewers on the GWG. You can't, one solution would be just to add more reviewers, right? But we can't. I guess that was the second to final thing. The final thing I would say, if you shift from 60-40 to 75 to 25, based on Gil's gray area, that's where the shifts would occur. It's unlikely, if you run Monte Carlo, to see shifts going into either the lower red area or the higher area. It's going to be within that range, and it's not going to make really a whole lot of difference. So we could quibble about 75-25, 80-20, 60-40, but I do agree with Gil that the selection itself should be the most important thing, because once you start ranking, you're ranking against 5. When you're selecting against the whole batch that you've been assigned. Thanks.
Thanks, Pat. Well, Christine, can I interrupt you for? Can I jump in front of you for just because I want to say one thing about what Pat talked about, and because it's what's worrying me.
You are really constrained, Gil. I mean, you're trying to work within a very difficult constraint, and that is a limited number of people on GWG. So I'll just remind people that when the NIH started its peer review system in 1946, it started with 10 study sections. And the way they dealt with volume control was to add more study sections. Now there's 240. So started with 10, and now there's 240 in order to manage the volume of applications that comes in. So that was their primary volume control system. And now they do a triage in which the goal of the triage is not to limit the number of applications that have to be read by a given reviewer, but to limit the number of applications that will be reviewed in the study section meeting. And that cutoff, that triage line is set at roughly 50%, not exactly, but roughly 50% of the applications are actually reviewed and are actually discussed. And that gives the study section the time to be able to put the kind of focus that's deserved into the applications that are judged as being in the upper half. But that kind of cutoff, that kind of triage line drawing, in my view, and I said this before, it's very hard to draw a triage line that is more than a thumbs up and thumbs down, more than a 50% line that says, no, this doesn't really do it. 50% is not so hard, but 75%, 80% that you're trying to do with some of these mechanisms, I think it's very hard. So just acknowledging, Gil, you're stuck with this constraint where you don't get to deal with volume control simply by adding more reviewers, and so now we're struggling, all of us together, are struggling to try to come up with a system that is science-based, as you said, but still allows the reviewers to be able to move through this large number of applications efficiently. And the criteria by which it's done, the structure of the application that allows them to do that efficiently in ways that, ideally all the reviewers will be looking at the same stuff. Not some reviewers feeling like they have to read the whole application, others that kind of pick and choose which parts they're going to focus on, and others that are just reading the abstract. You don't really want that, and I think that's the problem you're sort of stuck with.
Yeah, I'll just add, Keith, that the NIH has now gone to triaging 67%. The top third only get discussed at study section. Top third.
Yeah, I'm not a fan.
I'm not justifying it, but I'm not justifying it, but that's the name of the game. And the other thing is that for those who have done reviews on study sections, you know that each study section member has a different view on what innovation is and how they define it, what rigor is, et cetera, et cetera. So there's no way of getting around all this. And I agree with you that the CIRM team has a really challenging problem that is tough to get out of.
Yeah. Sorry, Christine.
Thank you, gentlemen.
Thank you. So 2 questions, Gil. One is about the total number of application that will go to the review, the grant working group. So far, it was my understanding of the current system that the CIRM team was looking at the application when they are reviewed and looking at basic completeness and overall completeness before they would send it to the grant working group.
Correct.
Will that still apply?
Yes, we will still look. Essentially, that's the eligibility stage, as we call it, ensure that the applications are complete, eligible, and so on. And so that's a process that we do in parallel, and certainly before they go to the panel.
Great, thank you. That's reassuring. Second, are there a list of guiding principles, and are they the same? I assume they will be different between a DISC application, 4 or 5, versus, let's say, a clinical application.
Yes. So they're each designed around the different programs. And again, I know we keep using the guiding principles. The guiding principles is the umbrella term that we use for what we want the portfolio to look like. And, so if you look at the first bullet in a lot of these is we want, something that's impactful, significant, transformative. And so that's going to align with also then the selection criteria that we would use at this step, as well as the review criteria. One of the other guiding principles is having a balance between the rare and prevalent diseases. That's not one that you would apply necessarily as a selection criteria or as a review criteria. And that's for us, CIRM, to have a portfolio that includes all of those elements. So I just want to make sure we're careful in terms of like how we transition the guiding principles to each of the selection criteria and review criteria. The selection criteria are going to be really focused on, is this project, for those first bullets, the transformative, significant type of project that's going to make a difference and that's aligned with our goals.
Finally, Christina.
I'd like to speak in support of this process in its totality. Selection is hard, and we're faced, as I think Keith said, with difficult decisions. I'm involved in a number of organizations that are doing this process, and I can attest to the fact that what CIRM is proposing and the leadership is proposing is rigorous. The other thing, having served on the GWG, I don't know now, 5 years, I want to acknowledge that the staff Gil's group does very rigorous training of the reviewers. So it is top of mind what our priorities are. And from my perspective, in terms of the way the applications are constructed across our different mechanisms, it's very easy to figure out if the applicant who's put in the application understands what we're trying to achieve. And I'm sure all of us that have read grants know that there are people that do that really, really well and follow the directions and hit all the bullet points. And then there are other people that are completely off the page. I want to reassure Isabel and others that the DEI elements are still viewed as very, very important in CIRM's mission. And they're not called out DEI for all the reasons we know about in relationship to the federal landscape. But the applicants are asked to provide us with clear information about how they're viewing in terms of moving a product or a therapy into the clinical area, how they're viewing the unmet needs, the diversity of the population that has the problem that's under consideration, the economic value proposition in terms of what they're doing to support individuals to come and receive this therapy. I just want to speak of how rigorous this process is and how, as someone who's a nurse, has a background in clinical trials, but is there as an advocate for patients, every comment the patient advocates make about DEI are valued, and we're part of the discussion. And the last review that we did with PDEV, every application got a fair review in terms of the science, in terms of its potential to move into the clinic. Was it a value proposition? Was it something that society needed? And we had some applications that were for rare diseases. We had applications that were more for common conditions. Each one of them was valued and discussed. So I just want to speak highly in favor of this, even though it's a very, very hard thing that we have to do. It's the best we have at the time with the resources that we have. And I just want to applaud the staff for putting it together. And I think you have to trust that the GWG is well educated, well informed to make informative decisions. And virtually every meeting goes over because we spend time on the amount of time that applications need to give them a fair review. And I'll stop there.
Thank you, Christine. Marvin, you're next, and then Leandra will follow.
I just wanted to support everything that Christine just said, supporting this change as someone who has spent hours and hours reviewing. I have to say that the change on DEI to access and affordability, if the applicant gets it, they get it really well. And if they miss it, we know that they've missed it. So I think the process is fully supportive. Thank you.
Phew. Yeah, I did that for everyone. And that's an important breath because I feel very privileged here as a patient advocate to be surrounded with colleagues that have such deep and intense subject matter expertise. That's why we are here. And with that said, I think, we all bring our various perspectives on the subject matter at hand. We also have perspectives on the process, but we are a board and we have a very capable staff. And I do think that sometimes we can tend to dance on the line between expertise and process. Process falls within the hands and the purview of the staff. Of course, we as a board can comment on that if there are concerns, but I think we have reached a point where we have to move forward. And I am very comfortable with the process that have been put forth. I feel like we've had rigorous debate. And I also trust and I want to honor my colleagues that spend hours and hours and hours poring over these applications and trying to do what's right. So those are my comments based on what I've observed and how I'd like to move forward.
Thank you. Gil, I don't see any other hands raised.
All right. Thank you very much for the comments and questions. So let me just go over the next few slides, which address a particular element that came up at the last science subcommittee of our use of selection with ranking versus using selection with scoring and whether that, may make a difference in the outcomes. And so we looked at this and ran some Monte Carlo simulations utilizing both rank and score. And so just to show you what the potential differences are, I'm going to walk you through some of these slides. So this is an expansion of the slide I previously showed you where selection means a binary decision, yes or no, and therefore a point or no point. That would remain. And then the rank, as I also showed you, gets converted to points. Well, if you look at a score instead of a rank, and if you have a scale of 1 to 5, that can mirror almost exactly a rank because you are converting things to points or a score. In this table, I'm just showing you the assumption that the score is going to be equivalent to the rank. It doesn't have to be. And one of the things with a score is that it allows you to score many applications the same, or a rank forces you to separate them. We looked at a different scale. So like 1 to 10 gives you more granularity in terms of what the outcome may be in terms of assigning an application some differences. So in this example, the top and the bottom row where reviewer 1 and reviewer 4 gave that application either a score of 5 or it got 5 points because it was ranked number 1 in their pile. There may be differences that a greater scale could offer of, distinguishing it as a 10 versus a 9 in that particular case. And of course, there can be more than that. And oh, wait, no, didn't finish making my point, sorry. And then at the bottom, you see what the composite score would look like for all of these. And they're, pretty much in the same ballpark. If you look at an example here of 5 applications that are ranked or scored by a given reviewer. You see applications A, B, C, D, E. And so in the ranking, they are ranked and they are forced to be ranked and given a unique value. So 1, 2, 3, 4, 5. But if you score, you can get a more variable outcome in terms of what a reviewer may decide to score these applications. Because remember, these tend to be, in the example of 5 out of 15, these are going to be limited to the top third of applications that they're looking at. So by virtue of that, the range of scores that they're going to utilize are going to tend towards the top because it's going to compress the scale. And so it is likely that you are going to get more assignments of the same score to multiple applications. And so I identified and kind of just read, the possibility that you could get, multiple scores of 5 or 4 in a scale of 1 to 5, or multiple 8s and 10s and so on in a scale of 1 to 10. And so what does all that mean? Well, it means that you have the potential for having more ties. The possibility is that you may end up getting more clumping of scores. But when we ran the distributions across this, they still look largely the same. And although there is the possibility of having some clumping here or there, it didn't necessarily pan out in all of the iterations that we did. But of course, these are just simulations. They're not actual validated in practice session. So we don't know exactly what we will find in terms of in real practice, whether rank and score ultimately make a significant difference. But at least in the simulations, they seem to behave very much the same. And so, the summary of the analysis looking at ranking versus scoring suggests that we end up with similar composite scores, similar distributions, and gray zones or cutoff. And of course, key differences are that ranking forces separation of applications, while scoring can result in score compression, which could create more ties. Scoring, however, does provide comparability across batches, and that may be important, but it is difficult to know if when you see any difference, it's due to the batches actually being different from one another in terms of quality, or if it's differences in the reviewer themselves, in that one reviewer was more lenient, another one was more strict. And so that's not something that you can assess. So when comparing these, I think the CIRM team in general prefers the use of ranking because we feel, at least at the onset to test this out, we think it'll provide consistent separation of scores, particularly at the cutoff. But like anything else, this is something that we want to continue to collect data on and make sure that what we're expecting in practice is working as we think. All right, so how do you establish the cutoff? So this is the table I had mentioned earlier. So the way we would establish the specific cutoff is we would set it at the closest numerical break where there are no ties to the target number of applications that would not exceed 10% more or fewer than the target number. And so in this example, you have the applications that ranked from 28 to 32, and you see that applications 29, 30, and 31 have the exact same composite score. Those are tied. And so we would look for where is the closest numerical break with no ties that occurs between 31 and 32. So that's where the cutoff would be established. And so anything that is 0.32 or greater advances, and everything else below that does not. Now, if for some reason, and again, the modeling suggests that it would be a rare occurrence if you can't establish this kind of cutoff, but if for some reason we cannot, then we will take the entire range that occurs, which is likely to be, say 5, 6 applications in an example like this, and randomize them in order to select the target number in order to avoid any specific selection bias in determining what advances. All right, so then the last step in the process is that once a cutoff is established, then the CIRM president, with recommendation from the CIRM staff, will examine the applications that fall below the cutoff and may advance a limited number of those applications that could represent a unique opportunity aligned with the goals of the particular cycle. And so this is, if you will, a sort of a rescue process that's built into this. Now, the other question that we addressed during the science subcommittee, which is a very important one, is the role of patient advocates in terms of this process. So although not involved directly in the volume control selection, what we want to do is have the role of the patient advocate strengthened at the GWG panel discussion. So for those applications that advance, one of the ideas that came through our ongoing discussions was to define and include a patient perspective score. And so this score would allow us to define some specific criteria that come from the patient perspective and utilize a score that then represents that we would bring to the board as part of the application. This is an element that we are still discussing with the patient advocate members that serve on the grants working group to better define it because it certainly needs more definition. And something that we want to bring back ideally in June to present to you as part of this package. And one of the other elements that we thought that would be important was to have a pre-discussion orientation that includes the patient advocates along with the grants working group chair to talk about how to leverage the respective expertise going into these reviews and making sure that the patient perspective is something that is included in the discussion when these applications are brought to the table. All right, so that's my slide. And I think, Rosa, it comes back to you.
So, can we discuss?
Yeah. Who's speaking?
Keith does.
Keith, there you are. Yes.
So I apologize, Gil. I'm still not clear what is the cutoff? What happens to the things that are above and below the cutoff? This is for further discussion to then decide on what gets funded or what will be recommended for funding.
Right. So the ones that are above the cutoff advance to then the full grants working group review. And so they are, assigned a full set of reviewers. So if it's clinical, for example, the full set of disease area experts, CMC, and so on, to have that full review and discussion. The ones that fall below the cutoff do not advance, and that's sort of the end for those applications.
Right. So this establishment, being above or below the cutoff is very critical, or getting the attention of the president if you're below the cutoff is really critical. It's really the ball game of whether you're going to be in the running for getting funded or not. And so that's why I think it's particularly important to sort of try to get this right. And so so when you say that a nice thing about ranking, and you're certainly correct, your nice thing about ranking is that you get good separation of applications. The problem is that separation is a false one. When you're ranking 5 applications that you've selected, the rankings by definition say that you believe that the quality of them, the scientific value of them, differs by 20% as you go from 1 to 2 to 3 to 4 to 5, that the number 1 is 20% better than the number 2 one. If you're scoring, right, that artificialness, you're liberated from that artificialness. And so a strong pool could have 5 selected applications in which 3 of them get a 1 and 2 of them get a 2. And in a weak pool, you could have, you know, the top, your top ranking one could get a 6. And so you're able to really insert an assessment of the qualities. The scientific value is what we're claiming that we're doing in this process. And I just think that, and so that when you ran the simulations and came up with the 2 processes, ranking and scoring, being relatively similar, it's because you essentially assigned the scores to reflect the ranks. But what I'm saying is that that's not, hopefully that wouldn't be true. There's no reviewer that would actually believe that the 5 that they've selected differ each from the other by 20%. That's not true. I mean, nobody would believe that. And so if you liberate the reviewers to actually ascribe what they think the relative scientific value of the applications is, then you get much more information. And as you said, you also gain interpool information. So I think the 2 are really not at all equivalent, and that the scoring is substantially superior. And I'm just, this may sound a lot like I'm quibbling over details, and I apologize to the group for that, but because establishing a priority above or below the cutoff is so critical, and it's really the ball game. I think we really want to try to get this right, and we owe it to the applicants who have done so much work preparing their applications to be able to do that.
Oh, sorry.
This is John Carruthers. I just want to say I support this process. In essence, you're taking a process that apply to the different programs and more standardizing it across the programs, and you're just using a slightly different way to get the cutoff for the final review. So you're taking 100 applications. You showed the distribution between the 2. They don't look horribly different. I know these were models, but I think the process will be the process. I am less worried about ordinal ranking because I think it helps differentiate eventually which ones are going to get funded, because we do have limited funding. It's always hard when you see scores that are all very tightly, even though that reflects the scientific rigor, and maybe there could be an option for 2 scores. But the ordinal ranking puts them in order based on the rigorous review on how someone's going to move forward based on the limitations of the resources that are have for that round. And so you have to differentiate it somehow. I guess the only thing that I would possibly agree with Keith on the ordinal versus score ranking is, how do you compare someone from round 1 to say round 5 in the clinical round when the rules have changed slightly? I'm less worried about the individual rounds, but that would be the only thing I would ask.
Mm-hmm.
Yeah, this has been a really useful discussion, and I do applaud the team for working very hard on this. At the risk of sort of trying to reinvent the wheel, did you consider a model just based on the yes or no? Is it in the top third? Because
yes,
when I was at the NSF, we had a similar problem with a very large application pressure and a need to volume control on the front end. And before I got there, they had a very similar process where people would pick their top, I think in this case it was 7, and then they had to rank them.
Mm-hmm.
And we were advised by the then director of the Social and Behavioral Sciences Directorate that it is actually very difficult from a social science perspective for people to put things in order. And in that case, it was for 7, but it was felt not to really add meaningfully to the process. And perhaps if you are only asking people to put them in the top third, you could actually have more reviewers do that and make that a sort of more robust number, and presumably still have a spread across a large number of reviewers.
So we did the modeling. I mean, so that's what we call sort of just a binary selection, which is similar to what we have done with positive selection. And so we've essentially done that kind of binary model. But what happens is that you end up with a very large gray area because the selection is limited in terms of its level of granularity. Adding rank or scoring, either one, gives you another level of granularity because you have another measure that you can use in order to distinguish application. And it allows that gray zone, that cutoff, to narrow significantly. And so it allows a better cutoff. But we did look at different approaches in terms of the modeling. Again, you know, a lot of this is in practice, what's going to happen. I think Keith may be absolutely right that scoring works better. You know, from our perspective, we think that ranking may work better. But, you know, until we do it, you know, we're not really going to know. So we want to collect that data, make sure that it's working as we expect. I also just want to clarify in terms of, you know, a couple of the points made. When we did the modeling, we did not assume that they would have unique scores. The scores were allowed to be, you know, the same, and that's why you end up getting the issue of score compression. And so it makes the use of the scale, it just really tightens it. You know, you know, for me, that is something that we want to be careful about. But again, it is looking at a nuanced element. It's a second measure, whether score or rank. I think they would both ultimately work. But, you know, I think at this point, ranking for us is what we prefer.
Yeah, I mean, I'm willing to support the proposal no matter what, but you might want to consult with a social scientist who works on voting preferences or something like that, just to make sure that you're not introducing an element that's not defensible from a human psychology standpoint.
Yeah, thank you.
Pat Levitt is on the line.
Yeah, I was going to say, when you, because I've been on NSF panels as well, and the gray area is very large. It's actually out of 4 bins, it occupies 2 bins when they define the bins as must fund, and then you have 2 bins where you're not sure which bin it should go in, 2 or 3, and then you have 4, you're not going to fund at all. So you have a very large number of applications that are sitting in the middle. So that, to me, is a problem, but that's how it's run at the National Science Foundation. The other thing I would say is I agree Keith's point that it will deal, scoring will deal with inter group you know, batch issues, but the problem with scoring has always been, and this is true at NIH and in every other organization that uses scoring, is that there's scoring compression. That is a social science fact. And you need a thin gray area here, and you need some way of differentiating, which the rank does. So I would really support trying the rank first and seeing what happens. Is your gray zone, as you predicted based on your Monte Carlo, and is there a clear break? And then if there's some issues, going to scoring is certainly worth a try. But I do think that reviewers tend to compress, and NIH has gone to extremes to try to get reviewers to use the whole range, and they never do. Congratulations, and maybe we're done with this.
Okay. Scott? We're taking a break.
If we're finished with this
Yeah,
portion.
Do you want to go to a point where you can pause?
This is
organically?
a point where we can pause.
Okay.
Okay.
He's done, in fact.
We will take a quick 5, and I know that means 10, bio break. And so please hurry back as soon as you can.
Thank you. Oh, that's so sweet. Thank you.
So let's rock and roll. So I'm now going to focus on how the guiding principles are applied in the last step in the application life cycles, which is the funding decisions which the application review subcommittee of the board makes. And so again, just to iterate, we are on that target, the green part right there. That's the funding decision. And historically, just for context, the CIRM team has provided recommendations to the ARS by providing an analysis, a programmatic analysis together with the grants working group summaries and the public comment. But ultimately, the ARS considers many different things in how it chooses to fund which applications. So what we are going to do now is propose a refinement. It's not a huge change. It's just a refinement on how the CIRM team recommendations assist the ARS in making these decisions, which is basically a more transparent way to provide programmatic recommendations based on the guiding principles. So this is the last stage. How do I apply guiding principles to the recommendations? So what does the CIRM team recommendations include? The first thing that they include are, so we're going to focus, just to focus the attention, the programmatic factors is where the guiding principles will go. But when we provide recommendations to the ARS of the board, we provide the available program budget. So sometimes, like today, we didn't have enough money to fund these 4. So we had to provide the budget available, the budget asked, and then the board could make their decisions. We also provide the grants working group a score and the comments, the summary of the comments from the grants working group. The programmatic factors, which is where the guiding principles will come into play at that portfolio level. And I'll go into specifics in the next slides. And then prior awardee performance, like if they have strong execution, that builds confidence, right? But if there are persistence delays, we might bring this up so that the board is aware that those things happen. And then any new information, scientific or administrative information that comes to the attention of some staff through direct communication with applicants or institutions or publications or other. So in the next slides, what I'm going to describe is how these guiding principles apply and are assessed and leveraged in the CIRM team recommendations at the programmatic level. So programmatic factors for PDEP and CLIN2. There are the 3 guiding principles that apply on how they are assessed. The first one is transformative clinical impact. The kind of information that we look at once there is a grants working group recommendation for us to provide to the board is that we compare that award to active awards and external landscape within the same indication. The second aspect is patient access. And there is a difference here between CLIN2 and PDEP. So for CLIN2, beyond providing any knowledge of the modality and route administration, which does have an effect on the patient, on how the patients will be able to access this, we also provide a summary of the access specialist reviewer comments. There will be access specialist reviewers in the grants working group of CLIN2. And then the disease representation. We compare to our active award portfolio by disease area. I think Ben Chow, sorry, I thought there was a board member that had the thing open. I have good eyesight, by the way. That's with glasses though. So the next one is the DIS4. It's not moving. Ahora. So for DIS4 has, sorry. So for these four, the three principles, the first one, the multidisciplinary research approach does not apply. We do not provide anything extra. There is an assessment that's already provided by the Grants Working Group Review Summary, and that's what the ARS will receive in terms of that principle. But then for the collaboration, network synergy, and data leverage, we are going to evaluate how the research approaches and the topic area compare to active DIS4 portfolio and pending awards to figure out if there are ways that by funding that award, we could be able to increase the network synergy, for example. So those kind of elements will be in consideration in that programmatic recommendation. And then the Neuro Task Force and other organizational priorities, we will, because as Kelly will describe, the DIS4 is open to all comers. We just have a focus area that will be in play at the selection criteria. So if there is one that's not within the annual focus area, but it makes it to review, and then we can only fund, again, 6 of 8, those that are not within the annual focus area, that would be a consideration. And then any additional considerations concerning evolving research landscape and funding needs. So finally, the CIRM team recommendation memo, this is what we will provide to the ARS for the meeting for consideration. And again, they can, it's up to the prerogative of the application review subcommittee to consider this. So we will include, and there is an example in the memo, a summary table and rationale for all the applications. Actually, we put recommendations there, but it's confusing. So all the applications that came in, we will have a summary table and the rationale, and we will have grants working group, whether they recommend it for funding or not, and then what the team, like if we disagree with that or not, right? And then based on all that, the analysis of programmatic factors for the minority reports, applications where the CIRM recommendation differs from grants working group, all of those will have an analysis of the factors that we just saw. And then any application, and this is a handful of an expression, but if we think about it, any application with a grants working group score higher than or tied with the lowest scoring CIRM recommended application. So let's imagine that, we had an 80 and there is an 83 that's not recommended, but we recommend for funding the 80 because maybe it has an access path that's amazing, and we think that it will make it, and it will be of a lot of value for patients. So we could actually be making anything that's over that 80 could be coming with the programmatic as well, because we need to explain to the board how they differ, and you need to have those details. So that's it. And now, other updates to funding programs. And I want to say something about Kelly. Dr. Shepherd, oh yeah, sorry.
It's okay. I wanted to make a comment about the ARS and the recommendations that are going to be coming and how those are coming, because I think that's wonderful. I think it is really important to have all of that information as our funding dollars get fewer and the demand stays the same and more things are recommended than we can fund. I think it's, I think how we approach the ARS is going to have to change a bit, or at least be considered differently, and how the presentations are done, and what information is given during the presentation versus what is in our boardable or board packets is also really important. And so I think it's something that Vito it has to figure out as chair of the ARS, is how we are going to approach the application review subcommittee.
You're absolutely right, Maria. We've just begun to have conversations on this. It is going to put a slightly, I expect, larger onus, a burden on members of the ARS to be, you've always been diligent, of course, in coming to these meetings, but an extra degree of diligence is going to be required because you're going to have a more powerful role in selecting applications for funding based on the information, the report cards, the review cards that are going to be coming from the team. So more homework will have to be done.
Is that something that we can start to discuss in June? What that process will be? I know this won't take effect. Will it start for the June meeting? No, it'll probably be September is when the first
The, f-
tranche of
the
applications.
first CLIN 2 will be in September because it's the April CLIN 2
So-
will be, yeah.
Great. So we should start talking about this in June so that by September we understand what the ask is.
I'll start talking today.
Madam Vice Chair, yeah. Okay, thank you. I just wanted to mention, we heard Jim DeClos today, the education, and the workforce development has been a set of STAR programs at CIRM. And I feel it would be remiss to not mention the role that Dr. Shepard has had in the development of our workforce education programs. She's also the lead for the Discovery program. So I just want to thank Kelly for all the work because for the longest time she was holding them on her own. Now she has a team doing that with her, but they are possible because of her efforts and her mentorship as well of those directors. So thank you, Kelly. And now come on over. You have these 4 focus.
Okay, yeah. Good afternoon, members of the board, members of the public. Oh, sorry, is that better? Well, good afternoon, everyone, members of our board, members of the public, CIRM team. It's my pleasure to come here before you today to propose to you the annual focus area for our next DIS4 funding opportunity. So this is just a quick reminder that when you approved the DIS4 concept last March, it included a rotating area focus. The first or pilot version of this program was our REMIND program, and it had a focus on neuropsychiatric and neurodevelopment programs. The second iteration was also a neural task force recommended area focus, which was neurological disorders. And the culmination of that round of funding is the slate of outstanding DISC4 applications that you approved this morning. And we were really thrilled to see the innovative science that came out of this initiative. So today we're here to talk about what the area focus will be for our next cycle that we would be bringing to you next March. So we've considered a lot of different possibilities on our team, consulting literature, and looking at the trends that we've been seeing in our discovery programs and kind of some of the opportunities that are out there. And what we have decided to propose as our first suggestion for the next area of focus can be described as immune tissue interactions or interplay in the context of disease and repair. So this is sort of a systems focus rather than a disease or tissue specific focus, because as you all know, the immune system plays a role in a lot of our tissues and processes in our body. It's not just about fighting infections. It makes contributions at all levels and stages of our health and our lifetime. And so this focus area is applicable to a broad spectrum of diseases that affect Californians. So that's what is meant to be depicted in that little figure we put together. It's showing that all these different organ systems, there are diseases that affect those organs that suffer when there are dysregulation of immune function. So it's applicable to a broad spectrum of diseases. It's a problem that is well suited for the DIS4 initiative. The DIS4, as you recall, is a large, multidisciplinary, team-based approach, taking advantage of cutting-edge new tools and omics-driven approaches. And so this is the perfect type of application of an investment to begin to unravel the complexity of how immune tissue interactions affect our development and how they go awry in situations of disease and how they contribute to our body's ability to repair itself after sickness or disease. This approach, because it is a complex and interdisciplinary and intersystems approach, it will enable identification of novel targets and biomarkers, for example, such as those that distinguish between maladaptive versus productive repair. This is particularly useful for identifying novel targets because you need to do your studies in a system where there are multiple different types of cell types working together to identify a potential pathway to target, which is something that would be missed in a system where you're only looking at one cell type for only cell autonomous defects. So there's possibilities to take advantage of these really new technologies to understand spatial omics in cells, cell-cell interactions, cell states, differences in cell states. Those are all open fields of investigation that might lead to new and interesting targets and biomarkers that could be of benefit and perhaps relevant in more than one disease context. This is also an area that really supports and takes advantage of the improved human stem cell and organoid models that a lot of our grantees are working on, building the next generation of assembloids that include immune cells in addition to the tissue cells of a particular organoid and other support cells. And finally, we think that focusing around systems like this will create strong opportunities for network synergy across funded programs. And indeed, some of the types of proposals we saw in our the cycle that you discussed this morning, we were seeing quite a few projects that were addressing neural immune interactions. And so those types of programs will find a home in this area of focuses as well. So this is our recommendation, and I'd be happy to take any questions if any of you have questions before we ask for your vote on this. Sounds like everybody.
Is that still in a cycle of one cycle per year, or will there be, is there any option? I know it's a lot of work and it depends on the money we have to have 2 cycles per year. I'm saying that because if people want to be given a chance to reapply based on some comments, 1 cycle for a year really is not practical. So I'm just curious.
Well, currently, the way our budget and request has been is it has been set for 1 cycle per year. And I know that we come and discuss budget with you every year, but this particular cycle is ready to, assuming we get an approval today, we'll be ready to post the PA in April and begin to start accepting the pre-submissions that would be due later this summer. So we would have that going before any discussions about this next cycle after that. But as Dr. Canet Aviles pointed out this morning, we also have another discovery opportunity called DISC-5, which supports similar types of methodological studies, just with smaller teams. And we're really hoping that some of the projects that might be suitable for these large multidisciplinary approaches could indeed also choose to participate for a subset of those activities under a future DISC-5, and they would still have the benefit, if receiving a DISC-5 grant, of interacting with and joining part of the network that we're building for our discovery program so that they can leverage data and build on each other's findings to the extent that we can leverage that.
Also, this did come through the science subcommittee, and everyone, and this isn't about 1 or 2 cycles, but primarily the focus recommendation was something that the science subcommittee readily endorsed and thought was a great idea. So thanks, Kelly. The question about 2 cycles versus 1 would be something Rosa can address in further detail. Anne-Marie.
Come up here.
Thank you, Dr. Dilesh. So thanks. So one of this program, the DIS4, was modeled after those public-private partnerships, like the Accelerated Medicines Partnerships. Those programs are actually multidisciplinary collaboration. They takes a long time to put together, review. As you saw, we get like 100 pre-applications, pre-submissions, right? And so the process is long. We can only do 1 a year, but also it was modeled through the NIH funding for these kind of programs is 80 million every 5 years. So we are actually having 84 every year, and that's why we complement with the DIS5, right? So there's a different nature to these kind of programs, and hopefully that helps also with the context. Thank you. Mm-hmm.
Kelly, Mark Fisher-Colbury's on the line.
Yes, thank you, Mark.
Mark?
Hey, thank you, Kelly. I'm very excited about this opportunity here because this further expands upon our strategic direction with respect to looking at things as systems, and having that kind of a broad perspective has the opportunity to make dramatic improvements about knowledge and about how things work. And so with this kind of approach and with this particular topic area, I think there is incredible opportunities for acceleration of discovery. So thank you.
Thank you, Mark. Well, if there are no further questions, thank you for your attention. And I think we will have Dr. Shyam Patel present the final slide here. Or penultimate.
Good afternoon. I hope this is working. Yes. All right, great. Thank you. Good afternoon. I'm Shyam Patel, Associate Vice President of Preclinical Development at CIRM. I'm getting the ear signal from my colleague, so I'm going to try a little harder here. Okay, so before I begin, I do also want to second Rosa's comment about Dr. Kelly Shepard and the team's dedication to the EDUC programs. I can see on a daily basis how much heart they pour into that. And I've said it before and I'll say it again, on a weekly basis, we come across people who are alumni of those programs. To give you an example, Dr. Diana Farmer earlier today spoke about her program. She mentioned Dr. Aizun Wang as her collaborator. He was a CIRM scholar. She also mentioned that the one big advantage that they have is the GMP manufacturing facility at UC Davis. The director of that facility, Brian Fury, was one of the first Bridges scholars. And so not only are they being trained through these different institutions, they're going on to have careers that directly impact CIRM projects. Okay, so with that out of the way, we'll talk about the PDEV cycle recurrence. So just a reminder, the PDEV program is a preclinical development program. It's designed to take every single project that it funds to IND clearance. So regardless of what stage it's at, they get funding to get all the way to IND clearance. They could come in having just declared a candidate, or they could come in midstream of the pre-IND meeting or after their pre-IND meeting. So at any point along that spectrum, the translational valley of death, they can submit an application to CIRM, get funded, and get the funding they need to get all the way to IND clearance. So when we first designed this program and we brought it to the board for approval last year, we had incorporated 2 application cycles per fiscal year for that program. However, having gone through the exercise of doing those 2 cycles, we're realizing that the key benefits of having 2 cycles are actually not being realized for this particular program. And we are recommending that the board approve moving to 1 cycle. I'll explain that in a lot of excruciating detail because it's important. But I'll start off first by saying that a reason to have multiple cycles is that if you have some sort of a key readiness requirement, that the projects have the ability to apply when they are ready. In this particular instance, we deliberately designed this program to allow for entry at any stage across that preclinical spectrum. So we have projects who are applying midstream a pre-IND meeting, or they're applying right when they've declared a candidate. So in that sense, when an application due date is available, if a program is within that spectrum of preclinical development, it should be able to submit an application. So that was one aspect of what we considered here. Another aspect was if you have multiple cycles, what that usually does is advantages resubmission, so that you can get a close to a fundable score, or maybe you got a fundable score, you are not funded. You can come back in for a cycle that may be a couple months down the road. However, because of the way we have to line up these 2 cycles to fit within 1 fiscal year, the cycles have overlap. So somebody who submits in that first PDEV cycle actually can't apply in the second one. They would have to wait for the 3rd cycle. So in that sense, that resubmission benefit with multiple cycles is not fully realized with these 2 cycles. And lastly, the allowance of having 2 cycles allows from an administrative perspective to be able to batch applications. You presumably can get half the applications going through review and funding in that first cycle and the other half in the second cycle. However, what we're seeing right now is that because of the really high volume and the high scientific merit across the board, that we're exhausting a significant amount of the program budget in the first cycle. And that happened in the first cycle of PDEV, where we spent about 120 million, leaving about 40 million for the second cycle. And so in that sense, it's just going to incentivize applicants to want to submit that first cycle anyway, because they get a higher shot at a bigger budget that they can fund. So with all that in mind, plus considering a lot of other factors, we are recommending moving to 1 cycle per year. So what this does is that now, just to be clear, 1 cycle per year does not mean that the program budget is halved. The program budget stays the same. So 1 cycle, everybody applies. They all have access to the full program budget. Everybody competes at the same time for the full program budget. To do this, we have to have a higher volume of applications to be reviewed for that full program budget. So the number of applications reviewed will go up because we have to make sure that we can exhaust the entire budget. Any unfunded applications would have to resubmit in the next fiscal year application cycle. I know on paper it sounds really bad, but actually if you go through the whole cycle, the next application cycle is probably about 4 to 6 months away. And then lastly, this allows us to optimize the timelines for all the program updates and the application life cycles as well. Right now, one of the things that we were struggling with having 2 cycles was how do we line up the application lead time, the review lead time, and the ARS dates. And as you all know, we have an additional factor we have to worry about, which is that the ARS dates not have to line up with the in-person ICOCs, putting additional constraint. And to put 2 cycles in there, we actually had the issue where 1 application cycle overlapped across the entire holiday period. And so we had to make it much longer because we had to give applicants enough time to be able to submit an application and not worry about staff being on vacation or the universities closing down during the holiday period. At the same time, on the flip side, we had an issue where the applications were awarded by the ARS and the contracting period overlapped with the holiday period. So we had to extend the timeline for contracting those awards. So those types of things, in trying to make 2 application cycles fit into 1 fiscal year, we can avoid that by optimizing that 1 cycle. So ultimately with this 1 cycle, what we hope we can do is line it all up so the applicants have adequate time to make their application. The GWG has enough time to do the selection and the review, and we have the adequate urgency and appropriateness to make it to get it contracted. Ultimately, it helps us fund and launch the awards that the GWG recommends and the ARS approves in an optimal manner with a single cycle. Happy to take any questions on that if there are any.
Any questions from in the room? I don't see anyone. Oh, yes, Marguerite.
Hi, thank you so much for addressing this issue. As having just gone through the second PDEV session, it was really tough to see all those great applications and not very much money left. So I agree something really needs to be done. There was a letter that was submitted. I didn't quite understand how the concerns that were raised there would really, well, how this change would result in those concerns that writer mentioned. Does the CIRM team feel that there's any validity there in terms of the issues that were raised?
So that's a good point. So there are a couple of questions that were raised by applicants and awardees in the sense that if there are multiple application deadlines, they would be able to not have as much lag with a CIRM award ending and the next one starting. And, in that sense, we can't fully account for the financial situation or the award life cycles of every single awardee. It makes it, but at the same time, because we've designed this program to allow applicants to submit at any point in their cycle, as you've noticed, we've had several active TRAN awardees actually submit for a PDEV application. And so in that sense, there is a way to overcome some of that. But those are concerns that are valid from the applicants that, I'm going to have a CIRM award end, And if I have multiple deadlines, then, I can reduce the lag time to get funded. We recognize that, but we can't fully account for that in all circumstances.
Joyce.
Thank you for the presentation. I'm also inferring, and correct me if I'm wrong, that as we are getting higher and higher volume of applications, and we are trying to therefore not burn out our reviewers, that this might also be good for well-being? You didn't put it, it wasn't one of the bullet points, but I would imagine everybody's talked about increased number of applications. Makes sense. Federal funds getting trickier and trickier. And we have human beings that we're relying on to review these applications. And so I think it's a valid thing if that is actually, that inference is correct.
Yes, to do it at a single cycle, we could ensure that the full program budget is being reviewed all at once and that we're deploying it. And so you have all the the GW has all the information to deploy that full budget, and so does the ARS. So, yeah.
And wellness.
Yes, wellness is important. Yes. Yes, and especially for the patient advocate reviewers who have to review across multiple programs.
100% supportive of this proposal.
Thank you.
Oh, thank you, Joyce. Anyone else in the room want to ask Sham questions? Anyone online? Or anyone in the public? Okay.
We do have one public comment on the phone.
Oh, there is one? Oh, I'm sorry, I didn't see. Now I see it.
Would you like to wait for the public comment until the motion is made?
There's no motion yet. So, Rosa, do you want to come up and lay this out as a motion to the board? And I presume it's going to be an omnibus motion for.
Thank you. And thank you, Sham. So to finalize, we have kind of an overall motion for all what we presented today. The CIRM staff requests, with the recommendation of the Science Subcommittee, for the board to approve the following. First, amendments to these 4 PDEP and CLIN2 concept plans that are also in the documents that we appended with this item. There are track changes on how the guiding principles apply to the objectives of the programs. The implementation of the proposed grants working group selection process in all programs requiring volume control. And then finally, the last 2 items that were just presented by Kelly.
We can't hear you. Can you hear us?
I can hear you. Are you ready for my public comment?
I don't think Rosa was done yet. I'm not hearing her.
Yeah, all the people offline are not, all the people who are calling in, I don't think can hear the main room.
We might want to all raise our hands to try to get their attention.
Try that.
I just texted Claudette too.
They may text it.
That's good because Claudette actually runs CIRM, so that's good.
Too busy having fun.
She was asking if we could hear now, but I said no.
Okay.
Yeah, it's good.
Oh, it's good.
Yes. Testing. Yeah.
Can it, yeah, we can hear you too.
Okay, send it. Try sending it to this.
Yeah. Okay. No, I think the bandwidth.
Okay, I have
Kevin's
my
good.
own.
Yeah, I can hear you, Scott.
Yeah, we can hear.
Start sending it.
Testing.
Okay.
Can you hear? Good, good, good. Yes.
No,
it's here.
Yeah.
We can hear now.
Can you hear? No, the speaker. Yes, yes.
Yes.
Yeah.
Yes, now we can.
Yeah.
Dr. Carson, please go ahead with your question or comment.
Not good.
No.
It sounds like an echo chamber. Yes, now.
Good. This thing right here. Yeah. Give it a try.
They put 3 hours for this item. They were not kidding. Hello, testing, testing. Is anyone out there? Can you hear us?
Yes.
Yes, we can hear you.
Yes, we can.
Great job, baby people. Okay.
Okay, Monica Carson, long awaited. You are first up for a comment.
No, I was just trying to get original attention about the lack of sound.
Great.
Is there any other board comment? Is there any public comment?
I do see a hand raised. That now went away. Oh, now it's raised again. Okay, so the number ending in 3158. If you can unmute yourself by pressing *6, you'll have 3 minutes.
Great. Hi, this is Evan Snyder from SPP. Thanks for letting me voice my concern about the proposed reduction in frequency of PDEV grant cycles, which I fear will have unintended and undesired consequences for CIRM's mission and breadth of California scientists, clinicians, businesses, and patients who benefit from CIRM. I've uploaded a letter to ICOC, so I'll not read that. I understand the work and financial burden the old funding model presented. I'll use my time simply to suggest we figure out other creative solutions. CIRM was initially conceived to immunize Californians from the politically driven vagaries of Washington, D.C. That threat is no less now than it was then, probably worse. The principles and approaches, topics, and patient populations we hold dear are under attack, and the surge in applications to CIRM is a symptom of that. I fear that reducing the number of cycles will soften our core mission. Robust preclinical development has defined CIRM. While I appreciate the immense burden this emphasis places on CIRM, its menu and timeline of funding opportunities have so accurately been in tune with the tempo and dynamics of cutting-edge preclinical development. Such preclinical and clinical research, particularly in academia and small companies, cannot languish unfunded and unactualized for an entire year. I know we cannot go back to the old models. I would simply suggest, before we make such a change, that CIRM, in conjunction with other California scientists, clinicians, businesses, patients, rethink creative ways not to lead to some of the unintended consequences I fear. Some suggestions might be giving smaller dollar amounts per grant while maintaining frequency. Many PDEV projects are not ready to go the full 13 million. Early PDEV only is an option. Late PDEV only is an option, particularly if you've been in the CIRM pipeline. Combined early and late PDEV would be a minority of projects, mostly from pharma and hospital-based entities. A shorter merit-based LOI pre-selection process would help streamline things, shortening applications for reviewers to read to eliminate redundant sections or details appropriate only after a merit-based fundable score. Thanks for considering solutions other than reducing PDEV granting frequency.
Great. Thank you very much. The chair recognizes himself for a comment, which I haven't had a chance to do. I speak in support of all four components of this motion, and I'm going to just focus in on one to make a comment on volume control. I am, I think, the only ex officio member of the grants working group, along with all our patient advocates and our nurse representatives. But so I have experience of the process. Every one of those meetings I attend. So I agree that we have to impose a process that will allow our reviewers to assess scientific merit, but to do so in a reasonable format, given that the number of reviewers we have access to is by law limited. The proposed system in this motion gives the largest possible exposure to each application, gives the largest possible exposure of each application to the greatest number of reviewers without physically exhausting them or diluting their critical apparatus. I trust the grants working group to give great effort in reviewing all components of an application, and I trust their recommendations. Thank you. Scott, if there's no more comments, either public or board member, we can proceed.
All right, and for the record, Don Taylor, alternate for Kim Barrett, has joined the meeting. So all those in the room in favor say aye.
Aye.
Those opposed say nay. Any abstentions? And I'll pull the members on the Zoom. Monica Carson.
Aye.
Judy Chow.
Aye.
Isabel Daron.
Yes.
Mark Fisher Colbury.
Yes.
Judy Gasson.
Yes.
Jeff Golden.
Yes.
Rich Lahara.
Yes.
Pat Levitt.
Yes.
Hala Madana.
Yes.
Chris Miaskowski.
Yes.
Marv Southard. Marv, could you give us your
Yes,
answer one more
again.
time? Perfect, thank you. Kevin Zhu
Aye.
and Keith Yamamoto.
No.
Thank you. The motion carries.
Okay. Thank you very much. And thank you, Rosa, not just for today's presentation. We now have an update, agenda item number 13 from the communications team led by our Director of Communications, Amy Adams. Amy, there you are.
Okay, so I want to start by saying you don't have to vote on anything in what I'm about to say, so you can all sit back. There's some popcorn in the back of the room. I'm going to try to keep this easy and hopefully short because I think I'm all that stands between you and the traffic. Okay, let's start. First of all, thank you, board members, members of the CIRM team, and importantly, any members of the public who are on the call. Okay, I always like to start my presentation with a reminder of our mission. Obviously, we all know what our mission is, but it is front and center in everything we do in communications. And the way that the communications and outreach team helps move that mission forward is by driving awareness of and support for CIRM's progress and accomplishments. A reminder, so my communication and outreach strategy has 4 pillars. And as with most of my presentations, we are going to go through these 4 pillars and talk about what we're doing within each. So we're going to start with creating the story. And this is a reminder, I'm taking us all the way back, way back to December, when I presented to all of you my strategy. So what I'm going to go through today is for each of the pillars, what I said I was going to do within that pillar in the strategy. And then we're going to talk about what we've started to do. Obviously, that was December. It is now March. We have not done a lot on the strategy, but we are making progress in several places. So you'll see for every, this is the actual slide you saw in December, under what we'll do, there's a handful of tactics. The green check mark, I think we all know what that means. The little purple thing, that means we're working on it. And the red X, well, that'll be the next board meeting. So this will be the format for every pillar. So first of all, within creating the story, this is the one place where I think we've really started to make progress already. You've already seen a version of the CIRM story presented by JT. And we're starting to develop many versions of that. Obviously, one story doesn't work for all audiences. The one you saw, I think, was really compelling and emotional, but it's not right for everyone. So we're starting to build out different versions of that story and making it easy for everyone to have resources they need to tell CIRM's story. And I'm going to show a couple of examples. We're creating a new deck, some new deck materials, and we're trying to make it easier for people within CIRM to have access to really visual and emotional slides, even for when they're talking about the science. And I'm just going to kind of go through a few just nice examples of ways that I think we can be looking at our story. Obviously, we do science as well as patients, so we've got some great science images as well. And I'm just going to create the story there and move on to some of the other stuff we're doing. So tell the story. What I told you would do is a whole bunch of things. One of them I'm going to talk about is make the website fully accessible. So we have an accessible website. We did that. Actually, the previous communications team did that. And I think they should be really proud of it. There are some documents on the website that were not yet accessible, and we made them accessible. So this is the kind of thing that's happening behind the scenes, but that I think is incredibly important as a public-facing organization, that we be accessible to all people who might want to access information. The 2 things I'm really going to talk about today is some of our expanded written and video storytelling. Well, I'm not going to talk about video. And a storytelling toolkit that we're trying to build out to help people talk about the organization. So here's some of the storytelling that we're doing. The 2 closer to cures images there, that should look familiar. Those are the closer to cures that you heard at the last board meeting. So these are, as we all see, this is a really fantastic content, and we're taking advantage of it. So we've written stories about each of the closer to cures. We've got the videos from those are embedded in the story. We're rolling them out over social media. And I will say they are doing extraordinarily well across platforms. I don't think it hurts that these scientists are well known and liked and are promoting it, and their organizations are doing the same. So I think it really helps get our word out when we're also able to take advantage of other organizations that have a great presence. We're kind of piggybacking on some giants here. And then that beautiful picture, I avoid right, left. The beautiful picture on the right. So this is a family that participated in one of the CIRM-funded trials. It's just an incredible story. The older girls were both they were born with a disease called LAD1. It's an immune disorder. Parents didn't know that. They were just getting sick a lot. And the parents finally got these girls diagnosed with this incredibly rare disease that has no cure, aside from a bone marrow transplant, which is not an easy road. The mother was pregnant with that little boy there when they found that out. So the parents, this family came to the attention of our grantee down at UCLA that was running this trial, Don Cohn. He reached out to them, invited them to see if they were eligible. But then this family went to LA, participated in that trial, and here they are at Disneyland, like the world's germiest place. And these little kids born with an immune disorder are smiling with their little mouse ears on. So this story also did very well across channels because it's such a great story. I want to move on to deliver the story. You know, the thing is, telling the story is one thing. It's great. So we wrote a great story. Yay, really cute kids, and it's a great story. It doesn't do you any good unless you get that story to the people who need to see that story. And that's a whole separate part of a strategy. And that really is email lists and social media and leveraging our grantees and all of that work. So I separate those two out because I think they are two different activities. Within delivering the story, a handful of things that we're doing. One, I've mentioned previously, we've hired a PR firm, and I'm going to talk a little bit about some of the work they're doing. We have started training, I say advocates. In this case, the advocates are our internal CIRM team. So we're starting to train the CIRM team in how to use our messaging and other materials to be able to talk about CIRM. Well, one of the items on here is create a strategy for communicating about CIRM, which we did. So let's talk about the messaging training, because this was a lot of fun, I thought. So we invited everyone in CIRM to this training. And this is a screenshot from one of the trainings. There were about 60 attendees from within CIRM, which is a good portion of the organization. We broke people into teams, and they were given scenarios and told, here's your scenario, and what would you tell this person about CIRM? So there were 11 scenarios, and each team came up with multiple choice, many options of how they would talk to that person, because there were a lot of ideas about how we should be telling stories. This is step one. I think there's a lot of more steps to be done to help people talk about the organization, but it's one of the things we've been doing. And then I want to talk a bit about our, the PR team is just doing a fantastic job. So at the last board meeting, you all approved the concept plan for Rapid. The PR team pitched that pretty extensively. And I will say that Dr. Sean Patel did hard labor. He did a lot of interviews in between everything else he is also doing. So that was great. And then both JT and Rosa have done quite a few interviews on Rapid. I have a couple of examples here. Genetic Engineering and Biotechnology News, which is an audience or an outlet that reaches a lot of the people who we really want to reach, did a nice story. Stat did a story, and I've got a quote here from JT that's just right on message about who CIRM is and what we're trying to do. Also, CNBC Cures, which is focused on telling rare disease stories. CNBC Cures, their main vehicle is they put out a newsletter, and that's where most of their storytelling happens. And the top of the newsletter was a long story all about Rapid with some really nice quotes from Dr. Patel. I think JT is going to talk about an interview he did that hasn't published yet. Okay, and then I want to talk about outreach, and I've got a couple, I want to look at my notes because I've got some numbers here. So since the last board meeting, the outreach team, they did 10 outreach events, reaching about 250 people. And at those outreach events, 8 different students from our education programs participated in those events. Then in addition, 2 people from my team recently met with students at San Jose State University to train them in how to do outreach. So that reached an additional set of students. The images here, just to go through them, So you've got Dr. Joe Gold and Isabel Durone doing 2 different presentations at Rotary Clubs. And then what do we have? We have Aditi, Vice Chair Bonneville, and then Jeff Lomax here. And that was at the, what was that, Maria? The Alameda Health.
County Public Health.
Yeah, exactly. So these are just some examples of the kinds of outreach that are going on. And then I'm going to only briefly touch on measure the impact. So we measure things all the time. Here's the thing. We just started really, we just rolled out a new strategy. So all of our measurements are baseline. So I could stand here and give you baseline numbers, but they're not going to mean a lot. Some number of thousands of people saw a post on Facebook. So what I plan to do is start reporting out on measurements once we've got the baseline, and then we can report out increases over time. And with that, I thank you all. And I'll take questions. Or I'll let you all jump in your cars and get in traffic. No? All right.
Yeah.
Oh, Pat. Hi, Pat.
Hey, how are you?
I'm good, thank you.
Good.
You're coming from the sky.
Yeah, well, I'm getting over my cold with this meeting. It's not helping. That was just a joke. I love this meeting. The getting out the story is like really important, and I think I'd be interested at another time to hear about how to do this in a broader way, because presentations to small groups are important, particularly key groups. But for it to become viral, as you know, needs a different kind of strategies, and it's not easy, but I think there are mechanisms through that. So maybe I just want to make the comment that the thing that you emphasize, having a great story is wonderful, and except that it's a secret until it gets out there. And so great progress, really. And this next step, I think, would be really critical. I'm happy to talk about it with you between meetings.
Thank you, Pat. I agree completely. Yeah.
Thank you. Phenomenal work in progress. What's the biggest obstacle that you look forward to overcoming if you could just pick one?
Oh my gosh, that's a really hard question for late in the day. It's the size of the state, the size and diversity of the state. Actually, I'm going to take it back, because, and this, I'm saying the same thing, but I'm coming from a different angle. The fragmentation of the communications infrastructure right now. There was a period of time earlier in my career where you could put something on Facebook and people would see it. And now you have to and it takes a lot of work. You have to rewrite things each time. You have to put it on the website and on the blog and in an email and on Instagram and on Facebook and on whatever we're calling those other things now, and go to clubs and think about, Pat's exactly right. It's great to reach these high impact places, but how do you speak to larger audiences? It's really fragmented, and each one of those audiences requires someone to take the time to do that. So I think it's easy to say, time, money, people, but I think it's really the fragmentation that makes it hard for a reasonable. We have a reasonable staff, but it just makes it hard right now. And I think that's hard, not just for me, that is where we are right now in the communications landscape.
Other comments or questions? I'm going to thank you again, Amy, right? The highlights of our meeting. Listen, I owe you, the board, a very, very short chair's report, which I'm going to do, but realizing that I'm the only thing separating the board from hearing from our president, who has some late breaking news. So let me do this very quickly. This will take 2 minutes, JG. And it's very appropriate, board members, that I follow Amy's talk on how to tell CIRM's story. I would like to report to you on a new initiative I've undertaken with the aim of bringing knowledge of CIRM's work and accomplishments to a wider California audience. And by that, I mean audiences outside the typical scientific and biotechnology worlds. Capitalizing on some longstanding relationships that I have developed over the years, I've begun reaching out to some of the largest veteran organizations in the state. Many of these associations have large conventions each year that attract members from every corner of the state. As a matter of background, there are now presently about 1.6 million veterans in California, more than any other state. To advocate for them, Congress has chartered over 50 veteran service organizations, the most familiar being the VFW, Veterans of Foreign Wars, and the American Legion. What members of all these organizations have in common varies. Some attract members who served in a specific war, like Korea, Vietnam, or in my case, the 2 Gulf Wars. Others attract members of specific ethnic groups. Some require a distinguishing characteristic resulting from their combat experience, such as the Society for the Purple Heart, the Disabled Veterans of America. Paralyzed veterans have their own organization, as do those exposed to ionizing radiation from Hiroshima, Bikini Atoll, or the Nevada test sites. The Navy, the Marines, the Coast Guard, the other service branches have their own organizations, and so on. The 2 largest and oldest organizations, the American Legion and the VFW, have invited me to address their annual conventions this summer. Given that all veterans groups are chartered to foster their members' well-being and that one of their leading concerns is health, my offer to acquaint them with CIRM's work was seen as consonant with their national mission. One of the reasons I think that this is an ideal audience to educate is that, as a group, they are involved actively in the affairs of their communities. They are also among the most interested citizen groups in learning about advances in medicine and biological science, precisely because, as a group, they have been so disproportionately affected by war, whether by exposure to toxic environments, think Agent Orange, depleted uranium, and other carcinogens, or to blast injuries that caused chronic and traumatic encephalopathies. I'm expecting several hundred veterans and their auxiliaries to be in each of these audiences. And when it's done, they will return to the cities, towns, and hamlets in every county in the state. Take Alpine County, if you recall the map that Jeff Lomax showed earlier. Population of Alpine County is 1,200 people, but they too have an American Legion and a VFW post. So it's my hope that all of these veterans will carry some knowledge of CIRM and our mission and our successes to date back home with them. Thank you. If no questions or comments, I am eagerly awaiting the president's report. JT, you want to come up? Or are you going to stay there?
Hello, I'm one of the public members. Before we transition to...
No, we're not taking public comment right now. Thank you.
Mr. Chair, Madam Vice Chair, members of the board, members of the public, and members of our esteemed team, we have a very significant hot off the press announcement to make, which is that Rocket Pharmaceuticals, which is a CIRM awardee, just had their product for addressing severe leukocyte adhesion deficiency type 1, or LAD-1 or LAD-1 for short, approved by the FDA, just received clearance on their BLA. So this marks the first product that CIRM has helped fund that is now proceeding into commercialization. So this is a very large event for Rocket and a very large event for CIRM. And I would like just to read to you briefly from the FDA website on which this news is posted, the official word from the FDA. U.S. Food and Drug Administration today approved Kresladi, the first gene therapy for the treatment of LAD-1. Kresladi is indicated for the treatment of pediatric patients with severe leukocyte adhesion deficiency due to a variety of things that aren't worth getting into extreme detail here, but just to note that this is a very serious condition. The family that Amy was referring to, whose extremely cute picture was at Disneyland wearing all the mouse ears, was the family that she wrote about that had this condition. And so this is for that family and for others with this condition, a very major development. So I thought everybody would be most interested to hear that, because we've been waiting for our first ever BLA approved product to come out of CIRM. David, you hear that? Okay. Joe, do you know the answer to that question?
$5,867,085.
That's right. So that is the big news. We have other pieces of news that, because this sort of dwarfs everything else, I think we're going to keep in the interest of time for the next president's report, other than to mention two quick things. One is another patient story, which I think is very important for you all to hear, was interviewed by USA Today a number of weeks ago. And on that call was a family with two children that had SCID, one of which was 10 years before the other. That one, at that point, the only treatment available was a bone marrow transplant. And that child who had that treatment has had numerous issues, was in the hospital for, I believe, a year. The family went bankrupt as a result of dealing with the healthcare issues. It was a very, very difficult story to hear in the course of this interview, and your heart went out to the family. The second child, by the time that child was similarly diagnosed, which is terrible in and of itself, if you can imagine having two children with the same condition. And oh, by the way, the husband's uncle, I believe, had two other children that had SCID as well. But by the time the youngest of the two in this family that was being interviewed developed the condition, they were able to use Don Cohn's approach to SCID to get treatment at UCLA, and that child is now thriving and has none of the lingering side effects that their older child had. And it shows, first of all, it was great news to hear, but more than anything, that's the primary point. But certainly of great significance is the demonstration of this as the hope that cell and gene therapy presents to patients of all sorts of currently incurable disease. So that USA Today article is going to be coming out shortly. We will distribute when the time comes. The last thing I want to mention is just to close on a very positive note. I'd like to have everybody wish Jen Lewis of the Oakland Lewises and Dr. Liz Noblin a very happy birthday today. So, Mr. Chair, I'm going to close on that, and thank you, everybody.
Thank you. Thank you, JT. And where are you, Jen? Happy birthday. And Lana?
Liz.
Liz. Where? She left. Oh, no. Okay. Thank you. And JT, we'll look forward to the addendum to the report at a future date. We're at the point of the meeting where we ask.
We have a comment by Mark Fisher Colbury.
Oh, thank you.
This is incredibly exciting news about Rocket, and paralleling the conversation around SCID is the fact that there are a whole slew of very successful patient solutions and treatments that have been achieved through CIRM supported funding. And just wanted to expand upon that concept with the perspective of we've had a lot of successes, but we do want to ballyhoo and support a broader communication around Rocket and what's happening specifically around a BLA process. So thanks.
Thank you, Mark. Before we go to the public, are there any members of the board that want to make any comment on our ARS process or on any item that wasn't on the agenda? If not, I know we have a member of the public that's on the phone, so we can let them speak.
All right, the number ending in 3427, if you would unmute yourself, and you have 3 minutes.
Hello, my name is Parijat Bhatnagar. I would like to make, I would like to bring something important to your notice, to the notice of the board. I have brought this to your notice twice in the past, but I have been waiting for some action on your behalf. Basically, the issue is that, so I have started a new nonprofit. It has been about 6 months old, or about a year old, and one of the requirements, eligibility conditions for applying for CIRM grants have made us ineligible. And I'm sure that it was not intended to be like this, but I was told that somebody would get back to me. I have not been contacted about this so far. And basically, there is a requirement that the institute needs to have at least one W-2 employee. I have put in a letter to the board on the website, and I was hoping if we could have a discussion on how this could be addressed. We have all heard today that how important the funding from CIRM is in bringing groundbreaking treatments to the public, to the benefit of the public. And I believe that we should also get a chance to help California and with groundbreaking treatments, as well as increasing employment opportunities for those who would be helping us accomplish this goal. Thank you. I'm looking forward to a response.
Okay, we know who this is that's speaking and contact information? We do. Okay, thank you, Speaker. We've got your contact information and we'll look into it. I appreciate your remarks. I just want to let everyone know that the next meeting of the ICOC will be on Thursday, 25 June 2026 at the Hyatt Hotel in Burlingame, California. It will start at 9 in the morning, not at 8.